be really careful.
predator in that area.
Fishing gear.
But
and
on the seafloor, we also managed to see a number of deepwater coral species, as well as deepwater sponges and invertebrate life,
things like that come
from brittle stars, sea urchins, and even a few large marine gastropods or snails that were surprisingly large. They surprised a bunch of our scientists here on the ship. So today we're going to be having the same, more or less the same goal.
Northwest of us.
And we're just a few moments from the bottom now.
Just about 40 meters out.
I'll also point out, too, that we're joined today by a number of scientists on the shore, all over the world, for that matter. We have a number of means through which scientists on shore can chat with us and interface with us to accomplish a lot of our science objectives. We have run a chat system where they can write in specific comments to us that we will relay to everyone on shore. And there's also a conference line that you might hear other scientists' voice pop in now and again to provide some context about what we're seeing on the seafloor.
I will turn to starboard. And we will line up on
maybe just kind of center
315 somewhere in there.
Yeah, center on slope. Looks pretty good. All right, I am backing.
And if anyone is actually on the call, if you would like to introduce yourself.
Good morning. This is Kevin Rademacher with NOAA Fisheries in Pascagoula, Mississippi. I'm a research fisheries biologist. Interested in reef fishes and all the fishes in the Caribbean and the Gulf of Mexico.
Good morning.
Good morning.
Slopes my...
Okay, I've got you in Fisheye. I've got you in Zeus.
Copy that. There I am.
Okay, so if you hold there.
Anyone else on the line?
Copy. I'm going to do the same.
I selected the waypoint 335 is the waypoint.
Copy.
So it looks like we just
and uphill is
had some seafloor in sight on D2, and we'll give the pilots some time to orient themselves before we start our planned dive for today.
345, Sean.
So this site is actually bordering a marine protected area known as the Marine Conservation District, located south of St. Thomas. This area is known for its extensive coral coverage in the mesophotic depths between 30 to 150 meters. But it's also known for spawning aggregations of red hinds and also Nassau groupers. So hopefully we can see some groupers today and also some queen snappers since this area is pretty protected from fishing.
And coming down a little bit.
Bring it up to 50 degrees. And 45.
45, copy that.
And I'm going to come down to give you a little more tether.
Copy.
Guys go to the bailout south.
Bailout south, copy that.
Sounds good.
Bridges are RV nav. Yeah, vehicles are safely on the bottom. Let's set bailout due south, please. Thanks.
So the planned dive track for today takes us over a range of different seafloor topographies. We're going to be starting off in a shallower, we're going to be starting off in a bit of a shallower slope and proceeding up a rather sharp incline, rather sharp slope, which levels out at around 300,
right.
at around
I'm gonna get your swing arm set up.
I'll just
60 meters or so,
65.
465 meters. And around that depth, there's a small mound on top of the, towards the end of the dive track, we'll be investigating, as these features are suspected to be aggregation points for deep water fishes.
So this looks like it could be soft sediment of some sort. Few small fishes.
All right, watch leads. Do you have pilot?
Yes, yes, pilot.
Copy that. All right, as you can see, yeah, we're pretty much on bottom. We're just adjusting lighting and some last minute details, and we will start the dive in just a moment.
Sounds good.
The current seems to be pretty calm down here. Great.
All right, swing arms are set.
All right. I'm happy to Z-bias. And we can start. All right, watch leads. We're all set. If you see anything that you'd like views on, just let me know, and we'll slowly work our way upslope on the track.
Sounds great.
Copy video.
Thorian on the right.
Looks like there might be a small drop off right there.
Yeah.
If you have time, can we zoom on the sea cucumber
kind of
there?
Yeah, copy that. Video, give me 1 sec. Let me center. These are lasers. You kill lasers, Copilot.
Got it.
Get down there. All right, video, why don't we go for zoom in case he starts to float.
Oh, he's going to take off.
Oh no.
That's a good thing. They look cool swimming.
Ah, very nice.
So this is a sea cucumber or holothurian, and that we see quite often down at these depths.
What's really cool about sea urchins, if they are frightened or they feel at risk of predation, they could basically spill out their intestines and then regenerate it the next couple days. So hopefully we don't see that.
Depends on how scared they get, I guess.
Yeah.
This is some very nice,
Footage.
yeah, camera hog here.
All right, video, whenever you're happy, let me know. We'll move on.
Okay, we can move on.
All right, come down.
Okay.
Ah.
All right, you can come wide, video.
Those,
feel free to
those sea cucumbers are
pass
some
the
we
skids whenever you want.
see pretty regularly in this area. Throughout the Gulf of Mexico and the Northeast. In Nypniastes.
The slip is pretty good. I think it's a little more than I thought.
Is that?
Slip seems to be a little more than we thought.
Yeah, maybe it like gets deep and then flattens like shelves.
Yeah, it looks like it's a little more than just a flat bottom, which is a little more interesting. Looks like there might be a drop off ahead.
Well, do we want to go ahead and move the ship?
I think we can.
Yeah.
What direction would you like?
It's just upslope.
Upslope?
Yeah, I think
340.
Unless you want to cheat over to this interesting edge here.
yeah, we can cheat if you want a little bit.
So I'm noticing also some
left
stalks and stems, but
like
I think those are, they look very familiar. They're zoanthid covered
like make a move directly
old sponge stalks.
Why do we? I guess it's just a little
They're actually
there. It's not
really common.
much of an edge.
They look like corals, but they're not corals.
We do a 340.
Okay. Zoanthids are
30
a type
meters.
of cnidarian,
It's a Pashon.
similar and closely related to jellyfish, but they are
Test. 1, 2.
They don't have stinging cells like that of cnidarian, well, of
I
anemones
can always
and
stop
and
them
corals.
halfway through.
yeah, they're a little
We did 30 meters, John.
Ah, there's a squid.
Yeah, 30 sounds good.
Bridge, this is our we
Yeah,
have.
it's got video.
Yeah, like a 30-meter move, 30 meters.
So what are the
bearing
deep water
340
snappers eating in this area? Are they
speed decimal
eating
2.
squids like the ones we just saw, or they eat other fishes?
Yeah, so some of the fishermen, when they pull them out of the water, they still have the prey items in their mouths. So they found shrimp and small fish and also squids. So those are some possible prey items of these deep water snappers. Here's a fish.
Video and do a quick zoom.
Looks
on
like
fish.
a crinoid attached to
Some old animal stock, yeah. And looks like a cutthroat eel.
I'm going to come down a little bit and turn to starboard, or sorry, turn to port.
Turn to port, copy.
I'll have to turn back to starboard when we
Copy. Alright, video, you can go ahead and come wide. Start to push a little bit.
Can we zoom in on the lasers too? There's a stalk there that looks unlike the other ones.
Yeah,
just
copy
a
that.
snap.
Video snap zoom this.
Oh, there we go. It's our first coral for the dive. A sea pen. Something we would typically find in soft sediment habitats like this.
Is that all you want, Science? Just a quick snap?
Yeah, a quick snap
Copy that.
is fine.
All right,
I'm
video.
sure we can get some
Whenever
identifications
you're
on that.
happy. Nice.
So sea pen about 12 to
Something dead ahead.
13 centimeters in height.
And, quick zoom video.
Yes. Thanks, pilot. Oh,
Ah.
shrimp.
shrimp.
And some black corals here. These orange-looking spiral corals are black corals, probably in the genus Stichopathes. A coral I've seen a number of times, but I still don't have a species-level identification for. But the genus Stichopathes, most likely, and black corals known for their black skeleton underneath the tissue here.
There's a couple small sponges also, right on the edge of the ledge, right there.
And yeah, Science, as we pass over, if you want any really good in-depth views, just let me know, and we can take a little more time.
I think that's adequate now. Yeah.
Copy that. Moving on.
Yeah, some encrusting
wide.
sponges there. Hard to ID.
Okay, I'm going to turn my head back to 340.
Copy that. Turning starboard.
So just to give you an update, we are at a depth of 777 meters. The salinity is 34. PSUs, temperatures 7.4 Celsius,
We're
and
lined
oxygen
up.
2.87.
Tilting my camera down a bit.
Awesome.
Now I'm at 60 degrees.
You see a couple of brittle
Maybe
stars
5 meters
scattered on the
Right.
floor.
Copy. 5 meters left to move.
I'm going to turn off your auxiliary light.
Here's a small little fish.
Gonna snap this real quick.
Fish
again.
or shrimp.
Copy. Got a shrimp. You can come wide.
I remember seeing a photo of the stomach contents of some of these fishes, and they were full of shrimp. It looked
Yep.
like, yeah. So definitely.
Oop.
Oop.
Let's go.
Oh,
Yeah.
took
Move
off.
complete. Thanks,
Oh.
Bridge.
Oh,
Moves complete, pilots.
Copy. Move complete.
Is that a pretty good direction? 340?
I think it's
Yeah.
good.
Okay.
Yeah.
Looks like right up slope.
Great.
Should we just go ahead and put in another one?
Yeah. Slope looks nice and soft, so
Yeah.
I think we can move pretty quick here. I'm fine with the same move.
Sounds good.
If it works for your track.
Bridge is RV now.
Sponge upper right.
Yeah, I'd like to move same as the last one. Range 30 meters.
like a
Bearing
sponge.
340 degrees.
Fallen
Speed decimal
sponge.
0.
We're just about approaching a rather steep slope,
Good copy.
and we would expect to find many organisms that have fallen down and are stuck at the bottom of this rise. So this is a good
Video
indicator that
now.
there's a rather steep cliff coming up.
All right. Those are lasers.
kind of looks like a coconut.
Video, you
but
want
I
to
could
come
be
on
wrong.
in?
It's not the first time I've been fooled by a coconut,
Lasers
but
clear.
I think by the
Copy.
shape of the
Pilot, you got it.
spicules, I think
Yeah,
it's more of
it's
a
a
glass
sponge.
sponge of some
Yeah.
type.
It looks like a coconut.
Would make some nice habitat for a shrimp or something.
Yeah.
We see shrimp associated with deep water sponges all the time.
Max zoom.
Looks like some sort of
Copy that.
crustacean down there living in a shell underneath some type of crab, hermit crab,
Back up.
living in a shell.
All right, video, let me know when you're happy.
Video's clear.
Copy that. Science, you happy?
Happy in the back.
Yes.
Awesome. Come wide. Keep moving.
Yeah, Roland, do you have the settings? So I can latch bridge, and I think it's not supposed to be that way.
Sorry, Dan. I think we both clicked it
You know what I
at
mean?
the same time.
Is there a button on the pilot?
You don't have to do it now, I guess, but. Oh, okay.
But thank you.
So the ROV now position has the ability to latch bridge. And I don't think we should.
It's a lot of power for the nav.
Yeah.
All right,
There's
video.
another fish.
Quick zoom as it swims.
This could be a slender frost fish. Kevin, if you're on the line, if you can confirm this, or no?
Those lasers are about 10 centimeters.
Nice. Go for lasers. Awesome.
This one's not too shy. It's coming right up. Normally you get the back end of the fish to have to do the ID, but this one is providing a nice profile view. You heard me say that.
Looking down at you, 75 degrees.
Copy that.
Very prominent lateral line running down the body of the fish.
And a lot of these deep water fishes have these really big eyes because of the limited lights in these habitats.
Yep. We're also on the lookout for potential parasites on these fishes. There are several scientists ashore who are always interested in examining the fishes for isopods or other arthropods that might be parasitizing. Oh, another fish. Parasitizing.
Hey, this is Kevin.
Hi, Kevin.
Let's go for this fish.
Hey, good morning. I've got about a 15 to 20 second delay on my video, so when you guys talk, I've got to wait until I can see it. This fish is an ophidiform fish in the order Ophidiformes. It's one of those a cusk eel or something, but not quite familiar with this one, but it's definitely in that order.
Excellent. Thank you very much. And this one, I've seen this one enough. It's some sort of halosaur, I think. But that's usually the best I can do.
All right, video.
Down 90 degrees.
It's going to come out. I'm going to push a little bit. Cover some ground.
You are 21 meters into a 30-meter move, so he's got 10 meters left.
Nice. Copy that. Thank you, Nav.
Yep. You want me to range your sonar in, or do you like it where it's at?
Put it to 30.
Okay.
So definitely I see some sediment slumping here.
It's a wide-eye fish.
Well, those could be tracks in the sediment from fauna as well, but it wouldn't surprise me to see sediment falling downhill here because of the increase in slope. We've got Stichopathes, black coral, a couple of them attached to the hard substrate. You can see where part of the sediment has sloughed away and it's exposed some of the underlying rock. Oh, something. Shadow. I think it's another sea cucumber.
Oh, yeah.
Yep.
Move complete. Thanks, Bridge.
Another Enipniastes sea cucumber. Really big.
Thanks for fixing that, Roland.
How much left in that move, Levi?
These are
not much. I think it's it's complete. It's complete. Yeah.
These sea cucumbers are
They just
deposit feeders. They're feeding on the organic compounds in the sediment that are being degraded over time. A lot of the material that falls down from the surface ocean is the primary food source for a lot of the organisms down here, namely corals who feed on marine snow, and when the particulate organic matter reaches the bottom, the seafloor, it's sometimes vacuumed up by these deep sea cucumbers who are putting a lot of sediment and organic material through their gut and extracting out any nutrition that's possible.
And these sea cucumbers are actually closely related to sea urchins and the crinoids that we saw yesterday. They're all family members.
Looks like a shrimp.
Shrimp.
Yep, swimming shrimp.
There's actually a lot more shrimp on this dive than yesterday's dive that we saw, well, in the water column swimming.
Pilots?
Nav, what's going on?
You guys are going uphill.
Roger that.
Ship should just be complete, so...
Ship has completed.
Yeah, so this is what we're doing. Going up this hill, and then we're going to go up this hill. 10-meter contour, so you can tell it's not super steep, but it's a little slope. Right now, we just got you guys going uphill, 340.
Wonderful. I'm lined up 340. Delta 14.
Go ahead, Bridge.
Be happy. Give a good push.
Understood.
About 6
Yeah, as long as your center of rotation is safe.
Sí, está escuchando.
Great.
Saludos, especialmente en el Eco Exploratorio en San Juan. Saludos a todos. Buenos días. Y estamos aquí noreste de la isla Vieques y sur de St. Thomas, en profundidades de 761 metros. Y vamos a buscar habitats de pez, de chíos, en aguas profundas. Y vamos a buscar áreas con corales y esponjas.
If you're just tuning in, we'll introduce ourselves as well. My name is Steve Oskovich. I'm one of the co-science leads on this expedition. I'm a PhD candidate at Temple University in Philadelphia, Pennsylvania. And right next to me...
Hello, my name is Stacy Williams. I live in Puerto Rico.
Stay, what, 3 to 4?
I'm a co-founder of the Institute for Socio-Ecological Research and Coastal Survey Solutions. I'm a coral reef ecologist, and very excited to be on this expedition.
See your front
Hello,
toes. You don't even see your shadow yet, so you're still pretty high.
Well, I'll come down a little bit.
You can tilt up, and that'll help with that. Help you keep a little lower. Roland likes it about there in terms of lighting.
We're seeing some break in the seafloor, some structures.
Yeah, there you go. 2 meters. That's about good. Obviously, as it gets steeper, you'll have to do a little more.
I can zoom from here.
Yeah.
Yeah.
Sure. Oh, well, we'll get an image of this fish first, since it's being very photogenic. So this looks like some sort of midwater fish, potentially. I have no idea about any of these midwater fishes, so I'm going to have to rely on our shoreside scientists here. Although some pretty great imagery.
Swimming on me.
The pectoral fins are pretty.
Yeah,
it's crazy.
It almost looks like it's motionless in the water.
Yeah.
I'm slowly zooming out. He's getting close to you.
Copy that.
Can let him go.
All right, let it go.
Couple of shrimp behind him.
Copy that, Bridge.
Thank you, video. Oh yeah, he's right inside there. Okay.
All right. Well, we're gonna continue moving upslope, but in the meanwhile, we have a little bit of time. Pilot, this is Watchley. Do you have a moment to introduce yourselves in the front row?
Yes, we do. Yes, we do.
Whenever you feel ready.
Copy that. All right. So we need to speak on OKEX, right?
EXRV and OKEX.
Yeah, that's right.
Down there. You got that one. Yep, perfect.
Do you want me to start off? Or? Good morning. The front row has just changed shifts. I'd like to introduce ourselves now. Speaking is Lars Murphy at pilot. To my left
is Andy Lister at navigator.
Sitting co-pilot is Jeffrey Lanning.
In the far corner is Roland Bryant with video.
And in the back row, I'm Emily Narrow. I'm in the video clipping chair.
And we are all part of GFOE, the Global Foundation for Ocean Exploration. And we look forward to this next section of the dive.
Thank you, front row and back row, part of the back row. So we're just getting into a little bit more interesting topography here, where it's not fully sediment covered, but there are still some hard substrate for benthic critters to attach to.
085.
Thank you, video.
Although I'm not seeing very much at the moment. Occasional crinoid or black coral, stichopathes, whip.
Copy that.
Up there looks like some fish.
Yep, something shiny over there
something.
as well.
Yeah.
I'll make my way towards it.
Is that a shadow or is it just very big?
I think it may be a fish in a hole, I guess.
Yeah, the sediment around here is definitely very bioturbated. There's a lot of small, tiny mounds for infauna that live inside the sediment or burrow or use it as some sort of
That was my bad.
refuge.
And a lot of times when you're
She's not
looking at
coming
this
out.
fish, you can see their mouth forms. If their mouth is pointing downwards, usually these fish are feeding what's living on the seafloor, like shrimp or other small animals.
That was my fault.
So you could kind of get an idea of what this fish feed on by their body shape and the direction their mouth is
We
pointed.
didn't scare it.
For everyone shoreside, if you're in the chat room, I think we're trying to get the nav data tab up and working. It might not be displaying the depth and temperature and CTD data that you might be used to seeing, so we'll just reiterate it every few minutes or so.
Feel free to come in on that fish whenever you get it, video.
Oh, that's right. Yep, on the quad display on the ROV Sirius panel, you should be able to see the depth at least.
I'm gonna zoom from here.
But the depth is
If you'd like to.
756
If the
meters
lighting's
right
good.
now. As we zoom into this fish,
Have we seen this before today?
First
this is
time.
a new
No, this isn't
observation.
Okay.
So
Probably take
these
the lasers off.
tendrils that are hanging off the fish below, right there by his mouth, it probably uses them to sense anything that's living within the sand. So probably maybe some small shrimp or worms.
Copy that, Bridge. Just let us know when you're ready for us to put in a ship move.
So the depth again is
Oh, they do another
757
heading
meters.
change. Okay.
Temperature is about
After that.
7.64, 7.6 degrees C.
About 340. That looks good still.
And
more closer to it.
the
Yep, coming in.
oxygen
Yeah, they said
concentration
they're,
is about
they're still trying to sort the,
2.82
forces
milliliters
up
per
there.
liter.
Oh, I meant our head.
Our heading. Yeah.
Our move.
Gotcha.
340. It's
Yep,
good there.
I'll put it in. Another 30 meters.
Yeah.
Okay.
Sounds good.
Okay, I think we're satisfied with that imagery on the fish.
Okay.
Coming out, video.
You can see it.
Yeah,
You could
foraging
actually,
around.
see it kind of sensed in the bottom.
There was another dark spot over to the left behind this one. I
Okay.
believe. Would you like to head that way? or-
Yeah, if it's a good direction for
It's
you all.
straight ahead now.
Yep.
Steve, this is Kevin Rademacher. That fish with the barbels, the silvery fish, that's Polymyxia loei, Polymyxia loei, a beard
We have a
fish.
move underway.
There are two
Not yet.
species in the
Ship is
genus.
making a heading change right now.
It's the only
Got
genus
that.
in the
I'll just
family,
explore
and
here
it's
a
the
little
only
bit.
family in that order,
We've got space
Polymyxia
to
formes.
Excellent.
See if there's anything
Thank you very
on the
much.
inside.
Polymyxia, the
Come
beard
down,
fish.
give you a little.
There's a crab in the hole we're flying over below.
Is that of interest, or should we continue on to this one?
Just notify them.
There's a sea star right below me. On the
Looks
bottom
like
left as well.
some
Or something.
maybe looks like some corals.
We'll continue on. We see the crinoid, yeah, the sea star looking thing. But we'll continue on.
You can just do a sweep towards the starboard, and that would let you stay within your tether but see new stuff.
Copy that.
And once the ship's
do I have enough room to push ahead to this hole?
Possibly.
Right.
You might feel a tug.
I'll push ahead a little bit and see if I can swing to the port.
Something.
Seeing it, if it's even a hole at all.
Yep. There's a couple things on the top of the hole that we'd like to look at. One is a sponge, and just to the right, out of screen right now, looks like it could be a coral.
Copy that. Right there, that just came into view.
Yeah.
Right. Which one would you prefer first?
The one that's straight ahead first. I think the one on the right is actually the same sponge, but just not in great shape. Could be, yeah.
Do we want to do a quick zoom before we?
Yeah,
sure.
Sure, we'll check it out.
Right.
Not quite sure what that could be. It's blending in very well to the sediment.
Go ahead in video.
Maybe some cnidarian of some sort?
Could be, yeah. It's very tough to tell.
You're drifting back.
Or maybe a sponge.
My first thought was sponge debris, yeah. Okay, we're satisfied there. We'll back to the other sponge on top of the crest.
Got some light flares going on both sides of the camera.
Kind of looks like a glass sponge.
Yeah, for sure. Looks like a type of glass sponge, also known as a hexactinellid sponge.
Would you like to come in on that video?
Possibly a euplectellid sponge. Something that is I would hesitate to say common, but something that has been seen in this area previously, and I think it is. Euplectellid sponge, possibly in the genus Euplectella.
In general, yes.
I don't think we need to get closer. I think, yeah, we would be able to see if there were any shrimps or crabs inside of it. I don't quite see any right now.
Copy that. Come out and we'll go to the specimen on the right.
Yeah, I think we can skip that one. It looks like a sponge as well, but just in a worse shape. But
Copy
maybe
that.
a snap zoom at least.
Yep. Go ahead and come in video.
Yeah.
Yep.
At least this one has a crinoid on it, but the sponge itself doesn't look like it's in great shape.
So I'm sliding back, that's why.
Yeah, you're just got a little momentum backwards.
Gotcha.
We're satisfied, pilot, to move on.
Let's swing over to the starboard then.
Yeah, just keep this facing view and come around starboard and look around. And hopefully we'll be able to get a move in shortly.
Are they still making the heading change?
Yeah, they're still making heading change. They should be coming to an end though.
Copy that.
Looks like they've settled on 105.
Copy that, Bridge. How's it looking right now? Do you think that's going to be a good set heading? Copy that.
Can we pause for a move if it's safe?
Yes. Bridge, would you feel it's possible to possibly pause and put in a move because we're pretty stretched out here? Okay. Let me know when it's good to go.
Thanks, Nav.
So while the ship is trying to move here, we'll just take a look
you feel
around.
like putting in a little bit, like maybe a 40-meter move, just because? Okay.
It's definitely a lot of sediment here. Not much attached fauna.
Okay, Bridge, it's going to be 40 meters at 340 at 0.2 knots.
How is that for
Good
like
copy.
speed and, you know, is that?
Seems good to me.
What was
Video's
that
the
about?
guy. Ask him. Oh.
Roland, are you happy with this little drive-by?
Yeah, this is good. I'd like to get a little closer to the subjects, though, to get better lighting on them
Okay.
in general.
Definitely. Looks like we got a little hole here. We'll back up, take a look at it.
Some bent points, maybe.
Yeah. Some kind of burrow.
Or so.
See anything in there
Looks
that
like
you're
some
interested
small
in?
cup
Yeah.
organisms.
Or scientists?
Yeah. See a small cup coral.
Negative.
Great. And that move should be going in, right?
Yeah, I'll keep you in view for now.
They look very similar to the ones we were seeing yesterday.
Yeah.
Let's, if there's nothing of extreme interest here, I may just see, get a better feel for the vehicle. If I potentially touch down, we won't be obscuring any video feed, right? Or useful video feed.
It's all useful.
Well,
Just kidding.
I mean,
There's a big coral.
I
yeah.
think you'll get a good shot if you tilt the
So
camera up at this angle.
I don't-
see some stuff.
I don't even know if that's
lions.
much alive with the original organism that was there, but maybe we can take a snap zoom for attached things.
On this coral here?
Yeah.
Right.
I think there were some crinoids and some... I see a siderid urchin in the background, actually, also, too. Up here.
Yeah.
Go ahead and come in, video.
So this former coral is now fully covered in hydroids, another relative of cnidarians, or it is a cnidarian, but a relative of corals. But this coral actually looks to me like it may have at one point been a bamboo coral, because I see some banding, some discoloration.
If you want, I can try and get the base and the tip.
Yeah, it don't work too hard to get it. It's it's definitely dead, but if on the way out, you
Copy
can
that.
scan the base.
Coming up.
Might be easier to see if it was a bamboo coral. Yeah, and I would say so.
There's a crinoid. Is that a crinoid attached? Yeah.
Yep.
Gonna just follow it down to the sediment.
So this was a bamboo coral at one time, and you can see the dark nodes and the mineral
Starting to lose
white color looking internodes.
Copy that. All right, feel free to come out, video. I'm gonna make my way. Is that an urchin there?
That is an urchin, yeah.
Is that of interest?
If you have time, yeah.
We should have time, right? You still have my light?
Your light pool's right there.
Yeah. Come in quick, video.
This is a different species of what we saw yesterday.
Yep.
Coming back.
Not so long of spines. It's actually missing a couple spines.
Yeah.
That's
It
very
looks like.
interesting.
Yeah.
It has zoanthids growing on the spines.
Maxine.
Copy that.
I don't think I've seen that before.
These are also known as pencil sea urchins because their spines are so thick that they resemble pencils.
Right.
Time to play catch up.
And they're
If video and
also
science are
relatives
clear, I think we are.
Video's clear.
Right.
of the
Science,
crinoids
are you all
and sea
set?
cucumbers.
Yep.
Yes, yep.
Thank you.
All right,
Thank
feel
you.
free to come out, video, and let me run back out in front of Sirius.
And for all those who are joining us, welcome. Today we are exploring some of the deep water habitats of possible deep water snapper
Go uphill
species,
a little more.
like the queen snappers,
So
and also some
somewhere in
groupers.
that direction, yeah.
And also trying to
Trying to
find
come right
areas
out,
where
right
we can
underneath me.
locate deep water corals and sponges and other organisms. So far we've been seeing some echinoderms, crinoids, and sea stars, a couple sea cucumbers, and some black corals.
Got your glimpse there.
And previously living bamboo coral, but I'm optimistic that we'll probably see another individual from. Oh, actually, just out of the screen on the lower right, there was a large fish hiding in a crevice.
Straight back.
We're moving ahead of it
Probably
now, so
guilty
just
if you
due
tilt
south
down.
of the middle of the screen.
Coming back.
Looked like a
Get
roughy
it from the camera tilt.
looking fish, orange or reddish.
Yeah, just tilt down.
Tilt down.
Yep. Grab it.
I'm all the way down, I believe.
All right.
Just caught it out of the corner of my eye.
So you're up.
Oh, yep.
The one underneath.
Yep,
Yep.
that's it.
I'll hold there. Video if you want to come in on the on the fish. There's a shrimp as well.
Yep. So I think that's a type of roughy.
You're backing away a lot.
I'm backing out. Okay. I'll try and come in.
It has this really distinctive line right there on its
How's that for light video?
It's getting better.
Getting better. All right, I am getting
bottom
closer.
mouth parts.
The head is very platey,
Yeah.
Okay.
big eyes,
That's good
Oh,
there.
wow.
and I don't think I see any parasites on this particular individual.
Pulling out.
Coming back up. Coming over.
Beautiful fish.
How much we got left on that move, Nav?
Video's clear.
Steve, I'm just now getting some good video. Yes, that is one of the roughies. That's Jephna darwini.
I didn't get the time. So
Don't recall the common name off the top of my head right now.
it's done.
Excellent.
Copy that, Bridge.
The
If
fire
you want to
barracks.
continue your heading change, feel free.
All right.
Okay.
Time to catch up. Sorry.
Video time to come out.
I know I'm
Copy that.
going to misspell that, but I'll look it up later.
Copy that. Coming up and over.
Something like that.
Nope. Looks like another glass sponge
Do
ahead.
oh, so I got the Sirius camera. There we go.
So we're getting into a little bit more dramatic topography now. We started off the dive in a more flat, sedimented tongue that was sticking out of this particular wall.
Pretty stable, so.
And now we seem to be
come across the wall,
hitting that
come up
wall, and
try
the
to get
topography is
some,
rising
shots
very
and stuff on it.
fast. The bottom is rising very fast.
Up to that.
Although not a
should I
lot of
come up
evidence
until
of
I'm in your view
attached
first,
fauna,
or
which we
should
would
I just
expect
start
to
heading
find
over
in
towards
this
the,
area.
port?
Do you think it could be maybe
I
caused
would-
by all
I
the
would try to
sediment?
do both, kind of come up and
Could be
into it.
sediment.
You're way down over there.
Yep.
Could
I'm
be
actually turning my
some
heading
of
20
some other
degrees to
environmental
starboard, so
parameters.
you're way off to the starboard. So you sort of come forward and up.
Copy that.
Oxygen has decreased since
Yes.
we.
They ask you to take
There's
zooms.
another fish.
We're in a stable position, so.
Looks like the
Can,
fish that
oblige.
we,
Okay. Coming over
beard
to the
fish.
the
port.
beard fish.
Yeah. So we would expect to see more attached fauna in these hard substrate areas because there is more space to attach to, but at the same time, currents sometimes can be accelerated around these
Geez, I'm 10
rocky
meters.
features. and that brings things like corals and sponges,
Yeah, it's very steep
more
here,
food,
so.
greater food supply.
I just realized that as soon as I said it.
Looks like maybe a sea star in front of us.
Oh yeah, that is a sea star.
Still aiming
on the rock.
2 degrees to starboard.
Pilot, can you do a snapshot of
is that watch lead 2 that's talking?
Yes.
Okay, I got you. Sorry, I had you slightly muted. I got you on now.
Okay.
All right, feel free to come in video.
Yes. Yep. It's a sea star.
Yep.
How is that for light? Would you like me to get closer?
Yeah,
no, that's great.
Yes.
Okay. If you want to zoom out a little bit, I'll move in.
I mean, it's okay for what
I think this is
just in general.
one of the slime stars, but I
try to light it up.
Haven't seen sea stars like this in a while, so I could be mistaken.
Copy that. Thank you.
Why is it called a slime star? Produces slime.
I guess when they're handled, when they're sampled, they are capable of producing quite a bit of slime.
Okay.
Yep. It's right about there would be what I would consider optimal.
Okay. We get a spatial. Okay, excellent. Scientists, are you all set with this?
Yes.
I think so. Thank you.
Okay. Feel free to come all the way out, video. Unless
no, that was full zoom out.
Gotcha.
So yeah, when you're scanning, you're going to want to be back a little bit, not this close because you can't see anything.
Yeah.
You want to just be a little bit further back from this so you still have light, you can see plenty of objects.
And then when you see an item, go in.
When you see an item, go in and get a lot of light on it, and then take the zoom in.
Gotcha.
It's helpful if you get yourself stable when you're wide.
Y la profundidad ahora es 736 metros y salinidad es 34 PSU y temperatura es 8 grados de Celsius y oxígeno es 2.7.
Copy that. Right,
so
come back to
this is the second individual we've seen now, and the
yes.
right-hand side of the screen looks like a type
so,
of.
So, like in something like this, is it better to lateral port? Starboard? Yeah, port.
Yeah. I mean, so you don't want to be totally squared up and just looking right in front of you.
Yeah, so like a slight angle. Like,
yeah, exactly. You want to see what's coming.
Right.
So that way, when they see something come in on the left side of the screen and they say they want it, you're going to slow down, but then you're just going to turn your head and square up to it.
Go ahead, Bridge.
And that way, you're not going to dust everything up as you try to do that.
Gotcha.
It'll make your life easier.
Excellent. Thank you.
So that angle looks really good.
Something like that.
Keep that and come along.
So
and we kind of do a slant, like keep this angle, come across, and come up at sort of the same time. And that way you're covering ground, but you're also, we have a broad view here, so you're able to see a lot of the sort of just zigzag.
Also, this is a reminder for any of the land-based science team. Please feel free to annotate in situ anything that you see or you think would be interesting, would be of great help.
It looks like there's a shadow up above it as well from something a little bit bigger, maybe. I don't know if you can see anything on Sirius.
Shadow of...
You see like that V shadow, or that V shape?
Yeah, there's a coral sticking out there.
Oh, gotcha.
Yeah, or a sponge. Could be a fish.
It doesn't seem to be moving that much.
It's a sponge.
Oh, yeah. It's pretty transparent.
Okay, I'm going to bring my heading to port.
Copy that.
You're coming back in view. Line back up with 340.
What about these little white specks in front of us? Those shells?
They look like some kind of shell. Yeah, if you have a moment, a snap zoom, I think they're old, either brachiopod shells or some sort of mollusk.
Okay. Coming down on it?
I think they won't spend much time on it.
Yep,
so
just quick.
Quick, yeah. We don't have to delay too much. Might be difficult to get an ID on them anyway, but it doesn't look like a brachiopod. Probably some sort of mollusk. The shell looks rather rather robust.
Okay, feel free to come out video.
Looks like it is a bi-shell.
Bivalve.
Bivalve?
Yeah.
Yep.
You're full wide.
Copy that. Thank you.
So the only other thing to think about when you're doing this, and it hasn't been an issue yet, so you're doing fine, but just keep an eye on those starboard swing arms as you come across.
Oh, true.
Because it's
starting to get steep.
starting to get steeper.
Yep.
And so my job as co-pilot is if I start seeing it happen, I can just suck it in for you and I'll let you know. But for now, just make sure you don't plant the side of your vehicle, and that's the only way you're going to hit is if you really bury yourself on the starboard side.
There's a
Gotcha.
But be
pilot. There's a
mindful of
fish
that.
in the middle of the screen coming up.
Yep.
Can we zoom on that?
Of course. Let's get a little bit closer, and then feel free to come in video. I'll start.
Ooh, rat tail.
All right, if you're happy with the lighting, come in as you will.
It's very good lighting right there.
All right.
So we're looking at
Give
here
yourself a
a
little tiny bit of
fish
thrust
we came
back to
across.
stop your momentum
Looks like a
forward.
type of rat tail fish or grenadier.
There you go.
These are fairly common types of fish at these depths in the deep
Right toe
sea.
might touch. Just be ready for it to turn the vehicle
but
a little.
Copy that.
Nevertheless, they have a good diversity,
Still going.
species diversity.
I'm trying to come back a little. There we go.
So we'll let our shoreside scientists chime in if they have
There you go.
Something like that.
any input
Okay.
about identification.
Sorry, I should do a
but
little bit more of a tap.
Likely it's going to be some sort of
reverse.
rat tail fish.
Yeah, so it's good. You want to, when you're reversing up to something like this, you want to get it on it early and give it a very small amount of thrust over a long period of time.
Yep.
That way you're not less likely to produce a large burst that will cause a lot
It's a
of
great
wash.
view of their eyes.
Copy
So
that.
the sooner you can start reversing
Yep.
and just easing yourself,
Bless you. No,
watching your, you don't want to start backing up, but watch your manip cam views, and you can really tell
gotcha.
that you're starting to stop.
No. Oop.
Excellent.
That's my bad.
Going for a full specimen.
Go ahead.
Slight tilt up.
Coming up.
All right. Whenever video has gotten its satisfied, we're ready back here, pilot.
Video's
Okay.
clear.
Feel free to come all the way out. Yeah, come out to lasers first, if we haven't got it already.
It's good.
All right, and all the way up. Let's continue. I'll keep heading towards the port.
What is that?
Looks like
Is that a sponge?
I'm talking
pilot.
right here.
Yeah, right to the right, pilot. This is watch lead 2. Can you possibly just do a snapshot of?
Yep. Feel free to come in video.
Looks like a tunicate.
Oh wow. With the brittle star.
Yep. Yeah. So it's a type of
So
non-vertebrate
floating
chordate
away, right?
correct, called a
Drifting
tunicate
out.
or a sea squirt.
Yeah, so what I think you're doing is pivoting.
And
Okay.
I
You have
can't
your right toe
tell
down.
which
I
which
think
end is
you're-
up, but I
Oh,
can
it's
definitely
down.
see one of the siphons on the right-hand
Yeah,
side.
Pivoting around your right toe, which looks like you're backing. It's hard to tell what.
To the left is a crinoid, a feather star.
Going for a full
So
specimen.
this is
Go
not
ahead.
one I've seen before, but it's good to document. Someone will be able to identify it, hopefully.
It's
It
pretty actually
definitely-
pretty large for
Yeah.
a tunicate.
Right. Yep. They get
Yeah,
pretty
let's go for
large,
lasers.
and there are actually some carnivorous types down here
Tilt up.
in the deep sea.
Coming up.
Occasionally we see these carnivorous tunicates that will capture prey and
Just run the
slowly
lasers right
digest it,
across the creature
rather than
itself.
filtering seawater, which is what
Video's clear.
Okay.
a lot
feel
of
free
the
to come
shallow
up.
water species do.
Sorry, and so you're clear with this specimen?
Yes, we are.
Yes.
All right. Continuing on, heading towards the port.
Full wide.
Pilots, just so you're aware, we are at a good ship heading as well, so we'll be ready for moves whenever you guys are.
Excellent.
Copy that.
So was my toe actually in the mud there? Did you? I didn't see any sediment come up when I lifted.
Yeah, I think just very lightly.
Okay.
He's probably the perfect toe hold.
So it seems like we're seeing a lot of interesting things as we're moving up this steeper slope. But in the meanwhile, while we're getting the ship to move and we're moving the vehicles, I'll give a brief summary of, the bigger picture, what we're doing out here specifically, rather than just at this particular site.
Circle on the right.
So we left the port of San Juan about on the 30th, and we conducted one dive so far. This is our second dive of this expedition,
to what we saw earlier.
exploring deep water habitats around Puerto Rico and the U.S. Virgin Islands. Over the next 3 weeks or so, we'll be out here diving every single day, mapping at night the seafloor, and diving with our remotely operated vehicles across a range of depths to accomplish
a couple of things
various scientific goals that range from understanding deep water habitat usage by fishes, presence and occurrence of deep water corals, diversity of deep water corals and sponges.
Coming up with you.
We're going to be looking in many different habitats,
Still heading to the port.
including submarine canyons, slopes, seamounts, escarpments, trenches, and any other poorly known deep sea environment, deep sea habitats we come across.
Yeah, way across the screen there.
All of the data that we're going to collect over the course of this expedition, from video data.
Lower right swing arm.
There's a fish right in the center.
Everything's good.
Okay.
Pilot, there's a fish right in the center.
Yep.
Thank you.
Coming to it now. I'm going to try and get a little bit closer to get the light a little bit better, and then and feel free to come in video.
Zoom in.
Okay. So this is a reverse.
Looks like another type of Jafira barracks.
Feel free to come off with the lasers.
Another roughy.
Nope. Airplane controls. All right.
Just in case I don't know all my roughies, we'll get some imagery of it nevertheless, and they'll be able to tell us back on shore if it is the same individual. Or not the same individual, rather the same species, at least.
Video's clear.
Oh.
Okay. Science, are you happy?
Yeah, I think we'll move on.
Copy that.
Might be the best we can do for now.
Feel free to come out.
Full wide.
Copy that.
Pushing past the skids.
Thank you. Good. So
I think we're ready for another 20.
there's
another barbel fish or
he got some goodies.
Beard fish.
Oh, you have something up there
moving too
to
quick.
the.
Yeah, do you want to go towards those, or do you want to go? You're heading 340.
340
There's something right to the right of the fish.
Yep.
Yeah, it looks like some trash. I think
20
that's
meters?
a aluminum can.
Did you say 20?
Yeah, 20
Okay.
for now, thanks. It's getting steep.
We have any interest in the
Bridge, RV nav.
debris?
Hey, you ready for a ship move? All right, let's do 20 meters, 340 degrees, 0.2 knots. Good copy.
So that we're looking at another beardfish, and then it looks like there's some trash, possibly a can, aluminum can of some type.
Oh, a
Yep.
Sprite can.
Yep, soda can. And to the left there is actually something I would like to get some imagery on, is a type of coral. This one looks like a stylastrid coral. I think it's alive. Yeah, it looks alive to me.
There's also a live tube worm just below it.
You got a worm tube.
Or something in a tube.
Oh, yeah, that's right. Yep. So just to the lower right of that coral, there's a sponge, and it looks like there's some sort of polychaete worm.
Is that full zoom video?
In a tube.
Now it is.
Slowly creeping out of that structure.
If we're on
Let's Give yourself a little bump up.
Am I?
You're going down pretty quick.
Go.
There you go. The verts are the hardest to get figured out. As soon as you get forward, back, left, right, nailed, it's another like, a few dozen hours till you
full wide
verts.
Okay. Oh, yep. So the coral we were just looking at, I think we'll try and move back in if you have time, is a type of stylastrid hydrocoral.
I'm going to come around to the starboard of it and get a different view. If that works with everyone. Copy that.
And what are the differences between hydrocorals and sclerotinian corals?
First of all, they're both Cnidarians, so they're both belong to the same family or same group of corals that has stinging cells in their tentacles. But this particular group,
small, you can touch down a shot.
Most representatives from this group don't. The hydrocorals don't necessarily make this hard, stony skeleton. It's one thing they have in common with sclerotinians, but their closest relatives are the hydroids we saw encrusting on the corals, the dead bamboo coral a while ago.
So still coming down, still going. Do a little shot to the side.
They
how
are
about that video?
Distant relatives, but they belong within the same group of organisms.
Oh, okay, I gotcha. So you want me to spin around a little bit more?
In general, yes.
Yeah.
Not on this particular.
It's this particular morphology, which I'll try to identify when we get a better shot, is one that is not uncommon in this area, or not uncommon in
I'll try and get to the side.
The Northeast Caribbean at least, but there haven't been a lot of
full wide.
There hasn't been a lot of research on this group in the deep sea.
And do you usually find these species? They seem to be small. They don't grow to, something like what we see in soft corals.
Right. Yeah, they can get quite large, but this particular species or genus, they don't grow very large.
So I think I should have just stayed where
It's unclear why they don't do that, if it's something about their biology or if they just don't get the opportunity to grow very large because they are living in a slope environment and there's potentially a lot of disturbance that could
try and come in there.
Reduce their
maybe also their skeletal structure.
Yeah.
They're lacking that.
Okay, so this is very good. We're getting a better look at this stylastrid coral, which I'm going to tentatively put in the genus Crypthelia. Known from these depths for sure. It actually occurs much deeper as well. With these corals, it's very difficult to identify it to the species level with imagery, so we'll take some very nice images and hopefully that will suffice for our scientists on shore. A number of these corals have been collected in the past, but
So
I think we'll leave this one alone for now.
Backing away slowly.
Gotcha.
And because the angle of your camera, it looks like
I'm sinking. Yeah.
You're sinking.
Okay. All right.
I think we're more or less satisfied if
So
video is satisfied with that coral pilot.
Get an idea whether it's a backing or a sinking, or know where your camera is now.
Yeah, I seem to get too focused on the main. I'll bounce around a little bit more. Okay, I'm gonna start making my way back to starboard, or more like up.
And certainly when you get to, when it's right on the bottom like that, and it's a really small thing, just plant your toe. That's like the ideal situation for that.
Okay.
Yeah. Looks like there's a few other crypthelia to the left, so they're not uncommon in this area.
So we are slowly rising. We're at a depth of 722 meters, now 721. Temperatures gone up just a little bit at 8.1 degrees Celsius.
Is that a ridge that you see above me? Up to the
You could see that there's a little bit more diversity of topography in this area. Hopefully we can see a little bit more organisms.
Oh yeah. Oh yeah.
We're seeing some colorful plankton, which we will be later on doing some midwater dives to identify some of these larger plankton and also survey the water column for the different water parameters.
No, it just looks like a ooh.
And are these all these like small little holes in the sediment? Are these organisms or tracks of organisms or maybe currents of some sort?
That's not
it
a
looks
fish
to
down
me
to my
like
lower left, is that clear?
Yes.
Yeah, it looks fish-like, certainly, right?
Right. Feel free to. Is that lighting good, Roland, or you want me to get closer?
Say, let's start this and then zoom in.
Okay. Yeah. Have that distance. Okay.
Ah, well.
Oh, looks like one of those grumpy fish.
Yeah.
Yeah.
Angler.
Yep. So that's
just slowly push into it.
All right, coming in.
I'll just
So as I push in, the camera should come up, right? Theoretically. Yeah. Yeah. Okay. Cool. Do a little drive by.
So we're looking at a first observation of this type of fish that we've made on this expedition.
You're asking for a lot here, Roland.
Although I believe I've seen it before. If I remember correctly.
Kind of looks like a toadfish in the shallower.
I think it's in
shallow reefs.
In the genus Chonax.
Oh, it's touched down.
A Chonax. Yep. Right. A toadfish.
Now, if you center him there, go tight on his eyes.
So the interesting thing about these fishes is that they have modified fins that allow them to sort of walk on the bottom. You can actually see it's starting to get up off of laying on the bottom now. Sometimes they scoot rather quickly.
Try that before he scoots.
You can see the barbel on between its eyes right now. It's laying down against its face, and they can use that to attract prey items. These are ambush predators. They'll lie in wait on the bottom and attract organisms like fishes or shrimps or...
So I guess with a pirouette, would you prefer, is it like a lateral or is it like a kind of more of a twist.
I'm guessing his beardy tendrils, he could sense, you know, something in the sand or
that's right.
close by
that
he could ambush.
Yep. Excellent.
Yeah.
Slight tilt up for full screen.
Coming up. Oh, that's down.
He does not look happy. He or she.
Slight down.
Yep.
I think they're just misunderstood.
Several seconds.
All right, I think we're satisfied here in the back row. When front row is?
Yeah, let's get lasers on. Just run it across it. Try not to hit it in the eye.
Coming up. Oh, yeah. Camera up.
That's good. Video's clear.
All right.
Yeah, that's right. And one of our shoreside scientists found or saw a type of crinoid in the distance, and I saw it as well.
Oh, did it?
We'll come across it again. I'm fairly sure it was a type of crinoid in the genus Holopus, I believe.
I'll just move my way forward here.
We'll be mainly seeing like two types of crinoids, the ones that are stalked and the ones that are attached by their cirri, which are these kind of like finger-like appendages that enables them to grab onto these hard substrates, while the other ones are based on a stalk. What's really cool is that, and you know, they found that, you know, before they thought that these stalk crinoids could not move, but now they know that they could actually detach themselves from these stalks and swim off if they're threatened by some fish or some other predator. And then they could regenerate that stalk.
We're doing okay on time as well. I mean, we only have to average a 0.07.
So another escape mechanism for the echinoderms, like that of the sea cucumber that we saw earlier.
Okay.
Stalk sponge.
Let's get this moving, then maybe we'll do that.
Say that again, video.
There's a stalk sponge off to your right, and if I call something out, that's just for
it's going to be 20 meters, 340, 0.29.
See if they want it.
Copy that. Oh, yeah. Is science, is the upper right-hand side of the screen anything of interest?
Yeah, the sponge there, the whiter, fluffier organism.
Copy that.
And maybe in the same shot you can grab the crinoid too.
I think I can manage that.
Yep. Oh, it's moving.
I think it's a stalk sponge.
If we want to zoom in. How is that for light?
It's a little bit dim.
Get a little bit closer then?
Yeah.
I don't know this particular sponge, but it's a glass sponge for sure.
In a little bit. Let me know when the lighting looks good, and I'll...
This particular glass sponge grows on a stalk rather than the Euplectella we saw earlier, which is firmly attached to the substrate.
Lighting is good.
Yep, copy that.
Although as I understand it, from what I've heard on previous expeditions, the characteristics we're looking for are how the stalk is attached to the rock, and the general morphology of the sponge tells you a lot about
the stalk and look inside
its identification.
Copy that.
You can go ahead up.
Coming up. I think.
Looks like there's a tiny gastropod right below the sponge. It's really small.
Oh, yep. Where's that, a hermit crab?
Or a hermit crab. Oh, it looks like a hermit crab.
Yeah.
Video's clear.
Copy that.
All right, we're satisfied back here, I think.
All right, come on out. And is there the crinoid behind that anything of interest?
Did we see that one already? I don't think so.
If you want to maybe zoom in so we can,
yeah,
I could
quick zoom.
Go ahead.
Show what I was talking about with the finger-like appendages, if we're able to see. Up, yeah. So if you could see, actually, it's
Copy that.
Yeah, there are some cirri on the bottom of the crinoid, which is the back surface, the adboral surface, that it uses to help attach itself or grab onto the rock underneath, although it could be anything. It could be rock. It could attach to corals or sponges. And then the feather-like appendages, or the arms, are used for entrapping planktonic organisms that may be drifting by.
Okay.
Moving it towards the mouth, which is a little bit obscured right now by the arms, but the mouth is in the center of the arms.
When science is clear, we're going to go ahead and push ahead.
Yep.
All right.
We're satisfied.
Thank you.
So we'll continue upslope. Our depth now is about 710 meters.
There's a big one.
Yep.
Or decent size.
We've seen that particular sponge already.
Copy that.
Probably in the genus Euplectella. Is that a fish or a debris?
Alright, turn by that little black.
Yeah.
On the bottom.
I think it might be debris.
Yeah, it looks like it could be debris.
Just snap zoom video. See if it's debris or not. Yeah.
Yeah.
Looks like bark of some sort.
Wood bark.
Feel free to come out, video. Thank you. Okay. I will push forward to the starboard.
Yeah, and even though it's terrestrial derived,
ship move correct. Copy that.
These woodfalls are important communities in the deep sea. There are organisms that aren't found anywhere else except on wood.
Oh yes, definitely.
Polychaete worms, sometimes sea stars also.
Got me at 49.
3 minutes.
Looks like there's a an eel of some sort.
So we haven't found yet any particularly large woodfalls, but we always keep an eye out for those.
Okay, I'll come in slow. I'll also come down a little. And it looks like there's another one of those sacks next to it. A tourniqueter, I believe. Okay, feel free to start coming in slowly.
Some kind of eel.
Yeah, this is a new one for this expedition.
I'm not sure.
It looks a little different than a cutthroat, doesn't it?
It might be a conger eel. Well, none of us will probably be able to tell you on the ship here, so we'll have to rely on our shoreside scientists to.
Are you happy with lighting video?
Yeah, it's pretty good for this fish.
If you have any more zoom, feel free to take it. Sitting relatively calm.
Maxing.
Copy that. Maybe we can convince him to turn around.
Tilt down. Tilt the screen.
Yeah. How about that?
Perfect.
Have we gotten a shot with lasers yet?
We did.
Not yet, I don't think.
Lasers were on as we came in.
Copy that. We can always come back out at the end and make sure we get a nice setup. I'm going to try and push ahead a little bit and see if we can get his eye. At least that's going to be the thought process. Oh, am I? Yeah. If you want to come out a little bit, Roland. Thanks. Gotcha. So.
Excellent.
Oh yeah, she's coming out. I'm going a little too quick, huh?
Full line.
Copy that. Thank you. Feel free to come in on him again. It's lined up just right.
We're just getting some scaling shots now of the eel, so that we can determine its size later on.
Oh, come over. That's it.
Very prominent nostrils. I can just see them now.
A little bit further.
You can see its small little pectoral fins.
Video's clear.
Okay. So you satisfied with this?
Satisfied, yep.
Yes.
Copy that. Feel free to come out video.
Nav, this is watch lead.
Looks like he ate something recently.
Go ahead, watch it.
Stomach has a little bulge.
How is our pacing? Are we really want to get to the end waypoint for this dive?
Sure, we're looking very good right now. We only have to average about 0.07 knots.
Okay, excellent. So we mentioned that at the beginning of the dive that this
come over to the port a little bit.
dive will take place at first over a steep terrain, but it will level out. But there's a large mound feature that we really want to investigate at around 465 meters or so. This has been
Sorry, I couldn't hear you. What was that?
referred to as potentially a mound that deep water fishes might congregate around or aggregate around.
360 or so, probably north. Yeah, okay.
Some of them are more important deep water fishes are those of the queen snapper, also known as cartucho.
You want a 30-meter move? Or you want to stick with 20?
This area is targeted by,
we're just going uphill, 30.
fishermen, and they fish these fish through vertical long lines.
Are we thinking maybe
and
more of those dips?
Much is known of their habitat and what they eat, how they reproduce. So this expedition is very important. And we saw that yesterday when we we saw large schools of silk snappers and other commercially important fish.
Hey, we're ready for another ship move.
And
we're going to do
St.
30
Thomas,
meters
and
at
also some
0,
misty
0, 0,
groupers.
0.2 knots.
So it's unique sighting, I believe, for fishes, especially the groupers. There are some pretty big groupers of misty groupers that we saw yesterday on the dive.
So on shore, Kevin Rademacher has wrote in about that eel we just saw, and he mentioned it could be a short belly eel, Dysoma, or a relative of that. Yeah. We'll have to scrutinize the video a little bit more for some details about the pectoral fins, but that's our running identification right now, tentative.
So for this, I'd probably want to angle up a little bit more, right? Okay. All right. There's a fish right there.
Yeah.
We've seen quite a few of those, right?
Yep.
Yep.
The beard fishes.
Beardfish.
And what about the one to the starboard?
Probably Mixia.
That one there.
I think we've only seen one of them so
There's
far.
something else. Maybe another. Looks like some fish of some sort. Straight ahead.
Okay. True. Do we want to just get a snap zoom with that? I know the lighting's bad.
Yeah, please.
Just to see what it is.
I think it's a halosaur. It looks like a halosaur. They have these elongated snouts Sometimes it looks like a duck bill.
Video's clear.
Yeah. All right. Feel free to come out.
Nice.
Okay.
Ooh, there's a shrimp.
There's some shrimp.
Shrimp.
Nice
Yeah.
follow.
It's. I don't think we're gonna get that one.
He's going pretty quick.
Usually the halosaurs, we see them swimming uphill most of the time.
I think I have the clockwise turn and the push down on the camera done pretty well.
I think they try
so
to
maybe I'll
ambush
just line myself
prey
up
as it
for every
moves
shot.
down
That was
slope.
Ah, okay. Come up from behind.
Yeah. They always seem to be facing
keep going
uphill.
up and out. And there's another fish there.
Oh,
it looks like another one that we've seen.
Beard fish.
Possibly. Something else that's laying down.
Green eye, maybe.
You said there was something else that's laying down.
Yeah,
that
where that
fish
fish
laid
was.
Oh, the one that just laid down. Gotcha.
The beard fish wasn't fond of staying still, so I think this might be something else.
Copy that. I will push in, get some better light on it.
Yeah, rotate around a little, get squared up with it. You got room to do that.
To the starboard, you mean?
Yep.
Copy
It
that.
looks like it might have a little bit of different
rotate
coloration
left,
also.
get yourself squared up.
There's another fish up above as well, to the starboard.
And since you're tilting up pretty high like that, you're actually you're like downhill from it.
Okay, so you want me...
It might work. It might look good, and you might get a shot without scaring it. But if you tilt it down and line
So
up,
a lot of the
it'd be
fishes
little
we've
closer.
seen so far, they're not these snappers we've
Feel free
been
to
looking
come
for,
in,
but
roll in.
nonetheless there are
We also have a very
a decent
small
number
thing
of
that's
fishes on the
squirting
slope.
to the side there.
Clear.
I don't know what
Drifting
that is.
back.
What is that little
Hatchet
fish?
maybe?
Is that what
Rat
that is?
tail.
I can't tell. Rat tail? Small. It's not a hatchet fish. I was wrong.
It's a small fish.
Fall off that quick.
That looks like a really tiny
come
rat
out,
tail
please, video.
fish.
Thanks. Alright, there we go. Okay, you can come back in on the little fish. I think I got myself situated. Okay.
Lasers are clear.
Okay, is that a full zoom or?
No.
You're also pretty
Are we interested in the little or the larger?
The larger, yeah.
Okay.
The little fish might be hard to tell anything on, but it looks like a type of rat tail, which is one we've seen before, but the bigger fish is a new observation for this dive.
Copy that. I should probably get closer.
Yeah, so you're going to touch now.
How's that for lighting or all?
It's a little dark, but you can
Little
use
dark.
it for this
Got you
shot.
at 80 degrees.
I can use it for this one.
How about that?
Better.
Well, you got a few seconds here before we
Okay.
take off.
Alright, go ahead there, roll.
Max zoom.
What should we have left in that move?
It should be coming to an end right now.
Okay.
Coming down for full. And
Got some oscillations. going
Excellent.
on.
yeah,
Any idea what kind of fish this could be?
Pulling out a little bit.
Well,
Family, ballpark?
Our shore team scientist, Kevin Rademacher, says that it might be a rat tail loose.
I
I'm
think
not
that
sure.
was the last
That's
one.
the last
That-
one.
The tiny one, yeah.
We're
This
getting,
one is.
some.
This one looks.
trying to get you a
Not
little
like
bit
a
more
rat tail.
stable there for video.
We've got enough of this.
Okay, I need to push out quite a bit.
Excellent.
I was lifting up when I thought I was lifting down.
We'll continue upslope then.
Okay.
We'll wait on an idea for that. Oh yeah, that's right.
So there's something there on the wall. Is that ship move done, Andy? Do you feel comfortable taking over now? Okay, I'm just going to lift you up a little bit. I'm coming up with the vehicle. Well, so science, we're just going to do a quick rotation of the pilot and the nav. Okay.
Excellent. Thanks, pilot. So while the front row is doing a quick swap around, we will, let's say, we're about halfway up this slope feature.
Co-pilot and nav.
Gotcha.
So I think we'll have plenty of time to reach the end of this mound structure that we've been targeting for the rest of this dive. We're crossing now.
Great, sounds good.
Just about 690 meters.
You got room. Give it a good push.
Yeah.
The top of this feature goes to about 600 meters or so, and then another 100 meters of vertical depth until we reach that mound structure. But everything is looking on track to meet that goal. We've seen a number of fishes so far on this dive. They've probably been the most abundant organisms on the seafloor here, but none of them are the commercial species that we intended to dive on to observe, but there's a strong evidence that as we get just a bit shallower, they should start coming into view.
Yeah, we still might be outside the depth range of these.
That was the same one, isn't it?
Of these deep water snappers.
Similar.
So when we get closer to the mound, hopefully we'll get some shots
on right now.
No.
Of these queen snappers.
Yeah.
That are known to be in this area.
So it looks like another one of those cyanogrops fishes.
I wonder how close we can get without scaring him.
Is there a common name for that?
Come on, video.
Not what I see.
Common name for Synagrops is, that Synagrops bella is blackmouth bass. This is Kevin.
Thanks.
Thank you.
Blackmouth bass. Yeah, looks like the same one we imaged, but this one, they seem to be behaving very well for the camera.
He doesn't seem too bothered by the ROV.
Oh. Nice slow pan down the bottom.
And like the last one, I didn't see any obvious signs of parasites or anything like that on these fishes. So, you know, it's sometimes worth taking a second look, even at the same species, if there's potential evidence for isopods or copepods or any sort of parasites that might be attached. Yep, I don't see any.
He's pretty clean.
Good job.
Hey, this is Kevin again. Just a little bit more on Synagrops. We get 3 species of Synagrops in the Gulf of Mexico, and I imagine down around Puerto Rico. The largest one is this one that we have on video, Synagrops bellis, the blackmouth bass. Then there's a Synagrops spinosus and a Synagrops trispinosus. And to differentiate them is the presence or absence of serration on the leading dorsal spine.
Video's clear.
Looks like you're starting to
Excellent. Yeah, so you could see how we can make an identification, but some of the details sometimes are difficult to discern using our cameras. So we're lucky we can get a species level identification on this one, but
who's trying to catch up?
It's always worth a second look in case there are fine details like that that we can pick up with our camera.
Satisfied on a move for now, right?
We're at a 50-degree angle, so I mean we're looking great.
You got it.
Quick zoom here video. I don't know what that is above the lasers.
Oh, yep. Yeah, that's one of the crinoids we were talking about earlier. Type of, I think it's a type of stalked crinoid. This one is called holopus, and I think I could be wrong about this, but I think it's restricted to the Caribbean and parts around the Caribbean. I don't think it's widely distributed in the Western Atlantic, but yeah.
I can land and get a much better shot if we want. Come on out for just a second.
I've actually handled one of these once on a previous collection, and they're actually quite hard. They're you know, almost stony. Yeah, they're not like the brittle stars or feather stars that jettison their appendages. They're quite beefy. Oh, okay, excellent. So holopus is found in the northern Atlantic on Meteor Seamount.
This is the first time we've seen this on the Okeanos when it's set up for a super.
How do you feel about fog lights for this one, Jeff? What do you think?
I'm in.
Okay. Let's go ahead, come in video. We're going to actually turn on some lights too.
Do a zoom
like this. I think he's finally closed up. I think he heard you, Roland, say that you wanted a better photo, and
yeah.
Still could be cool.
Not happy about that.
Looks like one of those pine cones.
Exactly, yeah.
Is that max zoom?
That's max zoom.
Copy that.
Oh, let us know when you're ready for a little extra.
Yeah,
light
video, you let us know.
In the background, there's definitely a cup coral there. I can't quite tell from here if it's live or dead. They're very difficult to tell unless the tentacles are extended.
Do you want it centered up, or do you like the placement?
So, okay, there. You can center it up now. I was just trying to get that.
Do you mind if we turn the lights on?
Base. Yes, please.
Okay, let's go ahead and try that, Jeff.
Coming on, it's about to be bright.
Yep.
Oh, that's the wrong button.
Oh no.
I'm not too much too sure about the feeding mechanisms of these particular crinoids. I know they have a very rapid retraction, so I wonder if they're potentially could be carnivorous or if they're strictly planktivorous or detritus feeders.
What's that video?
They might be all of the above.
I think it's all of the above.
Oh.
There are a lot of echinoderms that are carnivorous. They eat other animals. Especially in the deep sea, they tend to be...
Oh,
hey, look
there's
at
a
this guy.
fish.
Oh.
Yay.
You wanted a fish? We got you a fish.
Didn't even have to work for that one.
No.
Pulling out
It's another kind of eel-like fish, yeah.
He's stealing the spotlight.
That might be a small halosaur, actually. Just, it's very translucent. Very young fish, whichever it is.
You got any more zoom for him? Oh, you got a lot. Did you want a specific view of this watch leads, or is this pretty satisfactory?
Yeah, we're satisfied when videos
Whenever
had
you're
their
good, then video.
quota.
Okay.
I've seen outcroppings before where holopus grows extremely dense. Many, like hundreds of individuals. I can show you a photo later. They can get pretty dense on some lips and outcroppings.
Yeah, I'm a big fan of echinoderms. They thrill me the most. Actually, these look like stock sponges that we saw before.
Oh, yep.
Yep.
Yeah, there's a couple of sponges there. Ones we've seen before.
So red crab.
That one looks like a tunicate there in the lower left. That was like the tunicate we've seen previously. Gelatinous looking. So we were mentioning just a minute ago about echinoderms and how they're sometimes predatory. Definitely predators of corals and sponges. Haven't seen any predation going on just yet, but we're still very early in this expedition.
Oh, I see what you're talking about.
Oh, we're coming up to a, looks like a nice ledge.
Real quick, what is this?
With some growth.
Yeah, that's right. It's covered with sponges.
Oh, wow.
Got a big mass to the left too, just once you're done here.
Here are some more, looks like more glass sponges on the edge of this outcrop.
This is neat.
Yeah.
Did you want to zoom on these? Or do you want to just
yeah, just generally,
overview?
nothing specific here.
Okay, we can just do a general kind of flyby here.
Yep.
Great.
Just because it's a different morphology of sponge that we haven't seen yet.
Partial.
I
Some
think that's
look
perfect.
pretty fresh, some look older. So these are most likely glass sponges, hexactinellid sponges.
And the glass sponges differ because their spicules are made of silica and demospongin. Their spicules could be made of spongin or calcium carbonate, but we won't see those this deep, the calcareous sponges.
Yeah. There's a couple of demo sponges in there as well.
Yeah, the
like
orangey.
The one we saw that one yesterday, I believe. All right, I think satisfied here.
Great, let's come wide video. Let me go see what's over here.
I think it's just a hole.
I think so too.
It just looks cool from my view.
Yeah, it did.
Oh.
That's me. Watch leads.
Just feeding the locals.
Yep.
So this is probably the most substantial rocky hard bottom we've seen the dive so far. And this particular sponge seems to be restricted to this type of slope and hard bottom. I haven't seen this quite yet, but
this is full wide video. Is that
correct? You're full wide.
On other hard substrates, we've seen black coral whips, Dichopathes, attached to small exposed hard bottom.
And what we've been seeing on the West Coast through ROV and AUV videos and photographs, when you have these abrupt change in structure, like what we see right here, this outcrop, this is usually an area with high diversity of sponges and some of these deep water corals. And we see this right here with all these sponges.
Oh, actually, to the lower right, I see a whip.
I think you're clear.
Yeah.
Yep. I thought there would have been more on that particular outcropping, but maybe there's something coming up. More diversity. I mean, there's quite a lot of sponges, so.
Give us a bump ahead.
Ooh. Yeah.
Feeling another 20 meters.
Another 20.
Yep.
Shiny.
Copy that. I'll do 20
I think
meters.
that's one of those bivalve shells that we've been seeing.
We're still happy with that?
Let's
or
do 15. This is pretty bright, just in case.
Copy that.
Nice.
015.
Oh, sorry. Go due north. Let's do 15
Oh, I just saw a shadow of a large fish on the left-hand side.
Sorry, watch these. What's that? You want to look at a fish?
I just saw a shadow of a large fish on the left-hand side. Yeah, it might have been above the vehicle, but it may be gone. It looked like it was
Bridge
moving quickly.
Would like to call another move if you're ready. We're looking for 15 meters.
So you said it was where? Watch it.
Excuse me. Yes.
I saw it generally in the left-hand
meters at
side of the screen. It was
speed
a shadow. It looked like it was high above the vehicle.
Oh,
and,
it might be between Sirius and D2.
Yeah, it probably left. It was moving very quickly.
Okay.
But it looked...
good copy, yes.
It looked like the fin structure, the shadow of the fin structure looked like it could have been a larger predator.
Oh, well, we'll keep an eye out.
Yep.
There's a couple fish coming into lasers.
Yeah, I think these are going to be ones we've seen before, but we can snap zoom.
You want to do a snap zoom and see video? Oh, it is.
Halosaur.
Alright. Come on out, video. And then I thought I saw one more over here. Yep.
Yep, to the left.
I think it might be the same.
Yeah.
Alright, come on in, video.
Yep, another halosaur.
Okay.
It looked like a... Actually, the camera is so good you can see the Ketignaths shooting around when you zoom in really close. These are arrowworms. They're usually mid-water organisms, but they're hard to see sometimes. But when they accelerate quickly to escape or attack prey, it's sometimes very easy to see them.
They're ferocious predators. Definitely.
There's a circular thing diagonal upright from lasers.
Y buenas tardes a todos. Están escuchando especialmente en el Eco Exploratorio en San Juan.
Yeah, you can do a quick zoom if you want.
Y ahora estamos buceando en aguas de profundidad de 600 metros.
Got an edge here that's got a lot of goodies on it.
Yeah, I'll come right back to there now.
Muchos peces en
right up to it here and then buceo
y muchos crinoides, pepinos del mar. Y ahora estamos subiendo y espero cuando más estamos en aguas más llanos vamos a ver peces
we noticed that the shelling was stowed a little bit
chillos
in a different position than what it normally is. Do you think that would be causing that flare on
en este área.
left-hand side of the screen?
Bienvenidos.
Well, I'm also pretty far up.
Yeah, I think his flares are because he's tilted up so far.
Yeah, I'm very positive.
Gotcha.
Go around this corner here.
Can we look off to the left side? Actually, I thought I saw a yellow coral structure.
Absolutely.
Crab at the top right.
Stand by video. I think he wants to look at some yellow.
Yeah, kind of in the upper left corner now.
Upper left. Okay, I think I see it.
It looks like a blob from here, but I just want to make sure it's not a small coral.
Let me get a little bit closer to it. Talking about the one that I'm approaching.
Yeah, right ahead now. Looks golden or yellow.
Sure.
Lasers are just coming up to it.
Go ahead and we'll do a zoom video and we'll see if I need to set up.
Oh, it is a sponge.
A biomat.
Okay. It is a sponge we've seen yesterday, so
copy
it's
that.
a demo
so
sponge. Yep.
Might be a different fish
move
on
on.
the top of the ridge left.
Back to the crab if that's possible, or if not.
Yeah, we can go back there. Video thinks he saw a fish over here. A different
Top left corner, just see him sticking above.
Don't see. Oh, over there?
Yeah. We can snap from here to check.
Yeah, go ahead.
Just the way he's sitting looks like a tripod fish.
Oh,
Yep,
yeah.
it is.
Ah,
We can get closer if we want to get a good shot of him. Is this something you want to get a good shot of, video? Or watch it?
yeah, this is a new.
It's a new observation, yeah, for the dive. So
Alright.
we'll get as best a image as possible.
Not a problem.
Oh,
Pilots, I'm just going to play with HiPAC for a second, just so you're aware.
What are you doing?
Calculating the time
Oh.
until the end.
Oh,
just
yeah.
for my own reference, where I'm the last nav today.
Sure. Okay, come on in, video.
This is nice because this one's actually facing us.
As close to it as you can
Nope.
get.
Yeah, I don't
Maybe
want
not
to
too
scare him
long.
yet,
I
so
said
we'll
that.
start here and then I'll start moving
Jinxed it.
closer.
This is not one I've seen before. These looks like a type of tripod fish, but yeah, I've not seen this particular type of tripod fish before.
It's going to
I think
his
it's a
eye.
tripod fish.
Go ahead.
Accent.
Okay. Yeah, after this I'll try to move in a little bit more, but I didn't want to scare him away.
Yep.
You often like just turn around,
Look at his front.
get all lined up, and then he just goes
Yep.
90 degrees.
Yep.
Front tips of his feelers.
No thanks.
Yeah, those are cool.
Okay, you can make your move.
Okay, let's see.
Partial. Lasers are
So this one looks like it's a tripod fish in the genus Bathypterois.
Gonna just hop on over to you.
Good copy, Bridge. Thank you.
These are seen fairly regularly, so they're not really rare, I would say. But this is the first time we've seen one on this expedition and on this dive. Typically they
Okay, you have some zoom video?
face up
Pilots, would you
current
like another move?
or uphill,
Last one just completed. We'll do another 15.
and they're a type of ambush predator. They'll ambush animals
Actually,
or
don't have anything imminent ahead
prey
of
that
us,
are
so we could do
floating
a bigger
downhill.
one once he's ready.
Copy that.
But he's
Let's look at that front
in a good
feeler.
position for now. We might wait a second.
Standing by.
All right, I think we've got a good enough imagery for identification. If video is satisfied, we'll carry on.
Copy that, watch leads. We'll just finish up right here.
Slight tilt up.
You want the top feelers?
Yeah, both of them. There
Okay.
we go. And video's clear.
Great, come on out.
From the distance, those guys have a very unique stance on the sea bottom.
You seen this guy up here? Is that one?
In the upper left,
Yeah.
I can't quite tell.
Looks
I think
like
that might be
type
another
of anglerfish.
Let's go find out.
one of those blackmouth bass.
Is he
Oh, is
turning
it?
around right now?
oh yeah. Looks like it's...
Yeah, he just put his tail towards you.
Yay. From a distance, it fooled me.
maybe it's another toadfish?
Yep, it could be.
Slowly rotating.
A little here.
Yeah, that'd be great. Come on.
Or this one could be a goosefish. Yeah, that's right.
Oh, he's just going to give us...
10 degrees off track now.
Copy.
Yeah, I think this is a type of goosefish. It looks very similar to the toadfish, but it's a bit more flattened.
Laser's clear.
And their tendrils coming off of their,
Let's look at those
their
teeth.
ventral part
Go
look
ahead.
kind of like stylastards, like, to kind of lure
Yeah,
maybe some animals.
yeah.
Tilt down.
That's a good point. I never thought of that. I always thought they were, they resembled like polychaetes or something that would attract prey items. I never thought it would be like a habitat
Yeah.
that prey potentially would be attracted to. Or they could be sensory as well.
Yes.
We're getting a good view of the face now with the barbel laid back across just between its eyes. Not barbel, or rather lure, I think it's called. Scanning for
Looks like
prey
he
items.
actually has 3 of those little barbels, 2
Yep.
in the front and 1 there slightly.
Yeah,
back.
I believe they have multiple. That's right.
Okay, go
Another
full
kind of
screen,
ambush
video's
predator.
clear.
These fishes aren't typically caught around here. They're not targeted as a part of the fishery. No?
No,
To the best of
not
your
that
knowledge.
that I know of.
One more tilt up.
Okay.
Okay, excellent. Thank you. We'll move on when video is satisfied with their shot.
And we're clear.
Great. One out.
Full line.
Thank you, video.
Steve, it's Kevin Rademacher. Daniel Wagner identified it as Lophioides B-row,
Back starboard.
and there's
Back
another
again.
species that's very similar. The view that you have right now with the Esca, the lure between the eyes showing that it's dark, is how you differentiate those 2 species. B-row has the dark Esca, and the other species has the light Esca.
Yeah, I think so too.
Excellent.
Another 30 at 000.
Thank you very much.
Yeah.
Okay, I will call it in. Bridge, this is ROV nav. We would like to call in another move if you're ready. Range 30 meters, bearing 000, speed 2 knots, please. Good copy. Thank you, Bridge.
So there might be some students from the University of the Virgin Islands watching this video. We're presently on the edge of the Marine Conservation District,
We are keeping out
the
whatever
MCD,
this might.
and
Take a look for it.
we are now
I mean, yeah, we're gonna keep an eye on
getting
sonars, make
going
sure
up
that it doesn't
slowly,
sneak up on us.
What is that
trying
depth?
to reach
650,
a depth
430?
of about 400 meters, 400
Yeah,
and
we're in the 650 range. That so.
and
you're
65
gonna see a
meters.
steep wall. Another one,
And trying to
probably.
locate some of these deep water
Keep an eye out for it.
snappers, such as the queen snappers. So welcome to all of you that are listening to this line.
What's that in front of us? Did you see something close up their video?
No, I didn't. I was looking at the other computer screen at the
Just
time.
a quick zoom real quick. See if it was... Oh, maybe it was a fish. Oh, it was this guy.
Another Bathyterois, yeah.
Yep. Okay.
So right now we are at a depth of 657 meters, salinity's 35, PSU, oxygen 2.6 milliliters. The temperature right now is 8.97 degrees Celsius. We are slowly making our descent, hopefully reaching this mound-like structure.
Looks like we may have something.
Little ledge.
Yeah.
Looks like there's some break in the terrain.
Guess it just runs across here.
The wall looming in the background there. It's just barely lit up by your light. Some more interesting bottom, maybe.
There's a thing directly below us. I think a purple holothurian in HD2.
Oh.
Didn't even see it as we approached.
Yeah, we can do zoom.
So we're looking at here, looks like to be another species of sea cucumber, holothurian.
Okay, whenever you're good video, you can get whatever you want on here.
Just pan up the body.
As Steve mentioned before, these are kind of deposit feeders. You could see that they look for detritus and other dead organisms along the seafloor. These are closely related to sea urchins and the crinoids that we've been seeing.
Okay, well, yep, there they go, coming up.
You can see some
fecal matter.
Yeah, some casts.
Video's clear.
Come on out.
Yesterday's meal. It's an easy way to trace if there has been sea cucumbers in the area.
Yes.
In shallow water too, actually, they make large castings.
Good copy, Bridge. Thank you. Pilots, that move has been
there's
completed.
something like to the left.
To the, right there.
Or maybe another sea urchin?
Yeah.
This guy's weird. It's kind of like two big rabbit ears on it or something.
Yeah, we'll get a good shot of him. Come on in video.
So that looks like
wow.
Another type of pancake urchin, Formosoma.
Slight tilt down.
I'm going to tilt up.
And the big round things are modified spines, or is it pedicellare?
Like?
Are they 2 feet? Modified 2 feet?
Which ones?
The large kind of sacs that are hanging. Or those are the gonads?
No, I think they're for buoyancy and balance to keep it upright.
Oh.
But these actually, they're quite venomous, from what I understand. They hurt very badly.
This one looks pretty neat.
Yeah. So I think the sacs are filled with a type of gas that helps them stay upright.
Oh, okay.
Yeah.
We have a fish in the middle of the port cam too.
Yeah, but I do remember that. I've heard that these are quite painful if you get spined.
Zoom out for just a second, video. I don't know if this is a different fish that they wanted to see. Oh, he's finished. He's gone by. Where'd he go?
What's up?
Oh, there's like a dark
Where'd he go?
eel looking. I don't know if it's debris.
I think he swam off. All right. Are we good here, Watch Leech?
Yes, thank you, pilot.
You got an odd looking circle right there, middle screen. Yeah. On mine.
On yours?
Big black circle right there. Kind of
Oh, yeah, I do kind of see that. Okay, let's go see.
Shiny fish left.
Yeah, I just
there
saw him.
seems to be like, yeah, a fish right over to the left. This might be new to the expedition.
Let's go ahead and video.
Or, yeah. No. Man, I've been wrong every time.
I was hoping he was going to continue on forward with this.
Yeah, this is...
He did not.
This is new. Anybody on shore side, Kevin or Daniel, that can...
Extremely reflective.
If you know the species or genera.
He's up above you.
Yes, he is.
Pretty cool. See his like little pectoral fin moving rapidly right there on the sides.
Oh, I'm slipping just a little bit.
Does not want to stand still. Not a big fan of the ROV.
No.
It's odd, most fish are.
I know. Yeah, I'm actually surprised by the amount of fish that we are able to tape.
Stacy, that's in the order Zeoformes, maybe one of the dories. I need to refresh myself real quickly on it. With it being that small, I'm not sure if it's a red dory or possibly a juvenile.
Float with him this time.
One of the others.
Yeah,
okay, thank you, Kevin.
I can zoom in. I kept you at your...
Yeah, I was trying.
I kept you at what seemed comfortable. A nice angle of the lights there. Now you're getting real close.
Or is he getting close?
Yes. Lasers are clear. Now he's going back up.
Oh, jeez.
We're finished.
Okay, get out of here, fish.
Hi.
Stacy, looking real quick, I think it might be Cytopsis roseus, the red dory.
Circle.
Okay.
But my video's buffering again, and I need a couple of more looks.
I'm okay with that.
So what would
There it
tell
is.
them apart between the red dory and you mentioned another dory? What would be those characteristics?
The larger dory,
It's an urchin.
Xeniopsis conchifera, has bucklers, large enlarged scutes at the base of its dorsal fin and anal fin. Cytopsis roseus, the red dory, does not. When in hand, it has a reddish color, but down this deep, we may not be seeing the red color with the artificial light.
Okay, thank you.
We can get any zooms you want here video as well. I think it's like 4-ish.
The estimated time off bottom is 4:09.
So this is another sea urchin?
Local.
This might be one of the pancake sea urchins.
Copy that roll.
It's a great
Sorry, I
shot.
just had to move it a little bit.
You could see the tube feet kind of moving along. These are the really small
Oh, you want to go down to the bottom?
tube-like appendages that are kind of floating
get
around.
the feet going. They're talking about them.
This helps sea urchins kind of grab onto things. And actually you could see the pedicellari, which are modified tube feet that have these claw-like structures. Great shot. You also could see the 2 feet. It helps them move.
Slight tilt down.
They have this really
Down.
unique water vascular
There you go.
system that they use the seawater to kind of extend and retract these 2 feet.
Okay. Go back to the top.
So I'm here, since we have a chance, with Debbie Blaney. She
Go to
is
the center of its
the
back.
web coordinator for the Okeanos, and she's going to be sitting in for a little bit.
There's
While
the
Steve
center.
is on lunch break.
On lunch
Okay.
break.
Ah, there he is.
So welcome.
Thank you. Thank you, Stacy.
Sometimes that thing will open. Going full screen.
So I guess these are modified spines. They're like paddled-like
Tilt down
spines.
full.
So again, these are relatives of the crinoids and sea cucumbers. They're characteristic because you could divide them in 5 equal parts, so they have this radial symmetry to them.
Video's clear when science says.
Copy that. Watch leads, are you good here?
Yes, thank you.
Come wide.
How do you guys feel about another move?
How big was that? Did we put lasers on him? Just for my own curiosity. Oh,
Oh, wow.
it's a big one.
Yeah, pretty big. 20 centimeters.
Yeah, Sirius's sonar looks good.
What is it, 30 meters?
20 or
What's your
30.
depth right now?
It's 648. What's that ridge at?
30, 20, 10.
Okay, maybe 20 right now.
Right,
We're
20
we're
at
looking out for
000.
this. Sure, yeah.
Sounds
Yeah.
good. Calling it in.
We're supposed to gain 40 meters in altitude the next, like, very short time, so we're
Yeah,
keeping a close eye out for that.
sounds good.
Might be right here looming.
Yeah.
I can pick up and look.
Let's see.
So right now we're at a depth of 648
Got a gentle
meters.
one. I think it's just beyond this.
Temperature is slowly rising. We're at 9.2 degrees Celsius.
20 meters. Bearing 000. Speed decimal 2 knots, please.
So Debbie, are you getting a lot of feedback?
Thank you very much.
With the post? If you all do not know, we are on social media and Facebook and Twitter, Instagram.
Yes, and if you have any questions, please submit them over social media. We will be answering them as we dive.
Is it just a rock? What is that? It's a very,
Looks like it's a rock with a couple things growing on it.
Hey, video, you want to do a zoom right here?
You got a brittle star behind it, looks like.
Ah, there's a brittle star.
Huh.
What is that?
What is that?
Clear thing right there.
Clear thing looks like some type of... it doesn't look like a gastropod, but it looks like maybe a bryozoan of some sort. The shiny.
I think it's a bivalve shell.
Yeah, it's a shell.
Stacy, is there a common name for bryozoan?
No, that I know of. They're understudied group of organisms.
You never know.
Not much is, I mean.
Yeah, we're starting to get some terrain here.
Known of them.
It's a good solid return.
I believe they're hard to identify. I can be wrong.
Okay. It's kind of cool. But, sure, yeah, we can. Were you asking me video or were you talking to watch lights? Oh, okay. Oh, perfect. I usually am.
Okay, so we're just going to do a quick introduction around the room. Welcome to all those who are joining. Bienvenidos. Todos están escuchando. We are currently slowly getting shallower, and the mission for today's, or the objective of today's dive, is to try to locate some of these commercially important deep water fishes, silk snappers, and queen snappers, cartuchos. My name is Stacy Williams. I'm the science co-lead. To my left, we have...
Is that what that is?
I'm Debbie Blaney. I'm the web coordinator, and I'm currently sitting in for Steve Oskovich as he's having
a tiny
lunch.
little one.
An octopus, very bottom left of the screen.
Yep, copy that going.
And if we can, if you guys have time in the front row, pilot, co-pilot, navigator, to introduce yourselves.
Where'd it go?
Yeah, I'll go ahead and take the lead on that. Sitting navigator is Lars Murphy, currently piloting and going after what we believe is a small octopus, Sandy Lister, and co-pilot is Jeff Lanning. Over in the corner, doing the video is Roland Bryan,
come land here.
And in the back clipping is Emily Nero.
Thank you. Yeah, this looks like a small octopus.
This might be a candidate for fog lights in a little bit.
Are we doing any steals during this?
So octopus have these really cool cells in their skin called chromatophores that allows them to change colors and kind of camouflage with whatever
Let's go ahead and go with the extra
their surroundings are. This one's not that
Yeah,
camouflage since it's a reddish color. Oh, we kind of could see it.
And we just turned on some extra lights that we have for close-ups.
Not the greatest angle. He's a little guy.
Stacy, do you think this is a juvenile, or would you think it's an adult? It's fairly
we should
small.
be getting close
Yeah,
here.
it's fairly small. I'm not sure if this species
Copy that video. We'll wait.
grows
We'll give
to
him
this
just a second
size.
longer.
That's
Good
why
copy,
it's so
Bridge.
small,
Thank you.
or it's a juvenile.
Pilot set move is completed.
Oh, okay, you can zoom out. Thank you.
All finished watch leads?
Yes, we are finished.
Great.
Thank you.
Come on out, video.
So yeah, I'm not too sure if this is species specific size or it's a juvenile octopus.
I'm
gonna
pop ahead.
So we use those two red laser dots to tell scale. They're 10 centimeters apart and help us determine how big something is.
Yeah, it's easier to get distracted during these videos, and the scale bar helps scale. Well, these lasers help scale
I don't know. I don't want to set up if it's
their
nothing.
size, which can be useful measurements.
I still have no idea what it is.
I think it's a shell
Something
behind
shiny.
Oh.
something else.
Yeah, you can come back out. I don't think we're going to want to.
Stacy, we've seen quite a lot of fish on today's and yesterday's dive, and I assume we'll see more when we dive deeper. How do fish differ when they live in shallow water as opposed to deep water?
So as we noticed, the fish, especially on today's dive, we were a little bit shallower yesterday. The fish on today's dive are kind of like eel-like shape. They have big eyes. They usually have a metallic cover.
Have you seen this guy right to the right of lasers too?
Helps them camouflage against predators.
I guess we could just do a quick zoom.
And shallow water species, you get more vibrant colors of fish, and a lot of different morphological forms of fish. So that's a couple distinctions between the shallow water species and the deep water species. This looks like an anemone. This is an idarian, I believe.
Oops.
These are similar to corals in that they usually have stinging cells in their tissues.
Video's clear.
Okay, come back out. Maybe we'll just fly by the goosefish. I think we've seen them.
So yesterday's fish, you know, we saw the silk snappers and the big groupers, and those are predators. So they're usually this form of, you know, like a more torpedo type of shape to them, so they can move fast and hunt their prey in the water. So that's a little bit of a difference.
I mean,
not so much groupers, because those are ambush predators that kind of sit in the bottom and wait for their prey.
Two fish.
This looks like another sea cucumber to your bottom, bottom left.
I think that fish is going to be hopeless for a zoom. Yeah, come back out.
Okay, video swapping out.
Copy that.
So Stacy, here's a question for you for some of our younger listeners. What exactly is a sea cucumber?
Yeah, it looks like
so a sea cucumber translated, sea cucumber is is relative of a sea urchin. It's pretty cool. I enjoy sea cucumbers every time I dive.
Significant flat distance
but they're
ahead
deposit
of us,
feeders. They have these like kind of
after this, you mean?
appendages
After this,
that
yeah.
they can
So
expel and
they'll be able to make
kind
up
of
time if we're
feed
losing.
on whatever's
We're
living on the
holding time
bottom or
pretty
falling
well.
from the plankton. And they're actually used a lot of these sea cucumbers are harvested for medicinal uses, but also they're a food. In Japan, they harvest a lot of these sea cucumbers, so they have a commercial importance. So yeah. And as I mentioned to you before, a cool fact for sea cucumbers is that if they do feel threatened, they will basically expel their intestines, and they can leave those intestines behind and regenerate those intestines in 2 days. So this is called evisceration. So. Looks like we have something ahead of us. It might be another sea cucumber. And we've been seeing a lot of sea cucumbers in this area.
Yeah.
Must be a lot of food.
Yeah, I think I'm at whatever it is.
What do they feed on?
They feed usually on detritus, like scraps of other organisms.
That's good.
I'm not sure if there are sea cucumbers that are carnivorous that eat other animals.
Waypoint 3
they might be
is
omnivores, meaning that they eat whatever is available.
Sure. Yeah, let's get on top of this.
Good, copy that. Another at 000, you're thinking, what, 20 meters?
Yeah, yep.
Copy that. Bridge, this is our nav. We'd like to call in another move if possible.
So we're going to now see these orange glass sponges again that were pretty abundant deeper.
Does that green
Good copy. Thank you very much.
All right, Vidya, do you want to just do a quick zoom?
That looks like a
mat?
Yeah. Maybe it's a sponge?
Huh.
Looks like a sponge.
We can take a look at it if you guys would like to get closer.
Yeah, if you can maybe do a
come on out,
a zoom. There's also a polychaete worm.
We also have that same structure from before that we were very interested in.
You can see the arms kind of below of a brittle star that it's hiding under the rock.
I'll just pull you to me.
They usually hide their bodies under the rock and keep their arms out because they're waiting for anything that falls that they could grab and eat.
Oh yeah, I don't know if the fronts are going to do it on this one, but we can try them.
So yesterday we saw a brittle star on a coral. Was that brittle star feeding on the coral?
On that particular coral, we don't think so. It was just probably getting up higher into the water column. Maybe it's
Can you hear us?
Eating any of the organisms on that
Testing
that coral,
video, 1,
but
2,
also
3.
you know, higher above, he could grab the first kind of plankton or debris coming down from the water to feed on. So.
Okay, let's go ahead and zoom in, video.
Yeah, it kind of looks like a sponge. There's an old polychaete. Here's another one of those crinoids to our left.
Yeah, they're also looking at that guy.
Cythidium, I believe they're called. Yeah, that looks like a sponge. Yep, and there's one of those crinoids.
Oh yeah, look what's coming.
Oh, and another, looks like some type of crabalo.
Yeah. Okay, video, if you're clear. I'm gonna adjust my Z-bias just a bit. Thanks. So watch as you can kind of see in serious view over there what's coming up here.
Okay. Ah, yeah.
Is that full wide video?
It's pretty steep slope that we are
Yes, it is.
I'm gonna
now
come
going
up just
up
a bit.
to. Scattered with these yellow glass sponge.
Video, is that's full wide, right?
Yes.
I don't. Okay.
There are a couple crinoids hanging on to this wall-like structure.
Is
you
that
see
crack?
this
Okay.
crack. Looks like a demo sponge. The white, we see one of the pencil sea urchins in the front.
That's gonna be interesting. Still coming up.
Good copy, Bridge. Thank you.
So these sponges really like wall-like structures. We found these in deeper kind of wall-like structure, and now
Oh, it's like a little ridge, isn't
we're
it?
finding this right on top of this ridge.
What's that?
That's and these types of sponges you don't find in shallower waters. These are predominantly in deep water.
This would be a good angle to go check out that ridge as
Is there a theory why that would be?
These sponges are, actually, I am not too sure about the theory of why these sponges are only in deep water. Maybe of what they're composed of, as I mentioned before. They're kind of muscle tissue.
Probably.
It's not muscle, but its tissue is also known as spicules, are made of silica. So if there is somebody
do you
on
want
the
to zoom
line
on this
that
video?
is a sponge
Are you able to
expert
tell what?
in deep water sponges,
It's just
you want to chime in?
an old sponge.
This is like
Maybe
a dead
zoom out
sponge,
just a little bit,
or
so you can
it looks
fill this.
kind of alive.
Zoom in just a little bit, video. No, zoom out. All right, come on out. Full wide. Yeah, we'll look down this wall and then down that.
Wow, look at that.
Stacy, we often see solitary sponges, and here there are a lot on this wall. Is that just because this is a really rich habitat for them?
Yeah, this might be, you know, like, I'm guessing the currents are relatively strong in this area, so it might bring food source for these
Got a bottle.
these sponges, because these sponges are water. They
cool
filter
ridge,
the
though.
water.
That's kind of
They're
funny that
actually
it formed
really
like that.
important in shallow, and I'm guessing deep waters. There's
Yeah, I'm
some
gonna stop moving sideways.
well, some say that a couple sponges can filter the entire water
I'm
of the
actually gonna go along
Caribbean
it.
in a couple
I think
days.
we'll be
So they filter
just
a lot
fine.
of water. And we saw that there was another glass bottle, some other trash. So this is a pretty cool.
Okay, so everyone on shore side, our depth right now is around 607 meters. We're passing over some of the more dramatic hard bottom, hard substrate we've been seeing on this dive so far. Continuing to see sponges. I saw a Brisingid sea star just a few moments ago. Occasionally, we're seeing some holothurians or sea cucumbers.
Got a really good shot of the terrain over there on Sirius watch leads.
Yeah, that's right. Yeah, I'm not sure if the feeds are working to send to the shore side. I know there were some problems recently on feeds 2 and 3, but I think that hopefully we'll be able to send some screenshots. But yeah, the terrain is definitely this these terraced formations.
Yeah, thanks.
With rather large, 10 or so meters vertical,
yeah,
perhaps, and
back and forth.
Maybe 5 meters vertical.
So I'm going to follow it.
And then terraces every few meters or so, cutting into the side of the hill here. So we're mostly seeing sponges on these rock faces, although in the lower right-hand corner, we're passing a white structure on the rock there, just out of frame right now.
Sorry, Watch, do you want to take a look at it?
Yeah, if it's possible, and it's not too time-consuming to maneuver the vehicle, I just wanted to see what that was.
Not at all.
It looked like it was branching very low profile to the
other side, isn't it?
To the substrate, but it could be a type of fan or something.
I'm actually going to get on the side so we have a better view of it, watch leads.
That's ideal, thanks.
Maybe a little bit further back than normal. Okay, come on in, video.
Yeah, here. So there's been some discussion about this particular coral. We saw what looked like a smaller representative of it previously, but if possible, we'd like to get some tight zooms on the polyps on either the one on the lower left since it's facing us, or that might be better.
Copy that, lower left.
But this is
any more zoom video?
A type of coral. We need to make a positive ID on it, though.
Video, if you can latch me when you want to talk to me, because I don't have you latched.
I haven't said anything, but that is max.
I don't hear you, video. I'm sorry.
Looks like a shrimp.
If you latch me
on the base of the
there
colony
you go.
and the
latch me whenever you want to talk to me.
Yep, I was on EXRV. Sorry. That is max.
Copy
From
that.
a little bit further out, I was going to suggest that this might be a hydrocoral. It looked a little bit like a crypthelia.
Watch these. Do we need to try to get a little bit closer for this?
Openings in the center.
This is our max zoom here.
Swellings at the end of branches, which would suggest that this is scleractinium.
Yeah.
Yes.
I'm reasonably certain we saw something that looked like this, but I could see opercula a while back when we saw something like this. It definitely reminded me of crypthelia, but this, you're right. Once we get a better zoom on the coral structure, we can see if there are tentacles or if there's some structure to the polyps that might indicate that it's a scleractinian.
All right, watch these. Do you want me to move in some so we can get some better
that
zooms?
lower left side, we might be able to see if there's calyces inside those little pockets.
Yeah, to the lower left pilot, that colony that's facing us.
So this is our max zoom from here. Would you like me to move in to get more zoom?
The plates that divide up the inside of the calyx.
Yeah, we would like to get a little bit closer.
Okay, copy that. Come out, video. Now we're going to be looking more down on top of it when we come in here. Watch leads.
I think that'll be okay.
Copy that. Okay, come on in, video. I think we can kill lasers as well. Come on in, video.
Is this any better for the shore side? I do see very thin. Yeah, they don't look like scleractinian tentacles. They look almost like net-like or mesh-like projections coming out of the tips of the calyces there. Does anyone see what I'm seeing there?
Yeah, I don't see that, Steve. I'm not saying you don't see it. It's too small on the screen.
Do you want to try fog lights?
Yeah. I can't see into the cups well enough.
Okay.
All right.
Yeah, I see. I don't see well-defined polyps, tentacles on the polyps. They look more like the projections of a stylastrid, but like you said, we're bouncing a little bit here, and it's difficult to see.
Unfortunately, the background contrast isn't enough.
Yeah, there's not a lot of contrast to show that.
We can get a sample.
Yeah, yeah, it's tough.
Do we want to potentially collect this specimen
cool
to confirm later on? I think whether one way or the other, I think it might be it's representative of the structure forming corals at this site.
Yeah, I mean, certainly if you've seen multiples of them, it would be a good idea because then you could identify it.
Yep. So pilots, do you feel comfortable for grabbing one of these corals?
Yes, we can. Do we expect it to be real brittle?
Yes.
Sure, mine would be very brittle.
Float or sink?
Shatter, I think, pretty easily.
Yeah, it will shatter and it will sink. So it's heavy enough that it will sink readily. Yeah.
Come on out, video. We're gonna get set up for this. So I'm gonna have to try to approach it at a weird.
Yeah, these types of corals, these hard corals, they come in a few different varieties that we're trying to discern.
Could you hit my auto?
And one group are the hydrocorals and the family Stylosteridae.
Thanks.
And the other being scleractinians or the stony corals you might be more familiar with on shallow water tropical reefs. And we were just trying to discern which of the two this could be, and we're not able to get a very good image of the parts we need to identify, but we hope that with a collection of at least a piece of this.
And just so you're aware, all bio boxes are available.
One of these two colonies will help us greatly to identify this.
Okay, so we're going to go into port. Yeah, it's going to be the easiest. Doesn't really matter, I guess. I like it down here. Forgot about this. Okay. If you want to bring up Mini, and then we can move as needed. Yeah. Oh, perfect. That is throwing me off. Alright, what do you think? Think I'm going to be able to grab with this one? Okay, like more up. All right. This is such a weird angle. Maybe more like that. Force 4 is fine. Okay. It's the script force 4 and it's awfully slow.
Super brittle. It might just be the case where whatever you can get.
I didn't get anything on this one. I'm gonna go a little bit faster. And just try to scoop it.
That might work too.
Yeah, it's solid. Well, now I'm gonna have to scoop it. Okay. It's so brittle. Yeah, that's what I'm gonna try to do.
So for everyone on shore side, we're in a collecting operation here to try and
zoom
grab
on the main and then also on the mini.
Coral that doesn't have a firm ID yet, but we hope to better identify the organism on the surface.
Slightly.
And it was deemed by our science party on shore and on ship here that this is a good candidate for collection because it increases our knowledge of deep water corals in this particular area. because it appears to be a representative species of structure forming corals in this area.
Yes, please. It's just way too brittle. Nope. It's not going to pick up. Guaranteed. I mean, we could try scooping it. Come at it like this way. Yeah, that's better. Okay. What do you normally set the grip force for the scoop? Pretty high? Alright, okay. Okay. Why aren't we rotating? Ah. That's better. I got it.
Violet, would you like me to follow you on the mini Zeus?
No. Gotta go nice and slow for this. Got in there? Okay. Yeah, I'm not going to go any further up. Okay, yeah, now you can probably come out, I guess, for the getting in there. Yeah. Perfect, thanks. Okay. Can't really see it. Right there. Okay. I haven't used that scoop in a while. All right, watchers, we do have a piece in there.
Excellent. Thanks for your patience with that.
I think we're going to do a pilot shift as well right now, so I'm just going to stay here.
Everything turns really quiet when we collect a sample and everyone's holding their breath because it's a really difficult job. And what you can't see on shore is that the pilots are looking at a number of screens with different camera angles to help with depth perception.
So the pilots are doing a quick change out, and as soon as they're settled, we'll be on our way.
Gentlemen.
Navigator.
For your edification, we have one sample, the one you just saw.
Copy.
And for the remainder of your shift, we will be heading northwest to waypoint 3, which is a mound, and that's of interest, and we do want to get there. So.
Okay. So the rest of it should be pretty flat.
Right, and so we might see some relief when we get up here, but.
All right. What's that distance that we have to go?
600-ish meters. Let's see. Yeah, 607 from where we are now.
We have like 3.5 to 4 hours.
Right. Okay. Should be okay.
Yeah. Yeah. Okay. So we go and do a 30-meter move, and what direction have they been moving in?
They had
330.
They had been moving north, but I think that was just to get up this wall. I think our next one we'd want to do 300. 300. Yeah.
300. Okay.
Yeah.
All right. Yeah. Bob, feel free to zoom past the skid anytime you need.
Roger that. The camera is pointed down for the for this last collection, the last sample. You are now full wide pilot.
Great, thank you. All right, watch leads. We just had a shift change in the front row. We're just getting settled in here, and if it's all right with you, maybe we could do quick introductions of the new shift.
Absolutely, on your convenience.
Yeah. So short side, we just had a shift change in the front row. This is Dan Rogers, now on in the pilot chair.
Now sitting navigator is Chris Ritter.
And as co-pilot, Carl McCletchy.
Over on video, Bob Nutt.
And back in clipping is Caitlin Bailey.
And we're all with the Global Foundation for Ocean Exploration. Okay. Well, I guess.
Want to get that moving?
Sure.
Okay. What do you think? 30 meters?
Sure.
Y bienvenidos a todos, están escuchando. Estamos aquí sur de St. Thomas. Ahora la profundidad es 600 metros. Y vimos muchas esponjas. Aquí hay esponjas amarillos en la pared.
Nice.
Y vimos muchos peces en este buceo.
It's the fourth over.
Y saludos a Eco Exploratorio. Sí. Sí, está escuchando.
Anything of interest here, watch leads?
Yeah, can we zoom on those white balls that are projecting from the wall there?
Sure.
Just above the lasers?
It's like
yeah, I think those are tunicates as well.
We can do a partial video. See if we they want a closer look.
Sponges.
Do that.
Oh yeah.
They look like
these are different than the other stalk sponges.
Yeah, I can't tell if that stem is originally part of the sponge or not.
Kind of looks like it's like a crinoid with sponge attached to it at the end.
Yeah, something complicated going on there. The stalk seems to be coated with zoanthus as well and some brittle stars.
Okay, pilot set move is underway. 300 bearing.
Copy that. Do you need a closer look, watch leads, or are you?
No, I think we're satisfied with that.
Okay, we'll continue on.
I think we should do a round of introductions in the back as well. I'll start. My name is Stephen Oskovich. I'm one of the science co-leads on this expedition. I'm a PhD candidate at Temple University, and I specialize in ecology of deep water corals.
See the tip of the finger in the manip cam?
And to my right,
that shiny part
I'm Stacy Williams.
Okay.
I'm the other science co-lead.
Just in there.
I'm a co-founder of a nonprofit organization called the Institute for Socio-Ecological Research, and a for-profit called Coastal Survey Solutions, both based in Puerto Rico. I'm a coral reef ecologist. I primarily have worked in mesophotic reefs and shallow water reefs.
And then elbow all the way down, so
very
it can't
excited
sag,
to be on this expedition.
And then slowly
so
wrist
welcome
pitch into the little pocket behind the rock box. And then as long as it's not beyond the box, it'll be good for the hangar door, and then it's got a few inches before it hits the side of the box.
Sounds good.
Jaw pocket.
So I think those sponges are on stalks. There's another individual to the right there. That's the one we looked at. It's, just usually the stalk of the sponge sometimes looks fresher for some reason. I think.
Right, hydraulics are coming off.
That one looks really bio encrusted.
Here's some sea cucumbers.
Something.
Oh, here you go.
Yeah, so got something more substantial coming into view now. And the pilots are already making their ways to it, I think. So on this little bit of hard substrate, there seems to be some sort of coral, it looks like from a distance, sea fan. And we'll document this more closely.
We'll do a quick sweep of the lasers, and then we'll turn those off. Yeah, go ahead and hit it, Carl.
Aquí en el frente es un coral blando, pasible, con estrellas frágiles.
Can't seem to get the timing.
Y un pececito.
Have you had it work?
Yeah, you have to like hold it for a second,
but not too long.
Not too long. I think like about a second should do it. It used to be lit up, which made it easy to find.
Yeah, I don't know.
Let's try the fog lights on this one.
So that is
aft light bar. Yeah.
Vidi, you good for the front lights to try this shot?
So we're going to get some better imagery on this coral and be able to tell something about its identification.
Video co-pilot?
Perhaps, and as well as...
You're good for the front lights?
Yes.
All right, we'll try them out.
Something about its associates here. It seems to be a large brittle star.
Yeah, there we go.
Nice lighting.
Should we do a little still camera?
Sure.
It's, yeah.
You can go in tighter if you'd like video.
Oh, I've seen one like this before.
Hold that. That's a cool shot.
Where do we see the DSC?
So we're looking at now.
Sean will have to pull it up on his bottom monitor over there.
A type of sea fan.
I forget which router.
And right in the middle here, there appears to be a very large
the still cam,
stereochromated brittle star.
Yeah, that light does make a nice image there.
Now,
ROV still is on our router panel. You could do it monitor 12, yeah.
All right,
the characteristic of soft corals are usually that their polyps have 8 tentacles compared to that
that's the upper
of black
router.
Corals or hard corals,
see that?
Which could have 6
so let's get that back
tentacles.
to
multiple
Sirius.
Video will get that.
So I definitely think this one's an octocoral. At your convenience, can we pan through the colony from the base to the
I'm going to turn my heading to find you.
Sure.
Just so we have an idea of how it's attached.
Sure. Ship stop now, I assume.
Looks about stopped. Yeah, it should be.
Okay, there you are. Tether looks good.
Okay. Why don't we go ahead and zoom in closer on these polyps?
Max.
It looks like it has 8 polyps. So.
Yeah, eight tentacles on the polyps.
I mean, 8 tentacles on the polyps. Thank you.
All right, I'll try monitor.
Probably hundreds or thousands of polyps.
Polyps, yeah.
Yeah, I don't see RV still.
Much
on
more polyps.
Okay, it's one of those.
I have a
buttons,
guess, but I'll let our shoreside folks see if they can get a better view first.
And then it'll be black until we power it on.
So we have a request from a shoreside team.
Here we go.
The framing's off.
To zoom in. Pilot, this is watch lead 2.
Go ahead, watch.
Would you be able to zoom in a little bit more?
I'll have to pick up the ROV and move a little closer, but we can certainly do that. Let's come out video.
I'll turn it off so there's not too many.
Sure.
See what it looks like when you get closer.
Little like smudges on the Zeus in the bottom center left. You see that?
Oh yeah. The two or three little ones there.
Yeah. Okay, go ahead video. We're going to get a tight shot on some of these polyps here.
Excellent. I do see, no, that's just some discoloration. I thought I saw a small barnacle, but no, it's just some discoloration. So the colony does seem to have, it's mostly white, although some of the branch tips seem to be discolored, perhaps yellow or off-white colored.
Nice. I like that.
We'll let our shoreside scientists, if there's any thoughts on ID, let us know. Otherwise, we're just going to get some good imagery and move on. There seem to be some striations too along the skeleton, along the axis.
Digital still is secure.
Thanks, Carl.
Yeah, do you see that? This shoreside, you can see those striations right about now. Dark lines running up and down axis. And it looks to me like the bulk of the polyps are, they seem to have a small mound, a small lump of tissue at the base of each polyp. I haven't seen any retracted polyps, so I can't really give, oh, there we go. So it looks like the polyp is completely retracted into the coenenchyme.
Is there anything else you'd like to see here?
I don't think there's any more here. There's another brittle star that we just went past, so
there's a little
that's
crab or something.
Something there, yeah. Crustacean, perhaps a small squat lobster on the base. That's a pretty substantial base, though, for these types of corals. So I think we've got enough imagery here.
Okay, let's go ahead and zoom out video. I'll come back to you, Carl. And
yeah, Nav, what's the bearing now to the top of the mound?
So it's still about 300 degrees, which is what our last move was.
Okay. Yeah, well, I can chase you that way, pilot.
Okay.
Now, do you know why the Sirius shape is a circle instead of the Sirius shape?
The heading was off this morning, so
okay.
we didn't want to confuse
pilots
pilots by looking at HiPAX. We just made a circle.
So we just have to figure that out again.
But the heading on the GUI seems to be accurate.
Yeah, that's too bad. It was working for a while. If you go to hardware, is it turned on? If you go to administrator hardware, because we did turn a bunch off to do the calibration.
Looks like we have some demo sponges kind of scattered along the seafloor.
So it's over in the on
couple
the left
fish.
screen, the administrator.
So we're not seeing as many sea pens as we did earlier on, but there still is an abundance of soft bottom that sea pens or other soft bottom dwelling corals could inhabit as well. But it seems like the holothurians are dominant in this area.
Yep.
Yeah, Carl, I'm not exactly sure where to go on here.
Well, the shortcut's a little microchip looking thing. It's a little for hardware.
Oh, yeah, it wants to be to completely close survey.
Oh, no. Yeah, so you can't look at it in this version. Yeah, never mind. We'll figure it out tonight. It's better not to anger HiPAX.
Well, I guess maybe we'll get another move ready. Been moving it. What was the last one?
Looks like
4-0.
300. Yeah,
Or 3-0.
Yeah, I'm still
last
going.
one was 300.
Okay.
Yeah, we could try. It's pretty flat. We could do like maybe 50-meter moves. Just back up if there's a sample or something.
Sure.
Sounds good.
I think the fish would be on the mound.
Copies.
So now you're at 550
meters,
94 meters.
300 degrees,
Gently
decimal 2 knots.
getting shallower. The temperature is now 11 degrees Celsius.
Oxygen has declined actually since we reached bottom, so it's actually getting lower as we come up here in depth.
Can I take the mini Zeus?
Sure.
Is there, didn't it used to, the desired position was something like that?
Yeah, and then I think Roland likes to see between the toes, so that the bottom is just above the front of the drawer.
Okay. Yeah, I mean that's good for piloting too.
Yeah.
Trying to get something in between.
So for the most part, we've we've transited the steepest portion of our dive today, and we're going to be heading over some moderated slopes.
The minute cams seem kind of dark.
Pilot, in the lower left.
Go ahead, watch lead, sorry.
Sorry, in the lower left, there are 2 stems.
Yeah, I see that.
Yeah, either one of those could be something we haven't seen yet.
Okay.
Just wanted to get a closer look. So like I was saying, we were heading over some more moderate terrain now as we proceed towards a large mound feature that's towards the end of our dive track. And our depth now is about 594, and over the next, well, through the end of the dive today, we'll be ending at around 465, so covering only about
go ahead, video.
100 and a little over 100 meters of vertical left to go.
It's a little one.
Yeah, so we got a couple things here. We have a brittle star and a black coral.
Can zoom in tighter.
I think there are actually two here. One to the background. It's, yeah, it looks to me like this is a branching black coral fan. Rather highly branched as well.
Is that max zoom video?
Yes, it is.
Okay.
You know, I don't immediately recognize this particular coral, but we have some folks on the shore side that should be able to provide us some better identification here.
I've definitely started moving in my...
Getting out of focus. There we go. What's that, Carl?
Definitely started moving in my 50-meter move.
Okay. Are we going to want to stay here longer, watch lead?
No,
Or
I think,
yeah,
just
ahead.
As long as we get some good images, and I think we have, then we can move on.
Okay, let's come out video.
What did
Black
they identify
coral.
those as? Oh, black coral.
Yeah.
I thought they were sea pens.
The colonies are, yeah, about 10 centimeters or so in height. There seem to be 3 small ones in this area.
Yeah. Sea pens are coral, but not necessarily black coral, is that right?
Yeah, they're squishy, I think.
Soft corals.
Soft corals. It's a better word.
Okay, pushing ahead.
There's a few rocks in sonar, very small. If you want to bump that out to 50 meters, see.
Yeah, they're numerous here, so yeah. I think these other stem-like structures we're seeing are also these black corals.
All right, bump sonar to 50 meters.
Excellent. It looks more like debris, yeah. There's a lot of sargassum here on the seafloor that we noticed yesterday and today, and it looks like it's three-dimensional structure, but it's just phytodetritus.
Do you think it would be? Well, I guess you're gonna make it there anyway.
Like is that yellow blob-looking thing a sponge?
Or
looks
well, they're not necessarily big, but
would you like to look at this watch lead?
Yeah, can you do a snapshot, please?
Sure.
Thanks.
Okay, go ahead, video. Do a snap zoom.
Ah.
Oh, it's a terabellid. Terabellid polychaete, I think.
Polychaete or a cnidarian? Looks like it could be a cnidarian.
I think it's a polychaete. The terabellid polychaetes, they look like you know, an orange tennis ball, and they have lots of. I don't think it's a cnidarian. Second guessing myself now, but no, I'm going to go with polychaete.
I've come over 30 degrees. Starting to lose you.
They actually swim. The terabellids can swim by kind of oscillating their the top part of their body, the oral part of their body.
Do you want me to pause it?
I think we're okay. I'll just shoot back over.
Copy that.
Well, I think that answered our question that doing 50-meter moves, we shouldn't have you jog over far to the side to look at rocks, because it happens quickly.
Yeah. What does our average pace need to be to get to waypoint 3?
Stay by one side.
Sure. There's an outcrop over there on the left.
Oh yeah, Nav, did you get it passed on to drop targets every 15 or 20 minutes? Like, you could choose to do it on outcrops or...
I did not. Should I do that?
Yeah.
Do you want me to mark like features? Or should I
Yeah,
What
that's
is
interesting.
that? Oh, it's a jug. Yeah.
Debris.
Yeah, some glass bottle, large glass bottle.
You want to do a snap zoom video?
Yeah, Nav, just anything you'd like. If there's no feature, you know, every 15 or 20 minutes you can just mark.
And then I'm marking D2's travel?
Yeah.
Okay.
So it looks like we're looking at a large glass jug of some type here. No ships in there, but you know, at least there's a gastropod shell, a couple of them.
Okay, let's come out video.
And yeah, pilots, we're looking about 0.08, 0.09 knots per average.
Okay.
And these moves are we going at 0.2?
Yeah, that's correct.
Okay. So 0.2 half the time.
Yeah, so. Alright, pilot, you've headed over to my port side. I'm 10 degrees over.
Okay. There's a nice little vein of hard substrate here.
So we did manage to get a tentative ID on those black coral sticks we were seeing coming out of the seafloor just a few minutes ago, and they're likely stylopathes.
Get to the
or
top
close
fit, come
relative.
back over.
Oh, that's a big urchin.
Yeah, that's.
Would you like to take a look
sea
at that?
cucumber. It's a different species of sea cucumber, though.
Yeah, I don't think we've seen that one so far.
Okay.
You didn't get a twofer in this shot.
Yep.
I think I'm going to sit
Seem
down
to be
for
lucky
this one.
today with these 2-for-1 shots.
Stand by video.
So estamos viendo un pepino del mar y un erizo
That looks to be about the move.
en la profundidad de 592 metros.
Yeah, so sea urchin, possibly in a kind of thurid. And a new species of holothurian sea cucumber to the bottom that we haven't seen yet on this dive.
Let's zoom in on the urchin first.
Again, you could see the 2 feet kind of floating in the, well, not floating, but waving in the water. They use this 2 feet to attach to the substrate or attach something to camouflage itself. And what's interesting about 2 feet is that they have a water vascular system where they use seawater to extend or contract these 2 feet.
Okay. We can go a full specimen shot on the urchin. So hold that.
There also may be off to the left-hand side of the urchin we're looking at now, another urchin that one of the shoreside scientists has seen.
Okay, we'll pan over to the left after we take a look at this Thurian here. You can go in video if you have it. Okay, go ahead and zoom out.
So these guys also have 2 feet, but they're not really well defined. They're on their ventral system. So we could kind of see all the way to the left, the 2 feet attached to the rock.
Yep.
If we zoomed in, but
I have
yeah.
stop pilot.
Great.
There's possibly a sponge or a coral
Right,
in front of these things
back
closer
here
to
with
us.
Really? Okay,
yes.
you want to come out
Cucumber?
to a partial zoom?
It'll be white on the substrate right there.
Do you see the...
I don't see another urchin.
Right here.
On the bottom of the screen.
Bottom of the screen.
Yeah.
Gotcha.
Yeah.
Okay.
There's 2 of them. There's one on the sediment.
Okay, go ahead and video. I'll back up a little more. Okay, you can zoom in tighter.
So this looks like to be one of the deep sea long-spined urchins.
Hold that.
I have to remember the name of them.
It's like diatomidae or something. I think close to that. Are they in the family of?
Yeah. Is there something on that urchin as well? Looks like there's another organism in the upper right corner of the sphere. Yeah, I can't tell what it is right now.
You can zoom in a little tighter.
I think.
That's
yeah,
max.
that's it. Yep.
Okay.
Or that may be just part of the...
Might be it could be the anus.
It looks like it's chomping down on this sargassum here. Definitely an important food source for the for benthic critters down here. Okay, thank you, pilots.
Okay, let's come out full wide.
All right, so I've come 30 to port.
All right.
And I'm kind of looking down, so you have plenty to go starboard and ahead.
Hey, this is Chris Ma.
Hello, Chris
Hello,
Ma.
Chris.
Hello, Steve and Stacy. How are you today?
Pretty awesome. How are you?
I see you're checking out some of my favorite animals. So the word you're looking for is aspidodiadematid.
That's the word.
I don't know why you didn't just remember that.
I know. Shame on me.
So it could be one of 2 or 3 genera from this area, either Aspidodiadema or Plesiodiadema. And that was pretty neat that you watched it. I think it was feeding. Didn't get to see pronounced anal bulb, but still getting to see 2 urchins, a cucumber, and that brittle star. It's a nice sort of compendium. Also, we got to see the little hoof-like spines that the Echinotheriid uses to walk along on the bottom.
Get a better shot
There's
of
a
this,
fringe of spines directed along the oral surface that are horseshoe-shaped, and they use those to walk along soft bottoms.
Yeah.
So, and someone was showing me the star you saw earlier today, a slime star in the genus Pteraster. So, some definitely wonderful echinoderms that you're seeing today. And looks like you've got lots of nice Lebensborn and a lot of tracks down on the sediment as you're moving along. So a fine day for some deep sea invertebrates.
Agreed. Well, thanks for providing that context, and I look forward to
fog lights on this
having
guy
your
would help.
input if we find any, particularly noteworthy
Looking
things.
forward to the next thing. Thank you. I'll talk to you guys later.
All right, take care.
Seems pretty blurry. Yeah. All right, trying fog lights.
Oh.
There's a fish.
Photobombed.
Barble or beardfish, right?
Changing lights.
Yeah, beardfish.
Beardfish with barbles.
It's a little more detail there.
Yeah, it's a hard thing to image.
So this was Terabellid polychaete. It's actually a worm, funny enough. The body is most likely either on top of the sediment or in the sediment here, but what you're looking at is the oral surface, the mouth parts.
try to focus on that. It's awfully.
Yeah, right? It has very little contrast.
Yeah.
Yeah. All I can get is a couple of the feeding tubes in the center.
It's interesting.
And
put in a move when you're ready.
If perturbed, these do have the ability to
meters, 300.
move. They oscillate their this disc here, and they can actually move and swim short distances.
So the shallower water species of this are called spaghetti worms, and a lot of times they have these way longer tentacles that look like spaghetti. So those are also.
It's mesmerizing. You can watch the food particles like travel along the filaments. Yep. There's actually a video online of one of these swimming. I think that the EV Nautilus took a while back in 2014.
True.
I think in this same area.
High beams.
Sure.
But they're definitely characteristic of the soft bottom fauna in this area. So I suspect as we traverse this area of low slope, we'll see many of them.
All right. Headed 300, camera's at 45 degrees.
Copy that.
Everything's looking good.
Just gonna meander back and forth until we see something of interest.
Nav, this is watch lead. How is our track for getting to the waypoint 3 on schedule? Okay. Excellent, thanks.
The tracking seems really good and the calibration.
Yeah, it does.
I almost feel like these aluminum ones have always behaved.
It might
You
even
could see all the tracks
software.
in the sand. This
Sure,
could be
not
from
necessarily
all the mini sea cucumbers that we've seen throughout this dive.
Sea cucumbers, urchins, gastropods. Chris Moss had a word, Lebenspuren, that was, it's not a word we hear very often, but it's a word that is used often in the deep sea to mean signs of life. And I think it's a German word. And yeah, it's definitely something that when we look at photos or videos of the seafloor, if we can't see the organisms that are in the area, sometimes we can see, fecal casts or tracks or mounds or anything like that. I'm noticing that the fish do not like this particular area as much as they like that kind of more moderate slope.
Yeah, and we were told that for these queen snappers that we should look in the periphery because they do tend to shy away from lights and hide under ledges.
This is new for us anyway.
Can we zoom on this outcrop here?
Sure. Sit down for a minute, Carl. You just let me know if I need to.
Okay.
Go back over.
Still aimed 300.
I think there's a sponge off to the left-hand side, that white blob structure. Is that one we've seen? It doesn't look familiar.
Think so kind of looks like a geodia. This is a demo. It looks like a demo sponge.
Okay. When I get lasers, Carl, and video, you can zoom in. Good.
Lasers are off.
Yep. Great. Hold that.
Oh, there's a couple things here.
Yeah.
Let's come out just a touch.
So that could be geodia.
Geodia or
Perfect.
Xenia, possibly. We have a couple land-based scientists. It definitely is a demo sponge.
So I've also noticed to the left and right there are these small branching coral colonies,
What do you think about stopping the ship here and then moving me towards this line of rocks?
I also saw
doing that.
Sure.
To the right and to the top of this rock, there are some attached coral fauna as well.
Video.
Yeah, because I see rocks 25 meters ahead of you.
Okay, let's go ahead and zoom in on this here. Hold that.
Yeah, so the one that is on top of the rock here looks very much like the one we collected, this stony structure, possibly a stylastrid.
Surprised, do you want to...
Let's come out a little bit video.
A bump to the west then?
Hold that.
And I've also been seeing these small octocorals on the surface of the rock.
Or if you want to...
Or move me to D2, I guess.
Move you to D2, so I'll do a move 15 meters to 215.
So, pilot, immediately to the south below that white stony coral, there's this kind of feathery white coral fan. Directly below, on the center of the screen now. It's in front of the structure in the background.
Oh, I see it. Yep.
And I was wondering if you could get at least a zoom on that.
That's good. Hold on, actually.
And maybe the fog lights.
Yeah. Okay, let's go ahead and zoom in. And do you want to activate the high beams, Carl?
Yes, they're activated.
Yeah, so there's a couple things here. That looks like another stylastrid in the background, possibly. And then I'm not quite sure what these very small octocorals are that are all over.
Just a little bit, yeah. Hold that.
All right, well, I don't think we're going to get much better than that, but I think that's good enough for these.
Let's try one more time video. Go ahead and zoom all the way in.
Max.
Yeah, that's max zoom watch lead.
Yeah, I see. Okay. And in the foreground, the coral that's in front of it, can we focus on that? Might be more difficult. Okay, well, we got some imagery of it. Yeah,
okay.
I see
Let's
that.
go ahead and zoom out video.
Looks a little bit different.
Yeah, that might be a tough shot, so
yeah.
Try and see if we can move around and see another one.
Saddle around the left side here.
I'll turn the high beams off.
Sure.
Nav, are we good with the HPU? Alright, so I'm still... That move's probably about over or still going?
I think it's just about done.
Yeah. I guess I was hoping that would get us to the rocks and then we could aim back uphill, or back up to the mound. I'm not sure if it... Of course, it all depends on where the rocks go.
Do you want to maybe do... Yeah. So the rocks are pretty much in front of D2, right?
Yeah.
So D2's looking west. I could just bump you west now. 15 meters. Or just another bump.
I mean, this is, you see, I mean, there's no other rocks aside from this one in front of us,
so
right?
I guess you can just go.
And then we'll just, maybe we'll let Sears settle out, and then we'll just find uphill and do 50 meters or something.
Yeah, maybe.
Those are more
you can stretch out a bit.
Small stylastrids.
Anything you'd like to zoom in on, watch lead?
No, nothing in particular
there's a
here.
fish.
Yeah.
Upper left. Those beard fish.
Go ahead and zoom in video.
The small stony structures in the foreground look like the corals we've been zooming in on for a while, and the one we collected.
Hold that. It's quick.
Oh, there's a shrimp. Oh.
Or a fish.
Small fish.
Thanks, video.
Fast moving shrimp. Usually shrimps move backwards, right?
No, the shrimp. There's one shrimp right next to the behind the sponge right now, and then that was I think a small fish.
All right. Well, if there's nothing you'd like to zoom on here, we can continue to make progress upslope.
That's right.
Yep.
Okay. Let's go full wide video if you're not already.
Yeah. So there's a tall rock like 15 meters ahead of you.
Okay. At my current heading.
Yep.
280. Yep. Okay.
Just a good direction.
So I've been keeping a mental log, and oxygen is still decreasing through the dive today. We started out at around 2.9 milliliters per liter. It's becoming progressively lower, 2.7 now. Temperature has increased slightly over the course of this track, and we've traversed about almost 200 meters from our original start depth.
Yeah, we'll make this next move a 50-meter move and cover some ground.
That's 200 meters of vertical, not horizontal. I think we've
so that rock wasn't
we've planned about a 900-meter dive track for today, and let's see where we are.
At this point, do we want to realign to 300?
About 2/3 of the way through it.
Headed that way.
Copy that. And then when you're aligned, I'll put in a 50-meter move at 300, 2 knots.
Sure. You can go ahead and call it in, and we'll be there once it starts.
Copy.
What's that? Quick zoom here, video, partial zoom.
Okay. Up. This looks like.
Hold that.
Crinoid. Yep. This is a stalked crinoid. Okay. And right to the right is a small octocoral. We're seeing predominantly two types of crinoids, stalked crinoids and ones without stalks.
Okay, so yours is at 300.
Great.
There's something ahead. It might be a sponge. So this looks like maybe another stalked sponge, glass sponge.
Copy that.
Okay, go ahead and zoom video. Hold that.
Oh yeah, it's a glass sponge, and it looks like there's a brittle star underneath, attached to the stalk.
Yeah, so this looks like a Hyalinema, or close relative of it.
Okay, and zoom out.
Great, yeah, we're satisfied here.
All right, I think I'm moving.
Great. Go ahead and zoom in video. Oh, yep, it's quick.
So for those of you watching on shore, periodically, you might hear some noise in the background. Currently, they're conducting a fire drill. So I'm not noticing very much
25 meters ahead.
live megafauna here. Occasionally we're seeing fishes, very, very small fishes near the bottom that are very good at eluding the cameras, and then some sponge stalks.
Main deck fire team, muster to your secondary location. Main deck fire team, muster to your secondary location.
Fish to the right.
And for those who are tuning in, this is the Okeanos Explorer. We are presently south of St. Thomas to the east of Vieques Island, on the border of a marine reserve called the Marine Conservation District, also known as the MCD.
Okay, zoom more.
This area is protected from fishing.
And hold that.
Since 1999. I believe we've seen this fish before.
Yeah, I may have
earlier
been
in the dive.
I may have been at lunch. I don't recall this one. Pretty iridescent blue colors. Okay, thanks, pilot.
Okay, let's zoom out video. Line back up and push ahead.
Yep, that rock is about 15 meters in my direction now.
Great.
For the direction I'm facing.
A lot of sargassum debris in this area.
Yeah, there was an impulse of sargassum over the summer.
Oh, really?
Yes, that washed up depending on where on the coastline, but a lot on the east coast. So that's why we're probably seeing a lot of sargassum debris along the seafloor.
And sargassum is a algae that grows in the open ocean?
Yeah, this particular species grows in the open ocean. There are benthic species that will grow on the reef, but this species grows in the open ocean.
Interesting.
This looks a lot like the topography we were seeing yesterday. Occasional tables or outcrops of carbonate crust. Yeah. Can we zoom on the ledge there as we pass? Or
how much do we have left in that move, Sean?
About 15 meters.
Okay.
Ventilation has been secured to the hospital base. Ventilation has been secured.
Okay, let's get the high beam zone.
High beams are on.
Yes. Okay, let's go ahead and zoom in video.
So So what we're looking at right now are a couple of things. There looks to be a type of stylastrid coral. Some sort of stylastrid coral on the rock surface, and then a black coral here, more sparsely branching, but one that we have not seen yet this expedition. This could be...
Pilots, do you want me to pause that move?
Do you think you'll settle out over D2, or should we stop it early?
I think this could be some sort of antipathes, possibly.
So
black coral.
Cirrus might settle about...
Has a rather large brittle star here.
Okay.
So
my camera
and a small little crab.
Oh yeah, and a squat lobster.
Squat lobster.
Pilot
or
if you think if pilot thinks it'll be here a while.
Looks like a chirostylid crab.
Watch lead. This is pilot.
Yes, pilot.
Would I be correct in assuming that we'll want to spend some time at this rock, or do we want to just spend a couple minutes and move ahead?
Yeah, I think we're just going to scan along the ledge.
Let's come out video.
Yeah, around
Marshall.
As best we can and move on.
Okay, we'll keep it moving.
We're currently having some trouble, some lag with the shoreside scientists, so we want to give them a couple extra minutes to see what we're seeing in case they want
saddle
to
along the
make
right
a decision.
side, and I'll push out ahead.
On the far right edge of this structure, there seems to be a cluster of something that I would like to take a closer zoom on, but we don't need to set down.
Okay. This here on the right side?
Yeah. Yeah, I'm
Okay,
not
let's
sure
do a
what that
partial
was.
zoom video. Hold that.
Could we get a closer zoom?
Yeah, we can zoom in. Hold that.
So this looks like a mostly dead scleractinian coral. I don't see any live polyps or where there could be live polyps. There's a few to the lower left. There's a few solitary cup corals, but I don't see anything on this particular scleractinian that would indicate that it's alive, but it's good to document. Yeah, I think those are zoanthids on the right-hand side. I don't see anything that could indicate that it is currently alive, but we haven't seen any scleractinians this substantial living in the immediate vicinity or on this dive so far.
Right on the right-hand side, right in the center.
Tough to say.
Okay. What do you see?
Well, we got a bunch of little nodules coming out.
Can we look down a little bit more?
Yeah.
Yeah. Let's come out video. I'm just going to touch down here.
Yeah, very tiny sea star, yeah.
What do you see, watch lead?
Can we just zoom in one more time and then we can leave?
Sure.
It's very fouled, so I don't know if we're going to be able to make a very strong identification here, but whatever was living there was most
more zoom?
probably a type of scleractinian.
That's max.
Great.
Okay. Thanks for that.
Okay, let's zoom out.
All right, well, Sirius just stopped with you at the bottom of my screen.
Okay, I'll push
Hopefully
out.
we'll find a
get another
live
move in.
High beams
representative.
coming off.
Thank you.
It looks like there might be a sea urchin crawling. Looks pretty big.
All right, watch lead, so I'm going to push ahead and try to cover a little bit of ground. But the ship is stopped, so if you see anything of interest, just let us know and we can stop for you.
Okay, thank you. Sí, bienvenidos, saludos a todos. Están escuchando. Estamos sur de St. Thomas y el objetivo de hoy es para buscar chíos en aguas profundas y como chíos de ojos amarillos y de cartucho también. Ahora estamos en 574 metros de agua.
Yeah, I see. Your sonar has something
Y
off
en
in the
el
left
frente
far.
de cosa amarilla es un gusano.
Yeah, I probably should
Yeah.
push ahead.
Ignore that one. So
Yeah.
happy with 50 meters, same direction, 300.
Vimos
Yeah.
muchos crinoides y
So
corales
tempting though.
chiquitos.
Go over there. They don't seem to run in a line, so it's just kind of random.
En el frente ahora estamos
That last table
viendo
feature was
a
interesting.
crinoides.
It'd be nice if we could change the HD cameras remotely.
The settings.
Yeah. I wonder if they'll upgrade to that sometime.
Yeah, that'd be nice instead of having to plug it in on deck.
Yeah.
Okay, pretty well stretched out now.
Yeah.
Fire's under control. The fire's under control at this time. Continue extinguishing operations and follow up with a fire watch.
It's a long stock there.
Oh yeah, wow.
I don't know if I have enough tether to get out there, but be there soon. Seems like you're underway now.
Yeah, I think you can
push ahead a little bit.
Yeah.
Looks like something's hanging off of.
Yeah, let's take a look at that.
Very tall stock just to be out there on its own.
Oh, it looks like a C-pen.
Wow.
That is a really long C-pen.
So I'll do a sweep with the lasers. Maybe get lasers off.
Lasers are off. Lasers are off.
We'll get a few seconds of a full specimen shot, Bob.
I think this might be a Helipterid, possibly. But we'll get a zoom and see if which way the polyps are orienting. If at least one brittle star on there. Polyps in this individual seem to be coming off of all sides. Can we try to pan up and down or tilt up and down the image to
Sure.
scan the colony?
I'm going to tilt down to the bottom, and then I'll slowly tilt up to the top.
Yeah, so these C-pens are well adapted for soft sediment life. They anchor into the ground using part of their anatomy. Is this more or less at maximum zoom as well?
Yes.
I can move closer after we're done with this tilt.
Sure. So there's at least 2 associates, 3 associates, 3 brittle stars.
All right, I'm starting to look down at you, pilot.
Okay.
All right. Can we hold here for a second, kind of at the tip?
Okay.
I think this might be good enough. Yeah. There we go. Perfect. So it looks like there are some pretty prominent spines coming out of the polyps as well. Those might be the sclerites.
Can zoom in more, Bob, if you got it. It's nice.
Fabulous. Incredible detail. You can almost see, or at least we can almost see the sclerites in the tissue from here.
Like the lighting on that, Bob. It's nice.
Yeah.
I'm going to tilt up to the top.
So these are deposit feeders. That's why they have their polyps open. They're waiting for any type of plankton or debris.
Okay, I'm looking full back.
Okay.
Yeah, you can really see the 8 tentacles here with the pinnules.
We still have quite a bit left to go, right? Yeah. Okay, let's zoom out video. Are we happy with this watch lead?
Happy there. Thank you for some great images.
Let's hold that for a second. Just get like a medium view shot here. Let's come full wide. Get a nice departing shot, and then I'll shoot ahead. Okay, coming ahead.
300.
Yeah.
For those watching on shore and for our scientists ashore, we're at around 565 meters right now, heading up to a depth about 460 meters, so about 100 meters of vertical left to go. And we're transiting a fairly low intensity slope right now on our way to a large mound feature where there's suspected to be deep water fishes aggregating. We began our dive today around 772 meters in an effort to try and understand how these deep water fishes are distributed. We started to look at the lower end of their bathymetric range, and we haven't really found any evidence of occurrences yet. But as we move shallower, we'll be more likely to see these commercially fished species.
So we'll get someone, yeah.
Of course, all the information and data we're going to be collecting today is used by local resource managers, many of whom are watching right now, to help better understand how deep water fishes respond to habitat in the area and how they're impacted.
Yeah, a lot of what is known about these fish occur when fishermen catch them. So this is a really unique opportunity to see these important, or commercially important fish species in their natural habitat. And some of these deep water snappers, like the silk snapper, when they're small and they're young, they tend to be closer to shore, usually along the shelf edge, and then when they start
looks like
getting bigger,
they
move to these deeper habitats.
Copy.
We'll stay here for a minute until we get swapped out. Any interest in this urchin here?
Looks like an urchin.
I think it's a sponge.
Is it a sponge?
Debris. Sponge debris.
Tricked me. All right, zoom in.
We were looking at one earlier, and it looked like a coconut, so it's gone from coconut to sponge to urchin, back to sponge. So.
I don't know. It kind of looks like it's
zoom in tighter, video.
Moving.
Is it moving?
I don't know. I think it might be a sea urchin.
Oh yeah.
Yeah.
Hey. Wow, that's some intense camouflage.
So it's like fully uncovered in sediment.
Yep, there's a shrimp in the upper right-hand corner.
You can see all those small little, I'm not sure what they are, but you see them kind of buzzing around the base of the spines. There's a lot of them.
Yeah. Some like maybe mysid shrimp or some type of crustacean.
The tube feet are in there as well. They might be the tube feet.
Yeah, he's, I think, talking about all these like small little things swimming all.
That's right.
You want to hit joy lock, Carl?
Wow, that's almost completely covered in sediment. Huh.
And this is I think we haven't seen this species of sea urchin and so far these dives.
I couldn't even tell because any features are completely covered by sediment.
Okay.
Yeah, so this is probably a type of deposit feeder, eating small invertebrates in the sediment or other organic material in this floor sediments. All right,
okay.
Pilot,
zoom
thanks.
out video.
Yeah, man, it looks like a lot like those sponges that they just fall off and roll downhill and they collect sediment.
Hello.
Hello.
Hello, Chris.
Hello, Steve and Stacy. This is looking at your very interesting spiny urchin in the sediment. First of all, I just wanted to point out it's leaving behind a trail.
Oh, that's right.
Yeah.
And so this is what's called an irregular urchin in the larger groups known as the spatangoids, sometimes called the heart urchins. I also took a quick jog out to Possum has a wall chart of echinoderms from this area, of deep sea echinoderms from this area. So I ran out and took a look at it, and there's one that's kind of like this, very spiny, called Lineup Newsteeds. So I think this is what it might be. And so basically these are odd urchins that the word irregular refers to the fact that they're actually, in addition to being pentamerally symmetrical, they're also bilaterally symmetrical. So there are, they move forward, and if you look at the skeleton, they actually have a right and a left side. So, and these have a series of different modifications that they use to move themselves through unconsolidated sediment. And they feed by ingesting the organic particles in the sediment, in the sand or what have you. They're very spiny, but they can be very delicate. And in the Pacific, there are some species that are so fragile that they will literally come apart in your hand if you pick them up. But you guys had a really nice in situ observation of this irregular urchin. And I hope that, and there's lots of things like that live in the sediment that are, sometimes
50 meters.
We know them only from a couple of specimens, and I suspect that we'll get more interesting as we get deeper. So I'm glad you stopped for it. And thanks, and if you're looking at all of these trails and tracks, it's urchins like that, and maybe other sea cucumbers and whatnot, which have likely caused them, and provide some some interesting texture to the bottom of the sea.
Yeah.
Thanks. I'll be definitely keeping an eye open for more of these
rod still there? Can you
bottom things.
Thanks, Chris. Thank you for all that information. I'm glad to hear that there's another huge fan of echinoderms. I myself are definitely root for echinoderms, both shallow, mesophotic, and deep.
If you haven't been to Okeanos before, you'll get to know my voice real well.
Okay, that's good.
Pilot.
Everybody in the control room knows who I am.
No complaints. No complaints here.
So Nav, are we? Did we do 50 at 0.50?
That's correct.
Thank you to everybody again. Always happy to hear everyone's voice, even if I don't say hello to you on the phone. So
up here.
I'll speak to you later. Bye-bye.
All right, thanks. Bye.
Yeah,
bye-bye.
You're moving. How deep are you? Let me say.
So I have to go back to that original observation of that, what we thought was a coconut in the beginning, and take a double look at it.
But I think I did see an osculum in that, so I think that was a sponge.
Left on the bottom?
Yeah, it didn't look like a sea urchin.
Okay. Yeah, the track is a good a good indicator that it's living and moving.
D2 distance, about 400.
Look for that next time.
You got to average 0.07, or 0.08 if you round up.
So we're looking at
that's good. I think it was
kind of this low
0.09
slope
before. So.
Angle seafloor right now. There's a couple
slightly
of
steeper
live things here and there.
Steeper is a relative word.
We've seen some
50
Hyalinema
has been
sponges.
making ground. It's tough because almost every single time Sirius passes over.
Some sea pens, occasionally some
there's
fishes.
Always something, so.
Yep. We can always stop it too, and it's shallow, so there's no layback really.
We should probably do that because I passed over a few times.
Can't tell.
So you think you're
it's
underway?
A dead sponge stalk with
anemone
on it.
Nice.
But now I see
now they're everywhere. Yeah, you can see.
At least
I'm sure, Chris, you're watching, so you see all the tracks.
Actually, until just now.
The ground faults on D2.
They're good. They've been
sure they won't take it
all day.
Yeah. Sure they won't take it personally either that you're calling them irregular.
Co-pilot one out.
They look very normal to me.
Well, while we're here, video, let's just do a quick zoom on this fish.
Okay, coming in on fish.
There you go. I think this is a new monitor, so this one looks different than the rest. That's why. Different aspect ratio.
I think we saw this earlier. It was.
And it's like brighter. The backlight is like newer, I think.
Yeah. So video, we have some shots of this. I just wanted to check it out.
Yeah, it looks great.
Whenever
50-meter move, Levi. Yes.
Whenever you have your nice shot, can go back out. It's kind of cool looking.
It's cool looking.
Yeah, we need to keep ourselves entertained a little bit as we cross this
I think it's coming at me.
expansive
sea
seafloor.
A lot closer.
It's out of the lights.
Okay, coming out.
I was just going to call for the high beams, but...
Oh, gotcha.
He's out of frame.
There we go.
Yeah, he's gone.
Should get like a car high beam switch.
I mean, we could.
Add him on the new joy box.
Yeah. Put blinkers on D2 while we're at it.
So I think we're at 310, is that correct?
Yeah, 310 is the bearing of the move.
I'll head to port a bit.
I'm at 305 right now. I'll come over a little bit.
What do we have here?
Oh, okay. We'll take a peek at these if we have time.
Of course.
I think some shoreside folks might be very interested in these. I can already tell what it is. So I think there might be, the front two colonies might be the same, but there might be a different one in the back. So I guess let's take a look at the one on the right first.
Okay. Okay, video free to
Looks
zoom.
like we have a coral here. We're going to take a look at some different characteristics.
Okay, coming to port.
Yeah, so it looks like we're looking at an octocoral, although the polyps look retracted on this particular colony.
Okay, let's go in tight video.
Okay, coming in tight.
Wiggling around a little in the mud, so apologize.
That's
Tilting
okay.
my camera down to 60 degrees.
Copy. I guess once you get to 80, we can just stop because it'll happen quickly. They want to see one in the back too.
Sure.
Yeah, so this is looking like...
Okay, is that max zoom video?
I believe I have a little bit more left.
This is looking like an octocoral.
Coming in.
Okay.
Probably from the genus Calagorgia. The polyps are occurring in whorls.
Come back over.
With, I see between 3 to 5 polyps per whorl or so.
Down to 80 degrees.
Do you want me to stop the boat here?
And yeah, that's right.
Easy stop.
The polyps are...
Bridge is RV nav.
Oriented.
Yeah, we'd like a easy stop here, present position.
Looks like the polyps are oriented
Thanks.
towards the branch tips, which is another characteristic we can use
Pilot,
here.
will you tilt me up? I think we'll have a darker background up there.
There are several different species of Calagorgia in this area, which is why
just a little better.
It's helpful
That's
to
better contrast there.
get some input from
I'll stop. Thanks, Bridge.
everyone here, but
Stop, tilting down to 95 degrees. Everything looks okay.
Okay, watch lead. Anything else on this colony?
can we take a look at the brittle star?
Sure. I'll pan to the right. And co-pilot, would tilt me down?
Sure.
Where's the body? There it is. Perfect.
Center that up. There we go. Back to you.
Thank you.
Oh yeah, looks like another asteroschemated brittle star.
Try to get back to the center. Go.
Yeah, very spiny.
Central disk is moving.
Yeah, that's cool.
It's weird.
Okay, can we get a broader picture?
Yeah, let's go out to the whole specimen video.
Okay, coming out.
Or as soon as you're happy with the shot. And our laser's on.
They are now.
Excellent.
Yeah, I see it also. Okay.
Okay.
All right, I'll just tilt up a little to get the lasers on it. Here we go. Lasered.
Okay. I'm going to come out
So,
a little bit more so we can get more of the specimen.
just so you don't have to move,
But
can
that's
we?
full wide pilot.
Can you see if you can take an image of the coral in the background from here?
Yeah, we could try.
Just
okay, you all set, video?
Because
Yep,
we're discussing
set.
whether this could be a candidate for a sample, and we don't want to have to move too much.
Oh, I see.
So
Okay.
if we can look at that
I
in the
can get it
back while
right
we
here. Yeah,
establish
we can
some
zoom in the
things
back.
here. Oh, so that looks like
That's full zoom.
a seat pen.
Copilot, you want to start
So it's
setting
a
up for
C-pen?
a sample?
Sure. And we have the port outer, or the 2 starboard boxes.
Yeah.
Yeah, you only have one sample right now.
So your choice.
I'll see how it goes, but I'll probably do starboard inner just to save
the easy box for something.
Yeah.
Yeah, port inners.
Okay, you happy with that watch lead?
Yeah, I think that's satisfactory.
Okay, and is there interest in a sample of this colony?
Potentially, we have some lag on the chat here, so I'm just trying to give everyone an adequate chance to.
Well, while we wait, I'll just bump a little closer.
And in the meanwhile, there's always the left-hand colony we can image as well.
Yeah, Sirius is all settled out, so.
Nice.
Just out of curiosity, on the left-hand coral, we always like to sample the brittle star and the coral. On the left-hand coral, I didn't see if there was a brittle star that was easily sampled without, see they're kind of in the middle.
They're both pretty deep in the coral.
Yeah.
I don't think we could fit either of them in the box.
Yeah, we really do need the central disk, so I don't think it'll be advantageous to take that much of the colony.
Yeah, we'd have to clip some off the top and then clip the bottom, which would be a waste.
Okay, so can we? I think we're going to pass on this one. Can we move towards the C-pen?
Sure.
And get a better image of that? So this is a sea pen we haven't seen before on this particular dive, but it's probably belongs to the
free to zoom
to the genus Peninsula. Slightly, yeah. So it looks to have some irregular branching here. The center of the colony, which we're looking at right now, seems to be damaged in some way or growing irregularly. The tissue is red to pale, and the polyps are yellow.
Do you have any more zoom video?
I'm going to test that out right now.
All right.
That's max zoom.
Can we zoom up to the top of the colony when you have a good shot?
Very interesting extensions.
Yeah.
I don't know if I've seen like that before.
There's a little shrimp on it.
I can see some on the some yolky
How's
looking
our
eggs.
Pace to the waypoint 3.
Last I calculated was point.
Stand
0.07.
Can you come out
Zoom out.
partial?
Yeah, there you go.
Just lost my tail.
Serious to waypoint.
The coloration is something I have definitely haven't seen before.
I'll reset here. That's
I'll
good.
come out. Okay.
So we have one vote for a collection of the sea pen instead. I think.
Hi, Steve, Scott Brent. I agree with you. I haven't seen this coloration, but I'm not too used to working at this particular depth. Certainly,
Dan, looks like 0.08.
This is all new.
Copy
right
now, thanks.
And I agree with you. It sure looks like it's in the genus Peninsula, but I have no idea what species or if we could even tell that from the imagery, perhaps from the coloration.
Yeah.
Coming in more?
We'd have to go into the literature and see if there's any descriptions of that sort of coloration.
That's full zoom.
Right.
Yes.
So I think, yeah, we're getting some good images of it now. I think the sea pen is big enough that if we snipped a little off the top, I don't think it would impact the coral much. So I think we're going to set up for a collection of this sea pen, not the whole thing, just I'd say maybe 10 centimeters or so.
Okay. Would you like any more images before we snip?
I mean, aside from going around to the other side, which might be problematic and take a lot of time, I think
I'm just going to zoom out a
this
touch
might
for
be
framing.
good for now.
All right. Copy that. All right.
So, video's clear.
Yeah, let's get the lasers on it just to see how big it is. So if they want, oh yeah,
gotcha.
If they were 10 centimeters, so it's about 10. It's about 15 wide. Okay. All right, you can go full wide video.
Okay, coming full wide.
So one last recommendation, if it's not too much, to image the base.
Oh sure. Okay, video.
Okay, coming in.
Look how good tracking is right now.
So shore side,
wow.
It looks like there's a shrimp at the bottom. What would be the most informative?
Let me know
I know
when
it's
you're
a fairly
on a good
large
shot
sea
video.
pen, 50 centimeters. It's
I'm
really
good.
not
Coming
something we
out
can
now.
easily fit in a box. So is snipping
full wide.
some off the top going to be adequate for identification purposes?
Sorry, Steve, were you asking that to us on shore?
Yeah, yeah. Is taking some of it adequate?
All right. What do you think of that, Dan?
Yeah, it's just going to depend on sclerite distribution. There are some
setup, yeah.
sea pens where there are no sclerites basically in the polyps.
Kind of
So
bouncy in
maybe
the
in
sediment.
the peduncle or in the rachis, or along the rachis, the main
I don't know if they
axis
want
there.
the peduncle,
So
but
it's hard
good.
to say.
get it in 2 sections if.
I've heard just
but
a
if it's
snip
too
of the
big to
top
fit in
to
a
keep
box,
it
I don't
alive
think we
so
have
far.
much of a choice.
Yeah. So, pilots, I guess it might be better to collect the whole thing for identification purposes, but do you think that would easily, I mean, it's semi-flexible. I wouldn't say it's very flexible. You know, it may have some rigidity to it. Do you think it's possible to grab the whole thing?
Yeah, we could try to gently grab the base, pluck it. Has anyone done that before?
Yeah, I think we have. I would say maybe cut it in half.
Yeah,
it's pretty tall.
I think it would be hard to get the whole thing in the box.
Yeah.
But we could cut it in 2 pieces, do 2 collections.
Yeah. All right. Well, if the only way to fit it,
Yep. Bend it, you know.
Yeah. Can it be bent? Possible?
Is it very rubbery?
It'll pluck out of that sediment
If
very
you bend
easily.
it won't fall in half.
So it could be basically, you know, folded in, but I don't know how much trouble that is.
Yeah, I mean, I think we'll leave it up to the pilots. You know, I think it's still useful to have it, even if it's in 2 pieces, because the base is the important thing, it seems like. So I guess try and pick it up by the middle.
And then just try to pick it up by the middle and stuff it in?
Yeah, I absolutely agree with that, Steve.
Yeah.
Do you think get the base in, set it down, let go, take the end and just push it in. Then close the drawer and cut it in half.
Whenever we've grabbed the dunkel, we've always gone into the sediment.
Yeah, that's what I was thinking, yeah.
Pulled it out, but we could try it. I mean, we could try it. The worst that happens is it snaps in half, and we just do that afterward anyway, right?
Yeah, okay.
You've got your mini Zeus.
We'll try for the best case. Yeah, we bring it over to the craft.
And do you want to do port outer then, since it's going to be a little difficult?
Yeah, I think so.
Okay.
All right, hydraulics coming on.
Y saludos, todos están escuchando ahora. Vamos a colectar este octocoral o un coral blando porque tiene colores muy distintos.
Zeus camera as well.
Y es posible, es una nueva especie aquí en el Caribe de Estados Unidos. Se llama Sea Pen.
Grip force is 4. That's barely closing. That's pretty good. Video, will you zoom partially on Mini Zeus?
Okay, coming in partially. Let me know when you want me to stop.
Okay, stop there.
Right there? Okay.
That moved me.
Nice. So yeah, maybe zoom out on Mini Zeus.
Yeah, zoom out on Mini, please.
So
get
port
the box open.
inner is your only taken box. Got everything but port inner.
Okay, ready for drawer extension?
Yeah, ready.
I'm going to come over a little bit more.
Okay.
That is awfully tall.
Yeah. Something. Wonder if I should stuff it.
It will bend, yeah.
Yeah. It's kind of floating.
So this is that's
It's just more difficult because it floats a little bit. There we go.
Nice.
Okay, be ready to close it quickly.
Will do.
Okay, releasing.
Stuck to your finger.
Yeah. Yeah. Okay.
Clear?
Yep.
Retracting. Nice.
Port outer.
Okay, bring Mini over for stowage.
Sure.
Nice sample, Carl. Using the full depth of the box there. Getting your way in there.
All right. Okay, hydraulics coming off.
I'm gonna give the Zeus back to you, and I'll get the Mini Zeus realigned.
Ready for monitor?
Go ahead. And control back to you.
Excellent.
So on shore side, looks like we got the entire colony intact. So that's a good collection.
So I'm still
Peninsula.
facing 310, which is slightly off.
Well done, everybody. That's excellent.
No, I think it was
That's about right. Yeah. Okay.
There are. I think there were one brittle star on there. No brittle stars. Yeah, I didn't see any brittle stars.
I didn't see any associates.
So no associates on that particular colony.
I think we're still making a pretty good pace.
Yeah.
But we'll take a look when it comes to the surface if there's anything we missed.
Ian's sea pen in Spanish is pluma del mar. So,
yeah,
you want to shoot out ahead by the time the ship starts
para
moving?
todos
Yeah,
están escuchando,
like
mirando,
30 meters?
colectamos un pluma de
Or
mar
50.
Let's do 50.
y estamos en profundidad de 554
So I assume they're
metros.
probably going to want a little bit of time at the top to explore,
Temperatura
right?
es 12 de grados
Bridge is
Celsius.
ROV now.
Or carbonate, whatever. Fish.
Yeah, I'd like to move 50 meters, bearing 310 degrees, speed decimal 2. Good copy.
Video, feel free to zoom out the skid if you'd like.
Okay.
So we're
I'm sure this
still
shot
on
here will make
schedule.
it to the
We're
highlights. So.
again
I'm at
just
35
about
degrees in the camera.
We're just about through this area of sediment.
Come down a little bit and give you tether.
Very gentle sloping terrain. And over the next several minutes, we'll be increasing slope gradually as we head up this secondary mound in our dive track. What is that?
Looks like some debris, some sort, maybe.
Yeah.
Or is it some sort of cable?
Or a cable.
Yeah,
Ah.
potentially.
Yeah, Graciela mentioned that there might be some cable.
Okay, let me pause here.
You want to drop a target on that, Levi?
Huh.
Yeah, absolutely.
It's like a pile of steel cable.
I'm going to turn my head just a little bit.
Yeah, to see it.
Yep. Looking at 330.
So there's actually on the west coast a couple scientists that are trying to collect some of these cables. So this might be of interest to them.
Yeah, can we hold present position, please? Understood, thanks.
Just in case it goes up to a float partway.
Sure.
So single strand keeps going this way.
And I come up a little bit in altitude, and then I'll turn over to where you're looking. Wow, that is a lot of cable.
Yeah.
Okay.
So it's kind of north of you in a way, right?
Turning my head starboard. Looking just about due north. Tilting up 45 degrees.
All right, well, so we know it goes off to the east, and we keep looking towards the west, which is actually where we want to go and move more that way. Should we just do a move 20 meters due west to get away from it?
Yeah.
So I believe that these cables are old communication cables.
Yeah, maybe even a little...
or possibly previous fads. There are some fads like around this area, and fads stand for fishing like aggregated devices.
240-250.
They usually tether these cables to some type of anchoring system to the bottom, and then there's a buoy that is floating on top.
Range
and anything that is floating in the open ocean, it could be a buoy or some type of debris like logs attracts fish, smaller fish, which smaller fish attract larger fish like the pelagics.
Good copy, Rich, thanks.
So here's another.
Turn my head a little
Another looks like soft coral.
Camera's at 45 degrees. Nice. Looking 300. And that was a 20-meter move?
That's correct.
Okay.
Watch lead, any interest on a zoom here?
Just a quick zoom, because one of our scientists ashore who studied these Calagorgia was away when we imaged our last one, so.
Okay, lasers off, co-pilot.
Lasers coming off.
Okay, I have more zoom.
All right.
Coming in.
Yeah, this one has a chirostylid crab that was not present on the other colony. So
I'm drifting, and I'll try to hold here.
So we'll get a twofer here. We'll get our coral and we'll get our chirostylid crustacean people happy.
And brittle star.
And brittle star.
3 for 1.
Yeah. So pilot, where's the placement of the brittle star on this, just relative to any branch points that might be easily?
I'm gonna zoom out just a little bit.
Nav, let's stop the ship here.
Bridge, this is our nav.
Okay. Or I was just kind of going this way.
It seemed like you were getting further, maybe, though.
I think.
I'm zooming out, pilot, just
Copy that.
so that they can see the branching points.
The whole colony.
Yeah, I don't think we're getting further, Carl. We're kind of swinging around you.
Okay. Well.
Yeah, I can come down a little bit if you start to get tugged by the tether.
Okay.
Okay, that move's probably about done, right?
I think
Video, want to zoom on the end here?
Okay. Zooming on the top.
Top, yeah.
15 meters in.
So yeah, that guy is just 15, 20 from the top.
Okay.
Central disk.
Yep. So that's a reasonable size.
Yeah.
I just wanted to get a double check from our scientists on shore if, since we can get the brittle star and the coral, which appears to be different from the last one we saw, we'd like to set down now and grab those two, the brittle star and the coral.
Okay, coming out, pilot.
The tops.
There you go.
Excellent.
There we go. Move is complete, looks like.
Just for the record, on the shore side, there's another chirostylid on the bottom half of this colony, so that means in total there's 4 associates that we can see, 2 chirostylids and 2 brittle stars. Oh, is that a third brittle star?
I think there's 3 brittle stars.
Yep.
One.
Yep, that's right. One is hidden behind one of the other branches. Is that a fish, too?
Oh, yeah.
Yep, there's a fish in there.
Camouflaging itself.
That would be a good image to get.
Ready for monitor swap? And I'll keep an eye on this monitor up here.
Sounds good.
Can I zoom in, pilot, on fish?
Oh, yes.
I'll take your mini Zeus.
So, making everyone happy today, the coral people, the chirostylid people, the fish people.
This coral is like a palm in the desert.
Yeah.
Here and everything's gathering around it.
Yeah, so this is definitely larger
Move's
than
complete.
the last colony we
So
saw.
I'll try to get the jaws in between the 2 branches,
we'll,
just below the crinoid.
Take a look
kind of from
also at
below,
the
so
polyp
it'll be
orientation
pointing
for
down
everyone
when
on
I go to
shore
start.
so that we can check this against the last
And I'll do starboard.
Caligorgia we saw.
So you're going to
Instead of coming in horizontally, I'll try to come from a
at
little
an
lower.
angle so you can stuff in the box. Yeah, okay. Yep.
Okay, arm coming live.
Gotta go starboard side now.
Copy that.
Check, check.
And I will take your Zeus camera rotator. Sirius looks like it's settled out.
Okay, so that's the central disc about 10 centimeters above the jaws, right?
I believe so, pilot.
Yeah.
Yeah, I think so.
From video.
Okay. So I could go up a little more. All right, how's that look?
So the only thing is that it is kind of, the jaws are a bit perpendicular to the stock.
Yeah.
So it could push it away.
Yeah, you could push down on it.
Yeah, that's about 45 now. That's kind of what I was going for.
Okay.
Yeah, something like that.
Yeah.
Okay.
Nice. Okay. I'll follow you over with Mini Zeus. Ready for drawer extension?
Yeah.
Okay, extending the drawer.
Yeah, it is kind of perpendicular, but.
Rotate the jaws 180, and then
we haven't used the still cam yet, have we?
Oh, we did.
Yeah, we did.
Yeah. Oh.
I think 180 will stuff it better.
Well, I think then it'll be easier to stuff because you can put the jaws more in the front of the box, and the stock will extend toward the back. Instead of trying to get the jaws all the way in the back corner.
Yeah. Good idea. All right, that's most of the way in. Okay, you can close it.
Closing the box. Nice. Okay. Okay, Zeus rotator back to you. Lining up Mini Zeus. That should be good.
All right. Okay, watch lead, anything else here?
No. That's good, thank you.
Thanks for the tip, co-pilot.
Nice job with the sample.
It's still tricky. I think if that were one that floated,
could have gotten away.
Yeah, then you would really have to have,
gotten it at the right angle.
Yeah.
You could have, since it was a little bit longer, you could have also tried the outer box since it's a little wider at the back end, and it.
Oh yeah.
Might have worked even with the horizontal cut. That's still tricky. Okay, so we're 20 meters west of the cable. I'm looking at 335. I'll come over to 310.
Sounds good.
Unless, 320 looks pretty good on your sonar. How does that line up?
My sonar's blank.
I can change that, hold on. Alright, so from Sirius to the final waypoint, I got 311.
Okay.
Okay.
You guys want to move?
Yeah. Carl's ready.
You want 50?
Yeah, I'm ready. Yeah.
And then I'll stop it if we need to. And I'll do a 310 again, if that's alright.
Sounds good. We're at 310.
Bridge, this is RBNAV. Yeah, I would like a move range 50 meters, bearing 310 degrees, speed decimal 2. It's a good cap.
Pilot, this is watch lead. Could you zoom on the black feathery?
Right ahead?
Yes, right ahead.
All right.
That might be a crinoid, but.
Video, let's do a quick zoom.
Okay, zooming on black feathery.
If it's interesting, I can set down.
There we go.
Anemone.
Looks like a Cnidarian of some sort.
Is there interest in getting a better view?
That's on a stalked.
I think that might be a cup coral, but it's really fouled. There's also to the left a white branch.
Oh yeah, I see that. All right, I'm pretty much set down now. You can go in video.
Okay. That's max zoom.
Okay. Just keep the scene, see what they think.
All right, new estimated speed over ground we need to do is 0.09.
Good copy.
So if we go 0.2 half the time.
Okay, video, let's focus on the stalked branch now.
Okay.
That looks very similar to a larger branching octocoral we saw earlier in the dive, but very small. White with a slight yellowish color to the polyps. Okay, satisfied there. We can move on.
Okay, coming out.
Looks like you're moving, co-pilot.
Yep.
Thank you, video.
Tilting my camera down to 45.
So how many minutes are left in bottom time?
119.
So we've got 2 hours. Yeah. Well, that's if we leave bottom right at 4:15, right? We need to add a few minutes for the ship. So.
Copy that. How many minutes? 10 minutes?
Yeah, probably.
Okay. All right, now you're down to 1 hour, 48 minutes.
You could see some of the tracks of those irregular sea urchins as described by Chris.
So who replaces you, co-pilot?
Sean.
Oh, I see.
I think I'm on deck.
You are?
Yeah.
Okay. Who replaces you?
I took Sean and I took Fernando's watch.
Okay.
That's what happened.
I see.
So yeah, I don't know. I could check.
That's okay. It's just. That was
something in that hole, pilot.
Like just below the lasers?
Yes.
I can set down real quick just to.
Okay. I'm going to turn my head 20 degrees to keep you in view.
Alright, I'll just get the zoom down the hole and then I'll take off again, co-pilot.
Okay.
Alright, video, you're free to zoom.
Okay, it just moved again. It might have moved farther in.
Looks like a home of
Unfortunately.
some animal.
I think it went inside.
Yeah.
Alright, let's move on.
Okay, coming out.
For those who are joining us, you are watching or listening to the Okeanos Explorer.
So you came over 20?
We're on our last leg of the dive. We're trying to reach this top of this mound. Right now we're at a depth of 551 meters.
So we're almost to the part where it gets a little bit steeper.
We have about a little bit under 100 meters of vertical left to traverse. Okay, but we're starting our climb up of a more intensely sloped area
50 degrees.
Towards a large local topographic high that is suspected to be an aggregation point for deep sea fishes, especially snappers like queen snapper, which were reported from this area and from that depth range.
So a lot of these sites that we are exploring during this mission would not be possible without the help and collaboration of the local fishermen. They've guided us in many ways to these different sites. Looks like another soft coral ahead of.
You wouldn't necessarily expect it with this much soft bottom, but these primnoid fans appear to be the dominant coral in this area. This one's even bigger than the last two.
About 10 meters.
Okay.
Let me double check. Yeah, about 9 meters.
Can we get a zoom on the part of the polyps and also one of the brittle stars?
Sure.
The center
I'll set
disc.
down to face it.
Yep.
Should have a nice dark background. So you said the base?
Yeah, we can start at the base, and we're looking at close-ups of one of the brittle stars, preferably part of the oral disc, and
video you can go in.
Okay,
a close-up of some polyps that are easy to focus
on.
on.
Take the tilt.
Sure. So look at the base first.
Yeah. All right, video, just let me know when you're ready to tilt up.
I'm ready.
All right.
Coming up.
Try to just tilt slowly up. Let's go on the, we'll go the next one. Can you do lasers off, Copilot?
Sure.
Let's pause on the disc of this guy. Okay, you can go full in when you're ready, video.
This particular colony, it looks like the polyps are generally oriented closing upwards.
Tilting my camera down.
Okay.
60 degrees.
All right, will you tilt D2 down once you're comfortable? I'm really multitasking you here.
It's okay. Tilting down.
So here's a brittle star right now. It looks probably about as spiny as the first one we saw on this colony, or this type of colony. We're looking at the central disk in addition to the spine orientations because they are probably the most descriptive factors for identifying brittle stars.
Do you have any more zoom video?
That is full zoom, pilot.
Copy.
So I think polyps facing upwards in whorls of, I've seen mostly 3 polyps per whorl on the octocoral.
Okay, let's keep tilting up and get some polyps with the dark background.
Copy that. Tilting up.
Move complete. Thanks, Bridge. So move complete, pilots.
All right.
In order to make the top, we should keep moving.
Okay.
Yeah. So we'll get our imagery here of the polyps, and then we'll move on.
It's quite tall.
Yeah. It's kind of cool. There we are.
Can I get that as a screensaver?
Then loop it so they never end.
I think there was a screensaver for this stuff a couple years ago, right?
That was coral.
Yeah. Or for our images.
All right. So as far as I can see, the polyps are generally oriented upwards through the entire colony. The closed polyps I have seen are folding upwards, up axis.
And then before we go, we'll get one more laser shot.
Okay, I'll tilt down.
Yeah, that'd be nice.
Very nice background, though.
We'll
do that level of zoom. Yep.
Yeah. And then coming out now.
We hold that for a second. I'll tilt down a little bit.
So dark.
That's awesome.
Behind it.
Oh, it looks like a...
Yeah. Yeah, Kairos Stylid also, yeah.
Is this going to be in the highlights, Caitlin?
Maybe. Just gonna come in just like that. More black background.
Nice.
It's pretty.
All right.
Okay. Want control back to you?
Yes, please.
Coming out.
Okay, can we get lasers on?
That's full wide, pilot.
Okay, we have lasers on the middle there. Tilt down. There's the whole specimen. All right, whenever you're clear, video.
Okay, we're clear.
Excellent.
I'm full wide though.
Yeah, I'll back up. I just meant I wouldn't move until you had your shot.
Gotcha.
All right, so your camera's at 63.
Yes.
All right, we could probably do the next move.
Understood. 310 look good?
Yeah.
All right. Bridge, this is RV now.
Tilting up.
Yeah, I'd like a move, same as last. Range 50 meters, bearing 310 degrees, speed decimal 2.
It's 40 degrees on my camera.
Good copy. Thanks, Bridge.
Now, what average do you have to keep now?
Can I at least get on white pilot?
You are 255 meters away, and we have 97 minutes. 0.085. You guys made some ground.
Good. Still, actually, that's about when I started at noon. That's what we had, right? So, just barely keeping it. All right, so we have a ship move in. I'll pause here. Nothing on sonar.
Again, you could see quite a few of these irregular sea urchins lining up. Noosties. They look like those are the ones that kind of look like eggs, flattened eggs. You can see their track lines. It looks like maybe there's a couple sea urchins. In the view. So a lot of these are bottom feeders. They're most likely scavenging for any detritus or animals, if they're carnivorous.
Still don't know what it is.
Not sure.
That looks like a bivalve, the shiny shell. Brittle stars below it.
All right, a bivalve. We can go wide.
Okay, coming wide. Yeah, that's full wide pilot.
All right, you can lose the skids.
Losing the skids. There we go.
So we're still at a depth of 544 meters. Right now, salinity is at 35.6, temperature 12.6 degrees Celsius, and oxygen 2.8. That might be something interesting in front of us.
Yeah, what do we got there? Looks like another penatulid from a distance. Type of sea pen.
Pilot.
Co-pilot. So in the middle of a 50-meter move.
Yeah, it looks like one of those sea pens.
The terrain looks exactly the same as the one we collected.
But not a lot up here.
Copy.
Can we snap zoom just to document this and then move on?
Yeah. Video. Could you do a zoom to the specimen?
Snapping in.
Here we go. Yeah. So this is now the opposite side that we were looking at from the colony that we collected. More or less looks like the same species that we have in our bio box, but this is the side facing the polyps rather than away.
Okay, go ahead.
Si es un pluma de mar, es un octocoral or un coral blando.
And losing skids.
So nav and co-pilot, I think last we checked we had to keep a pace of 0.09 knots.
That is correct.
And we were doing 0.2 knot moves, so we're going to have to be moving at least half the time.
Copy that.
How's the surface current right now as far as recovery time? Oh, pretty
There's a
pretty
fish
low.
on the left.
Pretty low.
Great. Fish on the left. In the sediment there?
Yes. It looked like it was moving.
Something.
It's another bottle.
It's very
With
shimmery
the fish in
though.
it
Is
now.
that a bottle?
Looks, yeah, it looks like a bottle and maybe some
Fish in
fish.
front of it.
Yeah.
Mm-hmm.
Do you want to zoom in video?
Coming in.
Oh, it's inside the bottle.
Oh, wow.
Oh.
Huh.
Looks like some bass or some type. I don't know.
Yeah, I'm not sure. It has barbels. I just saw that.
Huh.
Yeah, well, that's a great little habitat there, but, you know, not ideal for the seafloor.
We got lasers off, co-pilot?
Yeah, laser's coming off.
Yep.
I don't know if the button is working.
So we
Oh,
have
there
been
you go.
documenting
Yep.
quite a bit of
Yeah,
debris in
that's
this area,
wrong.
mostly soda cans,
You can do whatever
bottles,
shot
jugs.
you'd like video for zoom.
A lot of
I'm sorry,
debris,
Pilot, can
I would say
you can zoom
more
in if
than
you'd
average.
like, or keep
Yeah.
the whole bottle.
Okay.
And some
It's kind of
old
interesting
cables.
either way.
Yeah, I'm going to keep it for now, but
Okay.
probably zoom in a little bit.
Yeah, that right. You
He
know,
keeps going back and
wouldn't
forth.
you think
Yeah,
so?
that's true.
That would be an easy way to sample a fish, right?
Collect it in the bottle.
Yeah. I don't know if that'll work too well, but man, if it was a super important species that we couldn't get a good ID on, that would be one way to do it. But no, I don't think we're going to sample. We're not going to sample the fish. That was a joke.
Can I
But
zoom
it
in a
is
little
an
bit,
interesting
Pilot?
use of
Yes.
Get him in the neck of the
habitat
bottle.
for as much as it is.
Right, pilot, I'm looking at you.
It looks
at
like there's a
80
brittle
degrees.
star also
Okay.
sharing that
Yeah, after
habitat
this shot, I'll take off.
and something moving inside.
Oh yeah,
Yeah.
maybe a hermit crab or gastropod. Okay, thanks pilots.
You're
We're
welcome.
satisfied here.
Video whenever you have what
Okay.
you'd like.
Zooming out.
All right.
Okay, that's full wide. There's another bottle in the background too.
Okay,
And there's another
310.
bottle right next to the other one.
Yeah.
Yeah, you can see the sediment getting a little steeper here. It shows up in Sirius sonar too.
It looks like we are climbing now. Looks like it's getting steeper.
Nav, is the ship move still going, or?
Yes, it appears to be.
Okay, great. We'll push out then.
See some stalked crinoids, some that we were documenting in abundance yesterday. I'll have to find the
Just
name
be
for those.
about,
I used to know
a
it.
couple more meters, I believe.
The stalked crinoids that are able to move on the bottom.
Yeah.
All right, my camera's at 45 degrees.
Very good. Quick zoom here, video.
Coming in. Good eye pilot.
Urchin.
Urchin.
Have we seen this one yet?
I think we have. Yeah, the one with the fouling on the spines.
Okay. It looks like it's right next to a glass sponge.
Yeah, everything here is, like we were saying yesterday, everything is super small now.
Yeah.
I think this this substrate, since it's a little bit more sloped, is perhaps a little bit more consolidated. I don't see a lot of loose substrate.
Do you think there's a lot of turnover from organisms? Like they are there for a short amount of period of time, and since it is such a slope and possibly with currents?
Possibly, yeah.
All right, should we, so the ship stopped a minute ago?
I think for attached organisms, I think it might just be a limitation of the hard bottom resource. You know, it's not firm rock.
Just
it's
excited
potentially could.
Are we happy with the current heading that we're on? With a 310.
Copilot's happy with that.
Yeah,
good.
I guess.
Unless we need to.
The returns are a little harder to your starboard, but is that the? Yeah. This is good. It's not that steep. So.
Copy that. When the next move's completed, I'll call on another 15 meters. It seems that you guys have been moving at. So if you're comfortable with that, I'll continue at that range.
Copy that.
I don't think we got a good idea on that fish in the bottle, so I'm just going to call it fish in
the bottle.
We'll,
it's a new song. Maybe let's call up Sting.
Fish in the bottle.
It looks like another sea urchin.
Yeah, I think that's been over for a bit now.
Bridge is RV nav. It looks like that last move was completed. Do you mind if we do another one? Copy that. It will be exactly the same, 50 meters at 310 degrees. Speed decimal 2 knots, please. Good copy, thank you.
So thanks to our shoreside science team as well. We've had a lot of participation today on the call and through the chat room. A lot of people to mention by name, but as we go through, I'll try to recognize every one of them. On this particular expedition, we had approximately 211, I think the count, and growing, scientists and interested parties helping us plan our dives and actively participate in the exploration of this area. We're taking into account a lot of resources from local environmental and habitat managers, conservation directors, as well as scientists from around the world. In the lower left of the corner, there seems to be something with branches.
I see that.
It looks very blended into the bottom here. And just for the record, after that, to the left, there's this sea star.
Okay.
We can get a quick snap zoom on.
Okay, there it is with lasers. Copilot, will you kill lasers?
Coming off.
And video, you're free to zoom.
Okay, coming in.
Looks like maybe a stock crinoid.
Yeah. Yeah, it does look like one of the stalked ones. That's right.
It's good.
Smaller than the other ones I've seen so far.
Okay, you're free to zoom in whenever you're ready.
Alrighty, coming in.
It's worth once in a while zooming in on these crinoids, even though sometimes they're hyper abundant, because there are interesting parasites that live in the grooves of the arm. This one doesn't seem to have any parasites that I can see right now.
It looks like some type of crustacean on the right below it.
Oh, yeah.
Tilting down.
Right there.
Yep.
Okay, before Sirius catches up, let's take a look at the sea star. Okay. So if you come out, Copilot, can we get lasers on?
You got it.
I think the sea star, it looks familiar to me.
There it is with lasers.
Perhaps not to the species level, but I think it might be.
Right, you can go in video.
Okay, zooming in.
Oh, no. My guess. I'm not gonna bother. I take it back immediately. I don't recognize this one immediately.
Coming in more.
This is, I guess, the first.
Yep, you're free to zoom as you'd like.
First we've seen this on the expedition.
Yeah, I can't tell if it's preying on something below. There's a small rock. Could be feeding. Could be just moving. Doesn't seem to be going anywhere in a hurry.
Again, you could see right underneath the arms there are tube feet, which allows it to move around and to capture its prey.
All right, pilots, thanks. I think
You're
we're
welcome.
satisfied when video has a good shot.
Okay, coming out. That's full white pilot.
Yeah, Nav, if you look at the manip cams, you can see they're both in, actually. When I let off, you can see the shadow getting farther away. This is very nice sediment. It doesn't stir up in clouds.
Yeah, I was going to say I didn't see any real indents or any clouds. So.
Yeah.
I think this was for the fish. No?
Quick zoom here, video.
Okay, quick zoom.
It's like a sponge.
Hey, this is Chris Ma.
Hello, Chris Ma.
Okay.
Hey, so
I'll
that
just
sea
pan up
star
past
that you
this.
were looking at, if I counted the number of papular rows correctly, that looks like an Ophidiasterid in the genus Tamaria, probably Tamaria halperni, which is named for Gerald Halpern, who was a student at the Rosenstiel Marine Science School that described many species of goniasterid sea stars. Ophidiasterids are kind of similar to the things that you might see in a shallow water habitat, like Lynchia or Ophidiaster, sort of like those ones that can regenerate really quickly, that can regenerate. It's not unusual to see them at shallow deep sea depths, but they're typically not deep sea animals as such. I don't know if you would notice that it was feeding on something. It might have been munching on some of the sponge or epizoic animals that were on the bottom, and the shallow water ones I think tend to feed on similar types of things, microfilms and whatnot. So, but definitely the first one for this trip.
The water seems to be getting more
Thanks
murky
very
as
much.
we come up.
Thanks for stopping. I'll talk to you guys later.
Yeah.
Bye.
Bye. Take care.
Bye.
Nav, this is watch lead.
Go ahead.
Could you give us an estimate of time on bottom?
Sure, give me 1 second. Looks like we have about an hour and 15 minutes left.
Hour 15, okay. So to everyone on shore side, there's about 1 hour 15 minutes left on bottom, and we're now on the mound that we had planned in our dive plan, this large topographic high point
All
at
right,
the top
so
of
what will the next move be? Are we still 310?
a rather dramatic cliff south of St. Thomas, east of Vieques Island.
Yeah.
So this area was suggested to us because of the potential presence of aggregating deep water fishes like snappers and groupers.
Yeah, I'll go ahead and call it in. Another 50 meters.
Y buenas tardes a todos.
Yeah.
Están mirando, escuchando, especialmente en el pico exploratorio en San Juan. Saludos. Y ahora estamos sur de St. Thomas y noreste de la isla Vieques. Y ahora está subiendo y estamos en profundidad de 15 metros. Y vamos a buscar hábitos, las comunidades de los peces en aguas profundas, como cartucho y de meros.
I'll try for this midwater video.
Is that
Okay, going for midwater.
a ctenophore?
Like a ctenophore,
I
yeah.
lost it.
I see him.
Yeah.
Drifting really quickly.
If he goes up, we can get him.
Can't really see the combs.
Sorry.
You're okay.
Gonna get lasers off, Copilot?
Yeah, lasers coming off.
Well, that's interesting.
Yeah.
It's not a ctenophore, or it looks like a jelly, actually. A medusa that I've really seen before. No, I think it's a ctenophore. I see some iridescence now. Although that could be just the refraction of the light from the vehicles. Yeah, so we're getting some indications from our shoreside scientists that it's probably a medusa. So this is a good segue into talking about our midwater section of the dive, and not on this particular dive, but towards the end of our expedition, we'll be doing some dedicated midwater surveys in which we'll go down and explore the bottom, and on our way back up, we'll do a series of 100-meter or so transects, or 10-minute transects rather. 10-minute transects at depths intervals of 100 meters, from about 1,200 meters to close to 700 or 800 meters, examining all the midwater fauna we find, such as these medusae and midwater fishes. There's actually a great diversity of life that occurs in the midwater, and they're sometimes can be highly migratory, often making hundreds of meters vertical migrations into the shallow water at night to feed, and then hiding back down deep during the daytime to avoid predation.
Some say it's the largest migration of any
clear.
type of animal.
Yep.
Thank you, Copilot.
Yeah, especially because most of these animals are at most sometimes centimeters, millimeters to centimeters. So I'm seeing on the seafloor still some of these sponges, many sideroid urchins. They seem to be dominant right in this particular area. As well as crinoids. There's a few of them in the frame right now.
Coming back to center, Copilot.
Right now we're passing about 500 meters depth. The final end depth for this particular dive is around 460 meters, 465 meters. Can we take a look at the shiny fish?
Certainly. Get a little closer.
Looks like a smaller midwater fish.
Okay, video, let's zoom from here.
Okay, coming in.
I'll try to creep in.
Is that
That something new or something hanging from
Say
its
again, Copilot?
mouth?
Do you want lasers off? It's kind of falling in between the dots.
So it's not really
yeah,
you can take them.
Is that one of the Neoscopulus? The blue iridescent fishes? Although I can't quite figure out what's going on with that.
Oh,
Oh,
there you go.
It doesn't look like a parasite then if it falls apart like that. Nope.
Oh.
No, I think that's something different.
Okay, I'll back up a little. Is that with the light?
That looks like something we haven't seen so far, but this is some great imagery of it.
Okay. Oh, will you put the spotlights on?
Yeah. Spotlights coming on.
Oh, whoa. Sorry.
Oh.
Oh, we should have asked you video.
That's okay. You're good. Oh.
Nope.
And he's gone.
All right.
So,
all right, coming off.
Thank you.
Okay, coming out, Violet.
Yeah, video in the future will.
Sorry. Oh no, yeah. I just wasn't like expecting it to be that bright.
Yeah, but we also, if you were trying to get a nice shot, we don't want to ruin it.
That's okay.
still seeing some sponges, crinoids. Nothing very large here.
Okay, it should be flattening
The
out
sediment,
again.
the substrate seems to be
nothing in sonars.
Fairly well consolidated. I don't see a lot of slumping of sediments, and there's there are some, but there's infrequent or fewer burrows, bioturbation, although there are a few mounds which are likely resulting from infauna or fishes that burrow, or crabs or mollusks that might be burrowing in the sediment.
How's that mood progressing now?
Looks like it's just about ready to wrap up.
Nice.
Good copy, Bridge. Thank you. Palace, do you feel like another 50 meters?
Some debris, perhaps.
Degrees, so
yeah,
in good shape.
Pretty far ahead. We should do it.
We'll do another one, same heading, and once we get a little closer to the top, we'll figure out where the slope's coming from. We'll be able to change from there.
Sounds good.
All right. Bridge, this is RV nav. Looking for another move, exactly the same, 50 meters at 310 degrees, speed decimal 2.
And it's walking.
Walking.
Like,
oh, I see it, yeah.
Good copy, Bridge, thank you.
Let's go take a look.
Pilot, do you see any relief coming up that might be of interest to check out in the sonars?
No, if you look at the left-hand sonar, which is mine, it goes out to 50 meters, and it's all kind of flat.
There's something crawling on the seafloor, straight ahead. Pilot, would you mind snapshotting that?
Yeah.
Oh, it looks like some crab.
Co-pilot, will you take put the lasers off? Okay, video, free to zoom.
Coming in.
Ah, just a hermit crab.
Oh, I scared him.
Fairly photogenic one.
Okay, coming to port.
Very pale hermit crab. With the exception of the eyes, it's completely white and discolored.
Is that a worm behind it to the left? Moving around.
Oh yeah, something. Yeah, something right here.
Is that max zoom video?
There was something right here that was moving around.
Yeah, I'll come to port a bit.
Just behind the worm.
On his
Okay.
face.
Just behind the crab.
Okay.
Okay, coming in.
Yeah.
That's max zoom.
Okay.
I think, are we
Yeah, I think
good
we're
here?
good. Thank you.
All right. Happy video?
5 seconds.
Sure, no rush. Just got stable.
Oh, yeah.
Okay. Thank you, pilot. Okay, that's full wide.
Excellent. All right, the ship is underway. Both sonars are clear.
We should make the top of this mound with some time to spare, so hopefully we'll see some deep sea fishes that we've been waiting for. Some of the snappers and groupers that we've been waiting for all dive might come into view.
So right now we are reaching kind of the lower limits of where we would possibly find queen snapper. We're about 499 meters. Some of the fishermen have recorded them all the way down to 450 meters, but still there's some question of how deep they actually can go or live.
So queen snappers, do you usually find them shallower, or can they, or are they
So they think they also have an ontogenetic migration, something similar to that of the silk snappers, where some of the juveniles are the younger fish are closer to the shore, maybe at the shelf edge, and then when they get bigger, they move to these deep water habitats. But to my understanding, that's still up in the air with the queen snapper. It's known for the silk snappers. But you could find these snappers actually all the way up as far north as Bermuda and North Carolina, and to as far south as Brazil. So they have a wide geographical range of distribution.
Are those crinoids on the right side?
Yeah.
I can't tell. There's at least a couple of them.
Yeah, they kind of look like stock crinoids.
Mm-hmm.
Oh, here's another one.
Yeah, that's right.
Behind. Looks like a different species, possibly.
Can we zoom on the, snap zoom on the crinoid that's to the right of the lasers?
Yep, copy that. All right, video, let's go for a snap.
Okay, snapping in.
Then Copilot, get lasers as you want.
All right,
looks like
lasers
this
coming off.
Oh, there's a small cup coral to the right of it.
Right. So I think it looks very similar to the last one we saw.
Yeah, it's not as poofy as the last one, though.
80 degrees.
I know these kinds of stalked crinoids can get much larger, but they all seem to be about the same size. Could potentially indicate some sort of recent recruitment event, or if not recent, then a recruitment event that gave rise to these crinoids at similar sizes.
Take it real quick.
Okay.
And then science, is that all you need from this?
I think that looks great, thanks.
Copy that. All right, video whenever you're happy.
Okay, coming in. Coming out.
Copy.
There we go, that's full wide.
Great, thank you.
Looks like we have another 10 meters in that move. If we're still happy, we'll continue at another 50. Right after, roll into it.
Yeah, we'll let D2 get out ahead.
Copy that. I'll let you know when that move's done.
Copy.
Crinoids everywhere.
They like this habitat.
I see something on the right-hand side, though, that might look a little different. Just went out of frame. Or is that on another crinoid?
Right here?
It
looks like it.
In the upper right corner?
Oh.
Further up?
Yeah, upper right corner, it looks more bushy.
Copy.
Yeah, I think that's also a crinoid, but
snap zoom video.
A different and non-stalked species.
Yep. Yep. You could see it's cirri.
Nope. Possibly some arm regrowth there. Tips of the appendages seem lighter in color.
Is that all you need, Science?
Yes, that's it. Thank you.
Copy.
Okay, coming back out, pilot.
Great, thank you.
Yeah, let's go wide.
Was that a coral or a sponge?
I think that might be a coral.
Okay. Take a look at it. I think it was a...
Great copy, Bridge. Thank you.
A Chrysogorgia.
Okay.
Completed. We'll let D2
In
push
which
out
case,
a little bit, and then I'll call in another 50.
we saw
that yellowish thing we just passed?
Sounds good.
Sounds good.
The
What's
yellowish
our range now?
thing that we zoomed in on?
Until waypoint 3.
Oh, sorry, if you zoomed in, I might be doing the same thing as yesterday, trying to see this small yellow black coral that looks like a crinoid.
Yeah.
Oh, excuse me. Distance is 100
Yeah,
meters.
I think it was definitely a crinoid.
Bearing is 317.
Oh, just 100 now.
So if we keep coming at 310, we'll be
We
able
thought
to
we had
come up
also
around
seen
it.
a small bushy translucent coral colony. It could have been a Chrysogorgid.
Right, and I heard you say that on the phone, and
Yep.
you know, there's a difference between the phone and the screen, and when I looked at the screen, I was looking at the yellow thing, so it's maybe just been a different timing issue.
Yep.
All right, I think we can call the next move in.
Copy that. I'll call it in now.
Great, thank you.
Bridge, this is RV nav. Like to make another move. Exact same thing, 50 meters at 310 degrees, speed decimal 2.
I would say there's been an increase in density, though, in these stock crinoids.
Thank you.
I agree. I agree to that.
As we move up on top of this mound, we would expect currents to be accelerated around the topography, potentially increasing food resources for attached fauna. Even though these stalked crinoids aren't necessarily attached, they are more or less sessile or not highly mobile. They can if they want to be.
They're able to shed their stalk and regenerate a new one if needed, if they're getting chased by some predator. Or they could drag their stalk along and move along with their arms.
Pilot, I'm just going to turn my heading to see if something in sonar follows me.
Copy that.
Don't be alarmed.
Pilot, this is watch lead?
Yeah, watch lead, go ahead.
If it's at all possible, as we get to the top of this mound, it may be of interest to look off in the distance more. If it's navigationally possible, to see if there are fishes in the distance that are keeping their distance.
Yeah, I can do that. Do you want to zoom in off to the distance or just have the camera pointed upward.
Have the camera pointed upward. Yeah, that would be ideal.
Yep.
And then we can zoom if we see something. But I figure if these fishes, like yesterday, where they were pretty skittish, and if we can at least see when they start to come into the light, then we can try to strategize our identification and sampling of their and zooms.
Copy that.
It looks like maybe we're coming up on another sea pen.
Do you want to zoom on this science?
It looks like the one that we identified before, so.
Copy that. Moving on.
But if you have a second and this is a fun view, it's okay, you're past it. Don't go back. Don't go back.
Is that a request to see it?
Yes, if it's any way possible, if you could zoom in.
Yeah.
Yeah, sorry about that. It's just when we were getting the really good close-ups before, we were on the back side of the colony, so we haven't seen the front face of the colony close-up, I believe.
Okay, coming in.
Oh, okay.
Well, this is an entirely different thing.
Yeah, it's a different species.
Like this is a black coral, I think.
Zooming in more, pilot.
Yeah, take
so that
it.
they can see what it is.
Yeah, this looks like an actual black coral.
Not a sea pen at all, actually.
Yeah, good call. Yeah, you could tell by that it's a black coral because of the skeleton.
Well, that helps. Certainly, you can see the tissue is so thin. You can see the main axis black. But it's because of the size of the polyps. If you look along those branches, you almost can't see polyps. It looks like there are tentacles arising directly from the branch, and that's just the structure of these particular black coral polyps. They're really, really thin.
Okay, video is ready.
All right.
Thank you.
Good. Come out. We'll move on.
I'm going to cycle your tether cam.
Copy.
There it is.
Did it shut off?
Yeah. Or something went wrong.
Yeah.
It's been doing it a couple times, or did it a couple times so far. Still nothing in sonars. We're moving along. Just 70 meters to the top.
So not a lot of hard bottom here, but the sediment appears to be consolidated. The majority of the organisms in this area are echinoderms, sideroidotrons, several stalked crinoids, occasionally a sponge, and one of the black corals we just saw. no evidence yet of deep water snappers or groupers. We've seen quite a number of fishes down deep. Nothing too huge. Depth right now is around 480 meters. Temperature around 13.9 degrees C. And oxygen concentration around 3 milliliters per liter. For the most part, oxygen and temperature have gradually increased over the dive.
I think that this is real.
Yeah?
Yeah, it's getting closer. Yeah, see.
Yeah, we're almost done with that other dive. We can change heading if you'd like.
Yeah, after this move, turn a little starboard.
So we are approaching kind of the top of this mound.
If there's a ridge.
Yeah.
We still have some ways.
Okay. I'll let this move finish and have pilot line up.
But the site is located on the edge of a really important extensive mesophotic reef system called the Marine Conservation District, also known as the MCD.
To see it.
Yeah,
this area has been fully protected from fishing since 1999, in full collaboration with the fishermen in the area.
So it looks like there's a bit of a ledge in the immediate distance. This could be the top of the mound here.
Good copy, Bridge. Thank you. Pilots, that move has been completed. If you'd like to set up for the next one to get us over the ridge.
Copy that.
Yeah, I'll start turning to starboard.
It's like a giant sponge.
Centered.
What is that?
It's a sponge.
It's like a sponge.
So
demo
D2's
sponge.
almost there. Maybe we should just
Possibly.
move me 10 meters or so.
Yeah, I'm looking 345, or you're looking 340. Yeah.
Yeah, maybe 15. That'll get me right over it if we're going to turn to port and head up.
Copy that. 15 meters at 348.
340 is my heading. Yeah.
340. Copy that.
Sounds good to me.
Can we zoom on the sponge?
Yeah, copy that. Let's give you 1 second video. Get a little closer.
Bridge, this is ROV nav. Looking to make another move. 15 meters at 340 degrees. Speed 0.2.
Get rid of lasers, co-pilot.
This looks like a sponge we haven't seen yet on this dive.
Good copy, Bridge. Thank
video.
you.
You can go ahead and come in.
Okay, coming in. That's partial.
Does it look like a demo sponge or a glass sponge?
I would say a demo sponge.
Coming back to center.
But I'm not sure. Kind of looks like a geodia, but if there's any shore team scientists that maybe possibly know this species.
I'm not sure. I suspect Nolan could offer an opinion here. If it is, it's a real clean one.
More.
Yeah, take what you want.
I don't recall. I think we're pretty close to the end, but if it is Geodia, that's an aspired collection target.
Mm-hmm. Yeah, we were basically right on the last bit of our dive, maybe about 30 minutes, or sorry, 30 meters to our last waypoint or so. So, we have some time here.
I'm going to scroll down video.
Okay, sounds good.
We want to make sure that we get around or follow this crest as much as possible to see if there are any occurrences of deep water snappers or groupers. But yeah, this is what are the dimensions on this pilot? Do we have lasers? When
I think it was just over 10 centimeters in width.
Okay, so not enormous. But
come out, pilot.
Sure, yeah.
So check out the lasers.
We got lasers.
Put them on.
Thanks.
Yep.
So yeah, science, you can see that right there.
Yeah, maybe on the higher end, maybe closer to 20 in length.
All right, come wide video.
Okay, coming wide. Okay, that's full wide.
If we can get an ID on that sponge, or even if not, we know where it is. We're not too far. We can always come back.
Do you want to drop a target on that sponge, Nav? Or
Will do.
Just a little bit behind D2 now?
Yep.
Yeah, science, if you want more time on that, just let me know.
We'll give our shoreside chat room an opportunity to respond. And, you know, we won't go too far, but it's good to check out what's on top of this.
More in the distance.
What are we calling this sponge?
Would you call it a buoy?
20 centimeter sponge.
Buoy sponge. So it's side to side. I'm going to take a step up now.
I mean,
looks like there's a nice feature in that head.
Yeah, it's cool.
Oh, we have a pretty big ledge.
Oh yeah.
In front of us.
So there actually is a bit of it's not as much of a gentle slope kind of mound as we were expecting. It's more of these terrace-like formations.
And a lot of times fish at this depth use these terrace formations for protection or refuge. So we might be able to identify some of these deep water species.
We did zoom on a couple of these black corals recently, right? Did we get an idea on those?
Do you want to check that? Check that out.
We can zoom again because
yeah. I was curious if they were the same as the stylopathes we were seeing earlier.
In the back.
They look very similar.
There's something like with that crinoid.
Do a snap zoom?
Snap zoom.
It's a rock.
Oh. Oh, it's
Good
a rock.
copy, Bridge. Thank you.
Okay.
All right, you can come wide.
Okay, bye.
All right, science. Anything in view you want zooms on? I think I heard some chatter.
Yeah, I mean, we got zooms on this particular black coral. Although, actually, can we go back into that black coral? I think I see it branching near the top.
The one dead center?
Yeah, and that might be interesting to note because I thought they were unbranched.
Yeah. Okay, I'll get lasers.
Yeah, you can see two branches at the base as well. And there's certainly some parentopathies that have a general shape like this that also branch. I'm not saying that there's a parentopathies, but there is still some things to be learned about the overall colony morphology of some of these black coral species.
Yeah, right. Yeah, because unbranched, it resembles stylopathes or a relative of it. And
I have more zoom pilot.
Why don't we go right on the branching center? You can take a video, yeah.
Okay, coming in.
There's also some branching at the bottom pilot that we can take a look at if we have time.
Yeah, copy that. All right, video, I'm going to scroll down.
I didn't recall seeing any branching on the last one we zoomed at just a few moments ago, but the chirostylid crab here. Yeah, so it's not a regular branching pattern, but there are two branches at the bottom and then one at the top.
Sorry, video.
You're good. Looks like there's also an arrow crab right at the base.
Video, whenever you're happy.
Okay, I'm ready.
We were happy back here.
Great.
Okay, that's full wide.
How about I move 10 meters closer so you can get to the wall. Yeah.
Copy that. Same heading?
Yeah, I'm 335 now.
Okay, should I change to 335? Copy that. 10 meters at 335. Bridge, this is RV nav. Would like to make another move, please. It will be range of 10 meters, 10 meters, at 335 degrees, speed decimal 2.
Oh yeah,
copy,
well,
Thank
it's
you.
okay. These
Watch
are
your,
some
upper swing arms
more
on it.
dramatic
I think you're good.
outcroppings. Yeah, I see that.
vertical.
In the center, top middle of the screen, just passing center now, there's a large branching structure.
I'm just going to do a quick
Yep.
side-to-side view. Watch leads.
Oh, that's a crinoid.
Yeah, it looks like there might be some things to look at here.
All right, I'll come back center, and then yeah, just let me know if you want views on anything.
Yeah, could you zoom in on this branching, white branching?
Yeah. All right, video. Go for zoom.
Okay, coming in for zoom. Okay, I'm going to hold here.
Copy.
Ah, this looks like bamboo coral.
Oh yeah, that's definitely looks like a bamboo.
Looks like there's
Although we'll
something
get
on
a
the
better
right
image of it.
attached to it.
Yeah, copy.
There might be a skate egg case on it on the right side
Get
there.
rid of lasers
Oh,
from
that's
the
right.
front.
Yeah, could be a skate or some sort of cat shark potentially. Haven't seen any evidence of fishes.
Yeah, and Stacy, there's also something kind of funky behind this bamboo coral on the slope. I can't tell if it's a really thick crinoid or some kind of coral
Anywhere
or what.
particular on this wash
Is
lead?
it-
Like,
It has yellow tissue at the base. I would suspect that this is going to be in the S1 clade.
S1.
Zoom in more.
Ah, okay.
Related perhaps to claderite.
Sure, yeah, just
For the polyps.
Thank you.
That's max zoom.
copy.
You know, I was just looking through my notes from the Anegada passage, and I have this exact same coral at about this exact same depth as well. So it was sampled once from the Anegada passage at 525 meters. So that's, you know, not too far from where we are now, and same yellow base, same branching pattern. So I also noted S1 clade. So that's good that we're in agreement there.
Pilot, can you possibly zoom on the crustacean on the bamboo coral?
Good copy, Bridge. Thank you.
Zooming out.
Copy that.
To look for crustacean. There we go.
Pilot, I'm going to turn my head just to see along the ridge, if that's
Yeah.
okay.
Yeah, go for it. All right, video.
Okay.
Do you want zoom back in on crustacean?
Oh, okay, sorry.
Sorry, that was my mistake. Should have specified. Is this what you want, watch lead?
Yes, please, pilot.
Yep.
Oh yeah. It doesn't look occupied, the egg case at least, but chirostylid is definitely, it looks like one we've seen before on some of the other black coral colonies and Caligorgia colonies we've come across.
Sorry.
You're
Coming
okay?
back
Do you want
out.
me to come out? Okay.
Go like that. Are you good with
There you
that,
go, pilot.
watch lead?
Yes, thank you.
Copy. Oop, kicked up some dust.
Excellent.
So uphill is probably the port.
So did you guys
Looks
see
like
that
there's a skate there to your left.
that sort of bluish thing that's behind the slope?
Ah,
Behind the-
right
back?
underneath the bamboo?
No, it's on the left of the screen now. Just the top of the slope. It was behind
Yeah.
that boom back
Yeah.
coral we were looking
Oh,
at.
to my right?
Yeah, so pilot, can we zoom just above that coral, kind of on the crest where the slope flattens out, there's a blue structure.
Yeah, video go partial.
Coming partial.
I think this is a another holopus. One of the crinoids we've been seeing through the dive.
Okay, great. I hadn't seen that before. Thanks.
Yeah. Yeah, we got some good imagery of it, much deeper, opening and closing.
Video's clear.
All right. Watch these. Do you want anything more of this?
Let's see. No, we're good here.
All right.
To the left or right? Where was the other coral? Can we look to the right? I think we looked left already. It's good to know too, we're seeing multiple of these black coral colonies. They're also branching fairly irregularly. This one just to the top left of the coral we just looked at. Pilot, we'll look at this fan directly below us here.
Copy that.
It's good to know that those black corals though are branching fairly irregularly, and the one to the upper left of the bamboo coral had a branch coming right off the midsection. And this one looks like an octocoral we're looking at right here ahead of us.
Yeah.
Would you like me to come out a little bit more? Oh, something.
Sorry, there's a
fish.
There's a fish.
I think it's gone. All right, let's refocus on the fan.
Okay.
This is a rather large coral fan for this family, possibly in the genus Chrysogorgia.
Yeah, this is a tough one, Steve. It's I agree. It's definitely in that family, Chrysogorgiidae, but
I have more pilot.
Color, the structure makes me think of something like Metallogorgia or Pseudochrysogorgia.
Right.
The shape definitely more Chrysogorgia-like.
Yep. I can see at least two associates as well. One smaller looking shrimp and one large shrimp inside. Any way we can get the back side of the colony to see how it's attached to the rock, pilot?
Yeah, video good, come wide.
Okay, coming wide. That's full wide, pilot.
Great, thank you.
I've also noted in the area there are some smaller colonies of Stichopathes. It looks like a different species than what we were seeing down deeper, but Stichopathes, black coral whips.
All right, video zoom in on that.
Okay, coming in.
Let's see if that gives it the base to us.
It looks like it might. There it goes.
So the skeleton looks rather dark on the branch tips and at the base, and it does look lighter in the middle.
Oh, sorry.
I'm sinking too. I'll come back.
Okay. Should I zoom out, pilot?
Yeah, that's all right. Bounce back. Yeah, go ahead, come partial video.
Okay. That's partial.
Copy. Well, actually, do you want a better image of the base, or is that good?
I think that's enough. Okay. Yeah, I think we'll work with that.
All right.
Coming out, pilot.
Great.
Okay, that's full wide.
We just wanted to make sure it wasn't attached to a longer stalk. It looked to be firmly cemented perpendicular to the rock underneath.
Copy that.
There's some really brittle stars that have really long arms. This is something new to this expedition.
Yep. There's a few here, brittle stars.
Go ahead, Bridge.
Feather stars.
All right, watch leads. At this point, do you want me to stay on the wall, or do you want me to
you would
get
like
on
to
top
move 10
of
degrees
it?
to port. Let me just check with the pilots, and I'll give you a quick call back. Hey, pilots, if we're going to be here for a second, the bridge would like to move 10 degrees to port.
Where do you think is more likely that we can...
yeah,
I mean,
I don't think we're moving.
we can always... we can always keep looking at corals around the edges here. They're likely to occur, but if we're looking for fishes, do you think it's better to be on top or on the ledge?
Just call down when
I would
you're
say,
set
can
and
we
ready.
be on top, close, well, maybe between the ledge and the top? Like,
yeah, just kind of ride the corner.
yeah, right on the, so maybe if we do see some fish in the distance, but yeah, maybe you could identify what's on the wall.
Yeah.
A happy medium.
From the craft there.
So we can see, yeah, so we can see down the wall, but see fishes in the distance.
I'm not sure, maybe. Do we try to move it?
No, there's not much.
Video is switching out pilot.
Copy. Thanks, video.
We'll see what Roland says.
We did glare yesterday, and I don't think it was that bad, but in like the top right corner, what eventually solved it was bringing the lower starboard almost all the way stowed.
You see fishes?
Okay.
No, I think there's another ledge.
Reduced our light
You
spread,
see?
But we had extra light, so we were okay.
Video, go for quick zoom. Is that?
Nope.
Oh. Oh, here's a shark.
Is that
shark?
Could that potentially be the fish that laid the egg? Or, you know, that species of
Video,
fish?
I'm going to sneak up to it.
Lasers are clear.
Copy.
Okay.
Is that another?
I got him.
Great, thank you.
I think it might actually just be a dead or dying chrysogorgiid coral, similar to the one we had just imaged, but it doesn't look very healthy. So we'll document this fish and then we'll move along the ledge. I think that's the consensus with our scientists on shore.
Copy that.
Sharks are fish.
I'm going to attempt to swing to get its side video, but it might move.
Okay, this is a partial at the moment.
Yeah,
So
if do a quick zoom, and then we'll come out.
So
it's
this,
still there.
yeah, this could be a cat shark.
Oh, sorry, video. You could do a quick full zoom in case it flies as I try to get a little closer.
This much movement is not usable in the video.
All right, when you come out then?
Okay.
Andrea Quattrini, one of our shoreside scientists, says it's in the genus Scylorinus, if I'm pronouncing that correctly.
Did you already postulate that is this who's leaving those egg cases around?
Yeah, possibly. Yeah, we did mention that. It's definitely a process that we see a lot in the Gulf of Mexico. Cat sharks laying egg cases on corals, and it's we've also seen egg cases like that on other expeditions in this area.
Good copy,
video
Bruce.
when you come on in.
Little tilt down.
Got it.
Pilots and watch leads, just to let you know, we have about 24 minutes left in the dive.
Thank you, Nav. Okay, I think, let's see, any particular part of the shark that is best to get close-ups, or just generally? I think we've got as good as we can get for now.
All right, if you're happy with that science, just let me know and I'll move on.
Yeah, I think we can move on.
All right, video, if you're happy, good, come on.
So I'm thinking
could we get lasers on that too before we go?
You got it. Sorry, got it.
Thinking we could stretch out a little further this way, which should be slightly downhill.
Yeah.
And then we could turn past back what we already saw and then try to go uphill to the top.
I was going to say, it looks like there's still a significant amount of height left to go.
Yeah, on my port.
Sounds good. Ride this edge.
So how tall is this? It's like 3 meters.
I can put my butt out, maybe. $3 meters?
Yeah. Nice.
Can we look at the star to the left-hand side?
Yeah.
I'm noticing some smaller colonies of that Chrysogorgiid that we imaged coming up the hill here. We're going to grab a quick shot of this sea star, since it's one we haven't seen.
Our video could come partial.
And I think this might also be one of the same sea stars we saw just about an hour ago or so.
Tamaria.
Tamaria, yeah.
All right, video.
Well, it's always worth a second look because it looked quite pale last time. This one looks a little bit beefier.
Put it center.
Possibly having lost some appendages. Some of the tips of the arms look recently regrown. Okay, pilots, we're not going to dwell too long on the star. We want to take a look at the fish.
Copy that. Video whenever you're happy.
Do you get a look at the thing on the left? I couldn't tell if it was a coral or an anemone.
Should we turn back for the peak now?
I think. Sorry, pilot. Can we go back in on to the left of the star?
Yeah.
Just for a quick zoom.
Go back on in, please. We go just to the left.
Looks to me like could be an anemone, although can't really see past it to see if there's any skeleton underneath. So I think that'll have to suffice for now.
All right. Our video, if you're happy, come on out.
Yeah, I think it's an anemone.
Watch lead. At this point, we could potentially keep following the ledge, but we also have a little bit of slope left.
Okay.
Do you want us to go towards the slope?
Yeah, let's hit that slope.
Great.
I had thought that this was the summit here, but we definitely want to reach the pinnacle.
Yeah, might be picking up one last bump.
Maybe you can look up past it for fish.
Hello, Chris Ma.
So I appreciate your taking the time to stop. The beast you're looking at is in the same family, but is in the genus Lynchia. This is Lynchia bouvieri, and it's one of the more commonly encountered deep-sea ophidiastrids. It usually has large, warty bumps on it, and not much is known about it. I think it's been seen before, but always happy to have another sighting. I also wanted to point out something kind of neat. You were looking at earlier at a lot of the soft crinoids, the Indoxocrinus, and they have kind of shortened tails.
Right.
And one of the things I'm noticing is that you also have a lot of sideroid sea urchins in the proximity. And the thing is, sideroid urchins feed on those. They do so by presumably a very slow chase, wherein they hunt down and devour that sort of the stalk or the tail. So that also might explain why you're seeing so many stalked crinoids with short sort of distal ends, because the sea urchins are probably chasing after and eating them.
Yeah. Very cool. Very interesting.
Yeah.
And there might have actually been an urchin perched precariously on one of the sponges, I think. The one that was next to the shark. I was doing something else at the time. But that might have I don't know that they've ever been seen feeding on sponges. It might not have been predation, but
got a little loop here left.
But if you see one again perched on something like that, maybe there's something else going on. They're just feeding on everything. Hard to say. But thanks again for stopping, and I know you have a short time with the rest of the dive, but enjoy the rest.
Thank you.
Thank you.
Have a good day. Bye-bye.
Bye-bye. Yeah, we did see a number of these sideroids feeding on a range of different things. Yesterday, we saw one very clearly feeding on sargassum, phytodetritus. Today, they seem to be on sponges, but we've also seen them on deep water corals, chomping down on the polyps.
A lot of times on the AUV and ROV work on the West Coast, I've seen them a lot on sponges. So I wonder if they are. There's a sea star right ahead.
I see it.
On the lasers, yeah.
Yeah.
Just a quick snap soon.
I have video snap zoom it.
This one looks like a goniasterid.
Is that all you want, Science?
Yeah, that's adequate. Thank you.
All right. Video, come on out.
We want to make sure we really get to the top of this mound, so that we can check that box.
So we are right now at 466 meters. Looks like a dark sea urchin to the bottom left.
Okay, pilots around
maybe.
Oh, there's something more substantial up here.
00 on the UTC clock. I will have you begin to start preparing to ascend. We need to be off the bottom by 20:05.
Sounds good. Well, looks like this is the top now.
Yep. For those of you who have stuck with us through the whole dive, we've reached the top.
So that's what we thought last time.
Purple blob.
Go ahead, Bridge.
I was a little confused.
So I actually do think this is the top, right? I guess we'll check the sonar when we get there.
Yeah, I'll peek over.
Good copy, thank you.
Is there a substantial current in this area?
Pilots, did you overhear that?
No, I wouldn't say there's much difference from current from earlier. It's pretty calm.
Okay.
You kind of see particles moving, but they're slow.
All right, so you still have uphill to your port about 20 meters out, right?
Yeah.
If you look in your sonar.
It looks down a little there. Yeah, watch it. It looks like there's still a little slope left. Do you want me to, 10 minutes left, do you want me to hang out here or keep following the slope?
Could we maybe keep on?
Yeah, we can definitely do that.
Thanks.
Yep.
So there was, we're pretty much done with that last move. Are we looking to start heading to the west now?
Yeah, like, maybe like 305?
305?
Yeah, that's the way your sonar says.
We're looking for 20.
Oh wait, but I am.
Where are you looking though?
See a fish?
Oh.
Fish.
There's a fish.
Yeah,
swimming.
I'm looking 305. That sounds.
Or is
good.
that a siphonophore?
Oh no, a siphonophore.
It's only.
See the slope there.
Yeah, it's only 12 meters ahead of you. I think that's probably the top. So.
Yeah, so like 300.
Yeah.
290 between.
Yeah, 300, like 20 meters. 15 or 20.
It's a number of these black corals
Maybe
we've been
15.
observing that have been branching somewhat irregularly.
Okay, 15 meters at 300 degrees.
A lot of the colonies seems to branch at least once
Sean, you can plant a
off the
little
main
flag on the
axis.
top.
Some of them branch
I forgot
twice
to pack the
closer to
flags.
the top of the colony.
Add it to the checklist.
I'm not sure if we made a positive ID on those, but we got some really great images. It could be either parentopathies or some relative. Yesterday's dive, we were around 270 meters depth, so we're about 200 meters deeper than yesterday. The substrate and the attached fauna look a little bit different, but we haven't seen any snappers or groupers just yet.
Bridge, this is RV Nav. Looks like that move's coming to a completion here shortly. Could we put in another one? Excellent. We're looking to go 15 meters at 300 degrees, speed 0.2 knots, please.
How's that little loop doing?
I think it'll
Good
come
copy.
out. I'll
Thank
just
you.
keep an eye on it when you stretch out. Copy. I should start thinking about that.
The top of this mound is still yielding more and more terraces. Kind of like a wedding cake.
What is that shiny object just right of center?
Right there?
Yeah.
Yeah, go for it.
Some of these stock crinoids. And a couple looks like small cup corals. Are those cup corals or anemones?
Yeah, several of them look like cup corals to me.
Okay, video's clear.
Alright, you come wide then. Alright. Oh,
ah,
fish.
there's a fish. Ah.
Success.
There's an octopus below you also.
Oh. Alright, go for fish first.
That's a snapper, right?
Laser's clear.
You can go ahead and get him for me, co-pilot. Yeah. Thank you very much.
I'm not. No. Any of the fish experts on land? Is this a queen snapper? I've only seen them dead.
Side video.
Its lips look sort of queen-like.
Yeah.
Well, if this was the only queen snapper or snapper we see, I think that we can consider that mission accomplished.
Yeah.
Shall we check out the purple blob?
Science, are you happy with this, or?
Can we hold it another 30 seconds or so?
Yeah.
Maybe in the last 3 minutes you can go and peek over the...
Do you have any more zoom out of curiosity? Oh, there we go.
There you go. Up. It is. Can we zoom a little bit more, if it's possible, to the mouth?
I can get closer and then resume. Would that work?
Yeah, if that's possible.
Video come partial for me.
Good copy, Bridge. Thank you. Pilot says that last move has been completed. I don't know if we have enough
video
time
go
for
here
another one.
in case it swims.
Yeah, he has enough tether to get right on top there. So.
Copy that.
Going in video.
And just to clarify, you all have 3 more minutes before we need to start lining up to ascend.
Max in.
One of our shore side scientists believes that this might be Atlantic rubyfish.
All right, thanks, pilot.
All right.
We'll take, I guess we have time for one more look off into the distance across the top of this.
Yeah, copy that.
Let's see if that was an octopus or no.
I think we have to skip it, so our video. We're down to our last 2 minutes. And we gotta get at the tippy top.
460 meters or so is the top of this knoll.
I'm going to start stowing the lowers.
Yeah, copy that.
In terms of attached fauna at the top of this, we're looking at black corals, relatively small colonies of black corals,
Zoom, is it
occasional,
nope, never mind.
Occasional stalked crinoids as well.
Thank you.
The sediment is mostly soft, consolidated from what it appears.
Yeah, watch it. I'd say with sonar and what we're seeing, this I think would be the tippy top.
Okay.
Okay.
For today's dive.
So nothing off in the distance. Not really obvious that there are large schools of any sorts of fishes here at the moment.
Upper's okay, co-pilot, to stow?
Yeah, go for it. Just seeing if I see any last minute fish.
Yeah, there's some small crevices in the carbonate, but I don't see any fishes from here. Well, just in our last 60 seconds or so, we'll recap what we have done today on this dive for everyone who's watching at home.
Great.
We started out at around 772 meters.
Set up 'cause I just
we
have
climbed
to port.
over a steep section of seafloor where we observed a number of attached organisms.
All right, do we back up a little bit?
Throughout the dive, everything was more or less
come up a little, just to make sure the tether's blown.
Sedimented, but there were occasional spots where rocky substrate occurred, and we have some samples of stalastrid corals from that area. We were able to also sample in some of the flatter and sedimented areas the ranunculid sea pen. And
if we push right underneath it, you can
a
use the PTZ.
clip of
rotate about it.
what we've tentatively identified as Calagorgia
back up a little more,
octocoral
we'll shift it
with a brittle star
right underneath your stomach
from about 550 meters.
That sounds good.
All right, Washades, that's about the dive there. We're going to start to set up and get off bottom.
Excellent. Thank you, guys.
Thank you.
So we've just reached this promontory at depth around 460 meters or so, almost exactly, about 5 meters difference from what our multi-beam bathymetry was showing, which is pretty good.
Okay, I can see it in the PTZ.
And we were trying to document deep water fishes, of which we saw, I guess, I don't know if the ruby fish classifies as deep water.
But yeah, I would imagine.
Yep. So I guess we did find one fish in the top of this knoll. And yeah, I would call this a rather successful dive. We documented many invertebrates, several fishes, especially down deep. We saw, I would say, a higher diversity of fishes in the 600 to 750 meter range. moving shallower. Not as many fishes, but diversity of invertebrate life, including sponges, deep water corals.
A lot of echinoderms.
Yes, definitely dominated by sea stars.
It's in my spotlights now because it's behind me. So
and feather stars, brittle stars as well. For the science party, who's on the line, we will be starting our post-dive call and immediately followed by the dive planning for tomorrow's dive, which will be at the Buck Island National Park.
Because I should be able to see it in my
and we will let everyone know about what we planned for that dive. Our dive track has changed from what we had planned yesterday, but we'll be sure to update you on how that goes. So in about, let's say, 10 minutes, we'll reconvene here for our post-dive talk, and we'll go from there. So thanks to everyone at home for joining.
Yeah, don't push
and
tight,
hope to see you back tomorrow.
We can see it.
Thank you all.
Copy.
Come up first for Delta's 11.
Still coming up.
Both turns are 0. That's good. There you are. Okay, Delta's negative now. Maybe hold there. I'll come up a couple meters.
Can't quite see its
camera. Oh, is that it in your camera? No, I think
no, that's closer to me.
I think it came out.
It's like in the tail's in the way.
Maybe just very gently push ahead. And Nav, will you zoom in on high pack for our position? Excellent. Yeah, so if it's there, I guess it'll rise up soon.
Looks good.